Okay. Welcome, everybody. Some of you have had to travel quite a long way to get here. So welcome to Sydney if you're not from around here. It's great to be hosting this meeting. We haven't had one for, well, in person for a long time. I can't remember exactly when. 5 years. Yeah, we had a virtual one during COVID times, but this is our first one since that period. Anyway, welcome. If you need to use Wi-Fi, here are the two options. You can use EduRoam. If you know what that is, I think you would know how to use it. Otherwise, we have UNSW guests, which you can use as well. And by the way, we're live streaming on YouTube now. And if you prefer not to have your talk live streamed and uploaded, just let us know. But otherwise, we'll assume that you do. Okay. First, I just want to do acknowledgement of country. So I would like to show my respects and acknowledgement of the Benedix people, who are the traditional custodians of the land of elders past and present on which this meeting takes place. And if you're new to Australia again, in case you don't know what this means, I invite you to just reach out to an Australian during the break and talk to them about it. What I've learned from my New Zealand friends and colleagues is it's okay to talk about this. Okay. So just some general housekeeping first. If you need to charge your electronics, there's some power cords or power strips up here in the first row. Just please welcome to the front row. Everybody's favorite spot. So there's going to be a break. There's going to be snacks at that break. And then we'll serve you lunch as well during the lunch break. Luckily, it's a really nice day. So what we recommend is just kind of go outside, look around campus a little bit, grab some lunch at the table. There's seating everywhere. If you go up the steps, there's some seating. Go down the steps, some seating. If you turn to the right on your way out of the retreat theater, there's seating there too. There's a lot of students from UNSW here. They can help you out as well. And so if you are a speaker, just log into Zoom before your talk, or you can also just transfer your presentation to this desktop computer if you prefer, which is a little bit easier. Whatever, but we can do both, whatever you leave a little bit of time for questions, and we'll give you a 5-minute warning, just to make sure things move smoothly. But anyway, in case you don't know who the ACA is, we're the Australian Centre for Astrobiology. And we have members all over different schools in the university. So we have people from chemistry, geology, physics and astronomy, and biochemistry. And what we do, well, first of all, our foremost mission is to support and give opportunities for collaborative research for original life and astrobiology related endeavors, as well as hosting international seminars and meetings like we're doing today. We also have a strong cohort that's interested in education and outreach. And we also provide guided tours of important geological sites, thanks to our former director, Martin Van Kranendonk. And we're part of a larger community in the international world, and I think we have quite a few, or about half of these different Origin Life Astrobiology Institutes represented here at this meeting. Okay, so today we have 3 sessions. So the first session is going to be prebiotic chemistry, origins and metabolism. Then we'll have space exploration and technology for session 2, followed by lunch break. And then we'll do the final session, which is outreach, science communication outreach. And if you want the full program, you can scan this or just go to the AAM website. You can see it there. We also have programs in the front or schedules in the front desk. Okay, so today's a little bit packed. Tomorrow it's going to be lighter. We'll have poster session. We'll have our keynote speaker. It'll be a lot of fun. I'm excited. Oh yeah, and for those of you that signed up for the dinner, we'll have dinner at 6:30. So don't forget about that. And last but not least, thanks to the organizing committee. This took a lot of work to put together. So especially Ann Wang and Martin and all these people you see here. If you see them around, give them a pat on the back for doing a good job because they really did a lot of work. All right. Thanks a lot. So we'll move right along into session 1. And our first speaker is going to be Jim Cleaves. So Jim, if you'd like to come up here.
Are the microphones or just, yeah?
So there's a mic right here, and then there's a mic for Zoom land.
Okay.
YouTube right there.
Okay. So it'll pick up the general.
Yeah. It should be good enough for, you can ask questions and the mic will pick it up.
Yes.
We just need to get rid of socialization. Steven, put it on the second screen. It is the same. Never use the, you know, maybe the black thing. No. This one. Where you just want to.
Yeah, that's.
Okay.
And can I get a reminder?
I can do the other screen.
So good morning, everyone. I'm Jim Cleaves. I've come from Washington, D.C. I'll try to tell you a little bit about some work we've been doing over the last couple of years about the analysis of complex organic mixtures and what they might have to do with the origins of life. Thank you. The first time was in Australia. In fact, first time south of the equator, so I'm very excited to be here. And I'm looking forward to meeting everybody here. Now,
Oh, now it's not working.
it's not working on the keyboard.
Say what?
Double adding just to the button on the top right. There you go.
There we go. Okay. Thank you. Thank you. All right. So these are the kind of big general orienting questions that we try to get at. This is idea of a heterotropic hypothesis that somehow in our environment, organic compounds were supplied, those somehow self-assembled by various processes and gave rise to living systems. Right? And we have some notions of how organic synthesis happens in early solar system environments. We have a lot of open questions about what the nature of those organics are. This is ongoing research. This is what we're trying to understand. What are these things? How are they made in the environment? How do they interact with the environment? How do they self-assemble? And how do they self-assemble living systems? And we'll talk a little bit about what the ambiguity of what a living system actually is in a moment. And then, you know, we want to understand the entire life cycle of organic compounds in the environment. How do they decompose and how do we lose the signal that organic material is functionally living in an environment? Right? And so this has some importance for the search for life on planets and Mars and so on. Okay. And so we want to understand how can there be diagnostics for living or non-living processes. And so before getting into that, I want to talk about the concept of chemical space, which is the general notion of how many different types of chemicals might exist, right? And that comes kind of the question of classification. How do you say one thing is different from another thing? But importantly, in organic chemistry, we have the concept of isomerism. We have multiple levels of chemical classification by formula. What are the ratios of chemical elements? How are those elements actually bonded to each other? What's the nature of the single bonding environment, and so on. So we can start to talk about more concrete concepts of more specific types of chemical spaces. So, for example, we want to talk about drug-like organic molecules, those that are of a certain size, that are of a certain type of solubility, don't tend to aggregate in solution, and so on. It's been estimated that that's very astronomically large number of something like 10 to the 33 to 10 to the 180 unique organic compounds in that space. For reference, the Chemical Abstract Service, which is kind of a catalog of all synthesized and characterized organic compounds, is maybe on the order of a few tens, maybe 100 million compounds. In contrast, what we've been able to detect from natural sources is maybe under a few scores, maybe 100,000 organic compounds. But when we look at abiotic organic syntheses, things like carbonaceous meteorites, we can already tell roughly, we can estimate that these might contain the order of 100,000 to 10 million unique organic molecule types. And that might be an undercount, right? Depending on the system. But in contrast, biology, the core metabolism of the biochemical system is maybe only about 500 monomer types. Right, so it's amino acids, nucleotides, deoxynucleotides, cofactors, specific lipids, and so on, that are winnowed down into a very useful space by biology. Right, it's the selection of this recyclable biochemical space. So the possible space is very large. What biology uses to create organic, biological diversity is very small. Right, and so we think of that as being a hallmark of natural selection. And so just to give you some concept, we've done some work trying to enumerate some of these small molecule chemical spaces. We have the number of isomers presented for various libraries we've generated in silico of all possible organic isomers. We have the number of isomers on the left, on the y-axis, and the mass of the molecule on the x-axis. And what I want to point out to you is that the top ticker on these things is on the order of a million isomers for a unique formula compound. That's still on the order of 150 atomic mass. Right? So that's something on the size order of the amino acid tryptophan. It's not a very large molecule, but there's an awful lot of redundancy, or potential redundancy, in a unique molecular formula. And so why I think that becomes important is when we're exploring abiological organic chemistry using high resolution mass spectrometry, we're able to get very precise molecular formulas, but we don't really have a concept of the isomerism that might represent. Right. And as I'll try to point out here, we want to have a way when we're exploring these natural systems where we have very complex organic mixtures of winnowing down the product space so we can actually identify what these molecules are, so we can understand how they might potentially serve as diagnostics of biological or biological processes. And we'll try to show how we can apply some reaction network metrics. So backing that up, I want to bring into mind, like I said, we don't have a complete definition of what life is, but I'll use some kind of broader concepts to try to get at, I think, what the periphery of the problem is. And there's this idea of autopoiesis that Martin Van Kranendonk came up with, maybe an elaboration of some earlier philosophical concepts. But it's the idea that you can define a system, a special type of system, which is able to recreate itself over time, right? And that's a unique class of system. And so to give you an example of what makes it special, you can think of an autopoietic system as being something like a car factory that can make car factories, right? As opposed to a car factory that can make cars. Right? A car factory that make car factories is an autopoietic system. The other is an allopoietic system. So it's a self-contained system, and that certainly seems to be true of biology. The second conceptual kind of model I like to bring into thinking about what other systems is Tibor Ganti's schematon. And Tibor Ganti, as opposed to getting into the specifics of what is a cell made out of, said that a chematon, a self-reproducing chemical system, is one that has some sort of system to define a boundary, so you can say what is and what is not part of the system, and maybe contain other parts of it. It has machinery for reproducing the entire contents of the system, and it has a blueprint or an informational system for specifying all the other components. Right, so it's completely designating and reproducing its via internally stored information. But what I think is especially significant about Ganti's concept is that it's so conceptualized that it doesn't depend on what it's made of. All right, you could make, in principle, this kind of system out of almost any kind of chemistry that works. The informational blueprint doesn't have to be DNA. boundary level doesn't have to be liquid, machinery doesn't have to be frozen. Right? Any chemical system that potential could be integrated to carry out all of those functions would be, in a sense, good. All right. So I'll try to show you some of the practical things we've been, trying to add to this discussion. That, as I said, this chemical space is potentially very large. There's a lot of things that would be really impractical to do in vitro at this point. There's just too many compounds and too many experiments, too many interactions that have to be explored. But we can actually use computational chemistry to answer some, or try to get a handle on where the interesting questions are in some of these problems. And so I think of this as a kind of hierarchical level of things that you might be able to use computational methods to help answer. So things like I showed you first over here, hey, what is the total potential number of chemical graphs, chemical species that could exist? How are they related by chemical reactions potentially? How is one able to be transformed into another by a viable chemical transformation? How does what we can see here? Is there, once you've started developing a chemical reaction network, are there avenues for feedback catalysis, or feedback inhibition in that network? And can you detect that computationally before you have to look for it experimentally? Can you predict the self-assembly of new phases in chemical reaction networks? Could you predict the development of energy transducing systems in these kind of systems? And ultimately, you'd like to understand, could you understand the origin of coding in chemical systems, right? How do things transfer information from one chemical level to another to have some sort of systemic meaning? Now, I'm not saying we've answered all of those questions, but I think these are things you can apply computational chemistry to. All right, so here we have some, a schematic of possible ways we think organic compounds might get to primitive planetary environments. We have things like atmospheric chemistry taking place via electrical discharges, ultraviolet light, so on. The input of extraterrestrial materials from meteorites and comets, possible geosynthesis from hydrothermal fluids, interfacial chemistry that takes place, from dissolved compounds interacting with light and minerals, combinations of these various processes. The point being that it could be a rather heterogeneous milieu that depends on inputs from things, which all may be characteristically different depending on their histories. And so to give you an example of the kinds of chemical reactions we're interested in looking at, purity experiment, and it's special in the sense that you start from a very small number of components, input components, and once you pass an electric discharge through the simple gases, methane, ammonia, hydrogen, water, you start developing radical species that recombine. These get converted into radicals which recombine again, and you start getting this combinatorially complex reaction network that takes a simple colorless, gas and water solution and turns it into a mixture of hundreds of thousands of unique organic compounds. Okay? Not all chemical reactions do that. Some do, some don't. The ones that do are the ones that we find. So just to give an example of the historical development of this kind of analysis, this is an example. That is Miller's actual original paper chromatography analysis of the products of his original experiment. So he basically separated, added compounds to a piece of paper, separated two solid systems, sprayed it with hydrogen, and was able to detect a series of amino acids. And you would perceive these as being kind of the major products of this system. But there's only a few spots, right? There aren't thousands of spots. But we went back and looked at some of these original extracts almost 50 years later, using more state-of-the-art instrumentation. And we're able to show that there were scores more amino acid products in these that would not have been detected by the technology of the 1950s. That's not a surprise, but it changes your perception of how complex these reactions are. And in fact, more recently, we've taken these same things and just looked at them by electric spray mass spectrometry, electric spray ionization spectrometry, and you can see easily that in fact, if you don't limit yourself to amine-containing compounds, there are again probably hundreds of thousands of unique organic compounds, with each one of these vertical lines representing the unique mass organic species, right? Or a unique mass molecular formula, which could be multiple organic isomers. And we see this phenomenon over and over in natural biochemistry, just the laboratory simulations of it. Here's an ICRMS spectrum of organics extracted from the Murchison meteorite. Again, you can appreciate the forested peaks. This is a spectrum of the Titan lab simulant. Again, same phenomenon. There's just almost a peak at every potential mass that can be possible. So this is a general phenomenon. There are these certain classes of chemical reactions that just generate enormous combinatorial diversity. And so the question is, are there other kinds of chemicals present in these mixtures besides things like amino acids and nucleic acids that might have interesting self-assembly chemistry that haven't been considered, previously, because we're trying to go directly from abiotic chemistry to modern biochemical systems or models. And so amino acids, in fact, in the Miller experiment, are thought to be largely derived from the Strecker amino acid synthesis, which starts from an aldehyde and a ketone, and via the addition of cyanide and ammonia to an amino acid, which is shown on the lower graph here. If you have a very low concentration of ammonia, you like to include cyanide, will produce alpha hydroxy acids, the cognate ones, shown on the upper mechanism. And in fact, if you go into a Miller experiment and you specifically look for these species, which become absorbed by the fragmentation spectrum, we find that there's a large abundance of alpha hydroxy acids. In fact, there's more alpha hydroxy acids produced than amino acids, depending on the reaction system. And so we're interested in this. Well, can you make some kind of polymer out of these, right? And of course, we've known this for some time, that glycolic acid and lactic acid, the two simplest hydroxy acids, are actually very easy to polymerize in acidic water. The free energy change for forming an ester bond and acid is pretty low. And you can push these into really long polymers fairly low temperatures. So shown at the bottom are just the electric fray chromatogram, and shown at the top are the fragmentograms of individual masses. And what you can see, if you can easily interpret mass spectra, is that each of these is in fact just a homopolymer, right, of different lines. And that's a very easy experiment to do with a normal laboratory mass spectrometer. But if you want to do more complex types of polymers with more monomer types, you need to use a high resolution mass spectrometer. And so we started taking our samples down to Tallahassee, Florida. There's the United States National High Magnetic Field Laboratory. It's down there with extremely high field mass spectrometers. There's extremely good resolution. And in fact, we tried an experiment of taking a mixed 5-monomer system with more amino acid type hydroxy acids and just created dynamic or combinatorial libraries of these. And in fact, you end up with a system starting with only 5 hydroxy acid types that contains tens of thousands or hundreds of thousands of unique oligomer species, right? So these are sequence-defined polyhydroxy acids, right? With millions of sequences of hydrogen. We calculated at the time it might have been the largest combinatorial library made to date. Okay, so what became also interesting about these is when you dehydrate these systems, they spontaneously form little globule systems, and we could easily predict using chemoinformatics which combinations of monomers would produce globules and which ones would produce completely soluble polymers. Okay, I'm not blitzkrieging you to that, but that's the point, that there's a lot of chemistry to explore in here. The next thing I want to talk to you about is trying to understand complex reaction mixtures using computational techniques. So reaction modeling systems. And so we during the pandemic, a really great team of undergraduates assembled to Blue Marble Space Institute of Science. One of them is here, Sid Sharma. And we decided to try to build a pipeline for analyzing these complexly generated reaction networks. And we used as a model, just to build a pipeline, the caramelization of glucose. You could use sucrose is probably the caramelization you're more familiar with, right? You take sugar, you heat it up, it turns into this sticky brown stuff. Feels kind of slippery on your tongue. It has interesting smells and flavors, right? You put on your creme brulee and all these things. It's actually a really complex mixture of chemicals generated by the hemolysis of common sugars. So it turns out it's very easy to model reactions in silico because organic molecules are graphs that are easily handled by computers. Right? And graphs in the sense that an atom is a node and a bond is a vertex. There's an edge, I'm sorry. So it's easy to represent organic molecules using matrix representations, and it's easy to represent organic transformations using transformations in matrices. And so what that essentially does is you can essentialize and represent as graphs any given input organic molecules. You create a library of organic transformations. These are allowed reactions. So these are the kind of things that we take in organic chemistry, the so-called named reactions. You have to use a little bit of intuition in this case as to what you experientially know will occur under the reaction conditions that you're studying. But as an example of how this works, you have an input graph that is broken up into an intermediate graph and creates an output reaction graph. And as an example here, this is an example of a benzoic acid rearrangement applied to cyclohexane dione and water. You have a reaction rule which can interpret that these two graphs can output this compound. Right. You're able to keep track of which atoms are moving to which atoms. And this, I should say, this is software written by Jacob Anderson at the University of Southern Denmark. Okay, so as I said, it's a pipeline. So now you can input any set of small reactants. You want to just start with glucose and water. That's a fine starting point. You can add your library of rules. The software will interpret what is the first set of products that gets generated from the input reactants. That's going to take that products and put them back in the hopper and now apply all of the rules to all of those molecules and iteratively do that, right? So each time you're ending up with a more complex pool of things that can build a more complex reaction network. And that's just here. Now, at the end of the day, we have an output graph of the network, and each of those molecular products can be interpreted using chemoinformatics software to see the kinds of properties the reaction products have now. And I'll just show you the kind of output that we get. So this is a representation of the reaction network of caramelization glucose, colored by the generation at which each species occurs. So that yellow spot in the middle is glucose itself. The green spheres are the first set of products that are generated. And as you can see, this whole network contains some tens of thousands of ultimate products after only 5 rounds of this iterative reaction development. As I say, now we have a table of all these products. We can compute things like their masses. We can compute things like their hydrophobicities, their solubility in water, and so forth. And we can look at how those properties develop as a function of the generation or the progression of the network. And so here's an example. We can predict what we think the partition coefficients, the alkaline water partition coefficients of the species ought to be as a function of generation, with the darker ones being more hydrophobic. And in fact, if you take caramelized glucose and you look at it under a microscope, it does spontaneously assemble into little phase separated globules. So there are, you want from a species that is completely soluble for generation, rather than soluble. Then we can go back just a bit at not measuring nonsense, right? whether there's a correspondence between an actual high resolution mass spectrum of this reaction and what the network model is predicting. And we see that there's a pretty good correspondence over the range that the computer can generate products and that the machine can measure compounds, right? So there are cutoffs in both directions. So we see good matches with even isotopic peaks in simple early phase of the mass spectrum. But we can look at higher order analyses of the mass spectral data using things like campus mass defect plots. And we see that the model, here's the open circles, predicts the general trend of the experimental data. Black circles in pretty good, pretty well. All right. What we see is actually a pretty good overlap in this, especially in the range with the two models, with measurement and modeling overlap. And noting that where there's a complete correspondence, that can still represent multiple isomers of the same level. So it's not just that there's a 1-to-1 match, it could be a 20-to-20 match. Again, one of the things we think is really nice about this is when we compare this to the total possible number of molecular types per formula for mass. In fact, this, if you say that the thing you're detecting has to be generated by a reaction that's possible, you may be compressing the search space for small molecules by a factor of about 1, which is it's nice if you're an analytical chemist. If you want to know what something is, you don't just start randomly grabbing stuff off the shelf. You can say, well, it can't be this, because there is a mechanism to get it. So that's it. I want to bring this in on time as best as I can. This is what we want to try to understand is how do you develop new types of chemical phenomena from complex reaction networks? And how can we best use this kind of understanding as a light detection tool that biological stuff that gets melted might also look rather complex in the geological record. And we're trying to refine these workflows to make them better, more technically sound. With that, I'll just thank you all and the organizers for having me. And thanks for listening.
All right, we have a few minutes for questions. Yeah.
So Lee Cronin has this molecular assembly theory, which he's saying as a way of distinguishing when you look at a complex picture of whether it's biological or non-biological in terms of its generation. What are your thoughts on that?
I don't think that's exactly related. I think it is related to this, but not exactly in the way that. Okay, so the concept is that something like a protein, which has a very defined sequence, right? When you analyze a mixture that contains that protein and you get a mass spectral peak and you fragment that, you will see that it fragments into a discrete spectrum. Whereas if you take a meteorite that has a species with exactly that formula and you fragment it, you will not get a fragmentation spectrum because it contains so many isomers of that same thing that you're getting 1 million very low level fragments.
Yeah, so he wants to put an organism on Mars and mass spec
Yeah.
experiment where he
Yeah.
collects this mass ring and then fragment. And he is saying that the number of fragments that you get out of say you isolate a mass 380 and fragment that. And it is also somewhere in that concept is embedded how many molecules there are.
That's the key. That's the key.
And I find this whole thing rather confusing myself. I don't know there's just because I don't find lots of stuff confusing. So
Well, no, I think that copy universe is really a key question. I mean, nobody's going to say that biological molecules aren't complex in some metric. Right? Some of them are, some of them aren't. But the fact that biology makes certain things at the expense of making other things, and then leaves a record of that behind, is what you want to measure. Right? But I mean, I think you would say alkanes are not particularly complex molecules, but biology on Earth is much harder than they are.
But
Yeah, Jim, it's exciting to see that complexity, we're making complexity is not difficult, right? They say it happens in few generations, measured in meteorites, all this. How do we simplify the biological system? like one of the extraordinary things in your opening slide is that biology only uses 500 compounds. How do you sieve all that complexity away?
Well, I think that's the key point is, I mean, this is just me speaking from what I feel. That at some point things start to have more control over the chemistry that they generate, and when they start developing catalysis and only making the things with fewer side products. More specifically, we have a very self-referential, self-controlled system, right? And that process is the emergence of life itself. Right? Does that make sense? So you have a system that has no control over where it's going, and then you have a system that has a very high degree of control over what it's doing. Right? That's a living system. That's a modeling system.
Somehow the energy reverses in that cycle.
How
I mean, in terms of splitting and differentiating and then recombining those together, there must be a point where those molecules recombine and so.
That's, yeah, so you're thinking about the concept of evolution by mutation, this kind of thing. Yeah, it's a good thing to wrap your head around conceptually. Maybe it's too complicated to talk about. We can talk about outside.
We have time for one more quick question. Faye, yeah.
Sorry, I just wanted to follow up on that and sort of asking the same thing probably in a different way. Starting with, 10 to the 33 molecules or how many possible, I mentioned with 500. And what kind of molecular gladiator game are we playing here? So you have to sort of have an arena. And how do you define that environment in which you can play that game consistently over time? Somehow over time, this continuous game will go back and forth. And at the end of the day, come up with 500 survivors. So I'm not sure by doing the current network prediction alone will get us there.
No, I don't think well.
Yeah, so
But it's useful for animals and systems at the moment. Yeah.
you need to have that analysis platform in order to ask the next question.
And I should say, the way we represent this here does not have any kinetic model, right? It's just a connectivity level. So you could model which things, right?
All right, I think we better move on to the next speaker. So let's thank Jane again. Next up, we have Laura Lowe, who's a third-year PhD student at UNSW with analytics group. Maybe we can actually. Just keep it up there. Yeah, this is.
Hi everyone, as Albert said, I am Lauren. I'm a PhD student here at UNSW, and today I'm going to tell you about some of the work I do on model protocells or model primitive cells. So I'm sure many of us here are trying to at least somewhat answer the question of how did life on Earth begin? And this is obviously a very, very big question not a single person can answer, but today I'm going to talk to you about a specific point in time where we started to see super simple primitive cells, perhaps composed of fatty acid bilayers, start to transition into primitive cells more akin to modern biology. So at this point in time, we've begun to see the emergence of phospholipids, some enzymes, but we haven't really seen the emergence of all the complex cellular machinery that exists in modern biology. Things like membrane transport mechanisms that can be used to transport nutrients and waste molecules into and out of the cell haven't really emerged at this point in time. So that leads us to the question of how the cell is actually able to transport these nutrients into and out of the cell, and how were they able to feed themselves, grow, and divide. So that's the question I'm trying to at least somewhat answer. I'm involved in a pretty large collaboration to build a propagating synthetic cell. And what I mean by that is an artificial cell that is able to feed itself, grow, and divide using the simplest components possible, in order to better understand how life is able to transition from chemistry to modern biology. So this synthetic cell is made up of a phospholipid bilayer membrane composed of POPC and POPG. So phospholipids are what our cell membranes are composed of. So we're using a similar system here. Obviously, it's a lot less complex than what our cell membranes are composed of. But within this phospholipid membrane, or this phospholipid vesicle, encapsulated inside, we have a system that can be used to synthesize fatty acids and eventually phospholipids. And these phospholipids can then be incorporated into the bilayer, allowing the membrane to actually grow, and then eventually leading to division. But one of the big problems with this system, at least at the moment, is that you can only encapsulate enough nutrients for this phospholipid synthesis pathway to actually go to achieve about 1 to 2% membrane growth. It's just not sustainable in order to get these synthetic cells to actually grow and divide. So that means we need some sort of external feedstock of nutrients that can actually permeate the bilayer. They need to be able to make their way from the outside of the synthetic cell to the inside of the synthetic cell. So that's the thing I'm trying to work on, and that's a lot of what my PhD work is about, is how do we get these nutrients to actually permeate the synthetic cell? And some of these nutrients of interest include things like amino acids, things like AMP and ATP, which can be used as energy feedstocks, and something like acetate, which we're hoping to use as a carbon source in order to begin that fatty acid and phospholipid synthesis. So to start off with, I needed some sort of technique that could actually be used to monitor solute or nutrient permeability, and the technique I used is known as a shrink swirl assay. Basically, that involves preparing vesicles encapsulating the fluorescent dye known as calcium, and then mixing the vesicles with your solute or nutrient of interest. What then happens next is water actually rushes out of the vesicle because of the difference in osmotic pressure. And then as water rushes out, the vesicle then shrinks up and shrivels, and we actually see a reduction in volume and then a reduction in the fluorescent signal. This is because the concentration of dye actually encapsulated inside the vesicle has increased because of a reduction in volume, and calcium is a self-quenching dye. So at higher concentrations, the fluorescent signal will actually decrease. Then over time, if our solute is actually permeable, it and water will then penetrate the vesicle, leading to it eventually swelling up again to roughly its original size. Now, because this is a technique that relies on fluorescence, this whole process can be monitored using some sort of fluorimeter or plate reader, and the data that we see is something like that plot on the bottom there, where we start with the fluorescent signal up high. With the addition of solute, the fluorescent signal drops down, and then if our solute is permeable, it will recover over time. And then because this is all related to a change in calcium concentration, it will be converted to a change in volume. So basically, we monitor the change in volume over time for all these vesicle samples. And the data I'll be showing you is just that late part of the curve there, because the initial shrinkage process actually happens really quickly. And the setup I have at the moment can't actually be used to monitor that, it just happens too fast. So to start off with, I thought I'd show you some examples of some permeable and impermeable solutes. So on the plot on the left, at the top, we have the control in 50 millimolar epis in the blue. That's pretty much just a flat line there. That's telling us that our volume isn't changing over time and just mixing our vesicles with the buffer that they were making here. So all these vesicles were made in 50 millimolar epis, which is above and hovers around physiological pH. Those two curves that we see underneath there, in purple and red, glucose and glycine, they have a characteristic shape telling us that the vesicles did shrink initially, but that volume recovered over time. So that's telling us that that solute did permeate those vesicles. These vesicles are permeable to both glucose and glycine, and they're slightly more permeable to glycine because that volume recovered more quickly. In the plot on the right, you can see that again we have the control on the top, the 50 millimolar heavies, but at the bottom we see a bunch of flat lines there. That's telling us that these solutes are not permeable across these phospholipid bilayer membranes because we don't see a recovery of that volume at all, at least over this time frame here. So that was kind of disappointing because, as I said earlier, some of these solutes would be useful nutrients for the synthetic cell, for things like the energy feedstocks that I spoke about earlier, or using them as a carbon source to synthesize fatty acids. But these results weren't actually particularly surprising. It's relatively well established that generally phospholipid membranes are less permeable than something like a fatty acid membrane. So we can see in this schematic here that something like glucose, it's further on the left here for the phospholipid membranes compared to the fatty acid membranes. It's telling us that it's less permeable across these bilayers. Something like sodium and potassium is a lot less permeable in phospholipid membranes, and then something like AMP and magnesium isn't even on the scale shown here for phospholipid membranes. So again, it wasn't particularly surprising that some of these solutes weren't particularly permeable. And this is telling us that maybe it's like potentially transition from something composed of a fatty acid membrane to something composed of a phospholipid membrane. There was a difference in permeability there. It was more difficult for a lot of these nutrients to actually make it across the bilayer membrane. But despite that, I wanted to explore what are the strategies that can be employed to actually make some of these solutes permeable, to make some of these nutrients of interest permeable. So today I'm going to talk you through two strategies that I've been working on. The first is through modifying the membrane composition. So as I've said, these vesicles that I'm working with are composed of an equal mixture of PVC and PVG. Both of these lipids are quite cylindrical in shape, so they actually pack together quite nicely to form lipid bilayers. But as we've discovered, they're not particularly permeable. Not a lot of these solute molecules want to cross the bilayer membrane. But what would happen if we were to, say, disrupt this packing a little bit by introducing a lipid that has a different shape? Something like lysophysi, which has a single carbon chain, but still has a relatively bulky head group. So it has a more conical shape. So when it gets inserted into the bilayer, it can lead to defects and pores forming in the membrane, potentially allowing some of our nutrients to actually pass through the bilayer. That was one lipid I wanted to explore. The other one was oleic acid, which is a fatty acid. It has a single carbon chain, a relatively small head group, and has very different properties to what phospholipids do. So I wanted to see if adding this fatty acid into the bilayer would potentially allow more nutrients to pass through the vesicle. So what I basically did was I worked with, again, the same base PVC and PVG system that added either 10% lysophysi or 10% glucose, 10% oleic acid to the system. And in the plot on the left here, we have one of our permeable solutes from before, glucose. We can see that in both the orange and the green, the addition of both of those lipids didn't appear to make too much of a difference to the membrane permeability. We can see that the green curve is shifted down slightly, but overall the shape of these curves is pretty similar. So that's telling us that the permeability of these membranes to glucose hasn't really changed. And we can see something similar on the right with one of our impermeable solutes from before, so glycine here. We still have three flat lines that's telling us that neither of these lipids actually changed the permeability. They didn't improve the permeability to glycine at all. And this was the case for a lot of different solutes I've studied. There was a much broader list than what I've shown here. None of the permeability of none of those solutes seems to change much, except for sodium acetate. So as we can see in this plot here, the addition of lysophysi didn't really make much of a difference. The addition of 10% oleic acid improves the permeability of sodium acetate quite significantly. We actually see that curve shape now. We can see that our vesicles are actually recovering, the volume swelling up as that sodium acetate is entering the vesicle, which was good news because, as I said, we wanted to use this as our carbon source for fatty acid and phospholipid synthesis. But again, overall, this is only one solute out of the many that I studied. So I wanted to keep exploring whether there are other strategies that would maybe be more successful in improving membrane permeability. So the next thing I wanted to try was through adding divalent cations to the system. It's quite well established that divalent cations affect the packing of lipid molecules within bilayers, so I wanted to explore if that would affect permeability. So the first thing I did was confirm whether or not these divalent cations did indeed affect packing, and I did that through a technique known as electrical field spectroscopy or EIS. Basically, that involves tethering a lipid bilayer membrane to a gold electrode and applying alternating current across the membrane. Now, this allows the impedance and thus the conductance and capacitance of this bilayer to be measured. Now, the conductance is proportional to the ion flux across the membrane, and the capacitance is inversely proportional to the membrane thickness. So this gives us an idea of how many ions are flowing across the membrane and whether or not our packing, or in this case, the membrane thickness, has changed. So what I did was I worked again with the POPG system, but I also but I also worked with plain POPC and the addition of the two other lipids, the oleic acid and the lysopC. What I did is I prepared all these bilayer membranes in a control buffer, and then I washed these bilayer membranes with our divalent cation of interest. In this case, it's either magnesium or calcium. In the plot on the left, which is showing conductance measurements, we can see that with the addition of magnesium or calcium, the conductance for all of these membranes actually decreased. So there was less ion flux across the membrane, and this was the case for all the membrane types tried. It was a lot more significant for the three on the right, I guess. These three here. These three systems all contain POPG, which is a negatively charged lipid. So it's likely that these three membrane types were a lot more affected by the addition of the positively charged divalent cations here compared to just the POPC system. But again, all these membrane types showed a decrease in conductance with the addition of magnesium and calcium, with it being more significant for calcium. In the plot on the right, we can see capacitance readings, and again, for the most part, we saw a decrease in capacitance for both magnesium and calcium. For all the membrane types tried, this decrease in capacitance is telling us that the bilayer is getting thicker. So the bilayer packing of those lipid molecules is changing, the membrane is getting thicker. So obviously, these divalent cations are having effects on membrane properties here. So from that, I went back to some of my permeability measurements, some of the shrink swirl experiments. Here, I'm showing the data that has the 10% oleic acid system. This is mixed with the sodium acetate that we looked at before. And I've also added magnesium chloride. So we can see just the plain sodium acetate in blue here. And then with the addition of magnesium chloride in the orange here, we can see that the curve increases at a sharper rate. So that's telling us that the permeability of this membrane to sodium acetate has improved with the addition of magnesium, which was good news. But again, I tried this for a lot of other systems as well, and it didn't really appear to make much of a difference. So it seems that sodium acetate with the 10% oleic acid is a unique case here. But I still want to further explore a lot of other possibilities, a lot of other nutrients, a lot of other divalent cations and lipid systems. But given that, so far I've been able to conclude that permeability can be somewhat modulated for specific solutes by changing the membrane composition, and it can be somewhat improved through the addition of divalent cations. But this only works in a few scenarios, and overall it's very difficult to improve solute permeability of key species. So this may suggest that protein channels needed to co-evolve with phospholipid synthesis in order to get those nutrients across the membrane, particularly if the evolution was evolving from something composed of a fatty acid membrane, which is a lot more permeable, to something containing a phospholipid membrane, which is a lot less permeable. And with that, I'd like to thank the entire Wayne group, especially Anna, for all the support. Thank you to my funding sources, and thank you everyone for listening.
All right, thank you, Lauren, for that great talk. We have a few minutes for questions. Yeah.
Thanks, Lauren. That was great. You started off using ester lipids.
Yeah. No.
There's also the option, isn't it, to use ether lipids, and Which does life prefer, the ester or the ether?
I mean,
You know, I don't.
I'm not super, I don't have a lot of knowledge about it, but it depends on the species, I guess, primarily. But the system that we're using, the PFBC and PFBG, we're actually trying to use a bacterial system that can be used to synthesize fatty acids. And that system relies on using basically those the PFBC and PFBG, particularly the PFBG, because it's negatively charged. So it works quite nicely with that. I don't know if it would still work by changing up the memory composition that much. Maybe we don't know enough about it.
Either it's been coordinate your ions, it will come this. So if you do that, you have to be careful about whether you are able to preserve any of your desired and structure.
Yeah.
So it's not as easy as, like,
So way back when, what would it have been?
I don't know. So
Wow. Good question.
so this cup, I think. Here's cup.
Thanks, Rob.
Yeah, you have this idea of permeability, right? And so you can get nutrients fluxing into the protocell easier. But you might want to, potentially think about how to trap those nutrients in there. It'd be a useful thing for a protocell to have, at least.
Yeah.
Have you thought about any strategies like that?
Not for this particular project, I guess. I guess one of the options is maybe like introducing further compartmentalization actually within the protocell itself. I don't think that's super necessary for this, but it's actually obviously something that would be super useful for a protocell.
Interesting, yeah. Yeah,
I have a sort of simple question about permeability.
Yeah.
When I first saw it in the slide, I saw it was in like 7 meters per second. And then in electromagnetism, and then in other things, measured maybe different units.
Yeah.
I kind of, finding what you're actually, what you're measuring exactly here is ions, like conductance, and then you have to convert that into permeability somehow.
So you can, for the second technique I spoke about, the electrical impedance spectroscopy measurements, that is an electrical measurement, so that is directly measuring the ions there. For the other permeability measurements I'm doing, it's what I'm actually measuring, I guess it's a change in volume. You can actually fit a model to those data to pull out the permeability coefficients, which would be in centimeters per second. But yes, there are different, I guess, varying ways of measuring permeability and different units for them.
time for one more quick question. Yeah.
I was reading about permeability organisms who can apparently maintain their membrane function. I was wondering if there's scope in your experiments to carry out experiments at more than one temperature or at 5 temperature.
Yeah.
That gives you some interesting information.
I think it potentially could. I personally haven't done any temperature dependent stuff yet. It's definitely a possibility that could be explored, and especially for like early primitive cells, especially in early Earth environments where those conditions are so extreme. Temperature would obviously play a pretty important part in what's going on in those systems. Yeah, so I definitely think there's potential to explore that.
All right. Well, let's thank Lauren again. So next up, we have Professor Anthony Poole from the University of Oxford.
Very good. Thank you. Okay, well, thanks to the opportunity to speak, and great to do some international travel. We've been stuck in our little islands in New Zealand for a while now, so we're coming up for the first time. So I'm a biologist, so this will sort of be the opposite way from what Jim Cleaves was describing. So I've got a different set of complexities to address, but I'll try and talk you through an experiment we've done trying to unravel origins of DNA using biological tools. So This is a schematic just showing how the four deoxyribonucleotides that are the building blocks of DNA are made. So you can see here these are RNA building blocks here, and there's an enzyme called ribonucleotide reductase, which is responsible for making all the DNA building blocks in pretty much every life form. This is how it's done. So this, of course, if you start with the RNA building block, you get an intermediate uracil rather than thymine, and sort of this extra pathway here. So one of my students, Apolline Dubois, is going to give a poster talking about this problem, and so I won't cover that more. But what we're interested in my lab is saying, well, if this is sort of a potential molecular fossil for how DNA building blocks are made, can we start removing those pathways? So Apolline's work is talking about removing this bit, and I'm going to talk about what happens if you take ribonucleotide reduction out of bacterial cell. So ribonucleotide reduction is a really kind of interesting thing, because on paper, for me as a simple biologist, it looks like a very simple reaction. You've got a 2' hydroxyl on the ribose ring, and that's reduced to a hydrogen. But if you look at how the ribonucleotide reductases do this reaction, it's fiendishly complex. So this is a simplified picture here. But what happens is that they have to generate a radical, and in some cases it's on a different protein from the one that does the reaction. That radical then has to arrive at the active site. So it's a cis-adenyl radical that's generated. And then there's a whole lot of steps required in order to ultimately reduce this hydrogen. So this is quite a complicated reaction for a protein-based enzyme to do. So not a simple reaction in terms of the enzyme chemistry. So I won't go into the details of this, but I will point out that in terms of what's known about RNA, this seems to be an unlikely thing to have happened in the RNA world. And so if you think about, well, RNA precedes DNA, could you have started making DNA building blocks using this reaction in the RNA world? So some of the things, like finding a cofactor that's central for generating this radical, yes, you could probably do that. You can even, in some of them, there is a cofactor cleavage step that's probably plausible for RNA. But the main problem is once you've got a radical generated, these radicals are not friends with RNA. It tends to result in cleavage. And in fact, the early ways in that structure of RNA involve use of radicals because they will cleave the backbone of RNA. So, in terms of then, for me as a biologist, what does that mean? Well, it looks like this sort of process of going from RNA to a modern system where you've got DNA and proteins probably happen in a couple of steps. You would have needed to have a decent ribosome and translation accurate enough to make something as sophisticated as a ribonucleotide reductase before you get to DNA. But not everybody sees it that way. There's another pathway that's very interesting that's being proposed among others by, so it was first discovered in the 1950s, but Steve Benner, who's in the audience, wrote a very interesting paper on pretty much life, the universe and everything in 1989, where he proposed that you could probably get to DNA in a much more simple pathway. So if we look at how modern cells do this, they take the RNA building blocks, they convert some fraction of those into DNA building blocks, and then they can make their genetic material from those. But there's also this recycling pathway, sort of salvage pathway, where blocks that are not used can be shunted back into central metabolism. And that reaction is reversible. So the argument that Steve made in this paper was that perhaps you could have had DNA before you had complex proteins because of this alternative reaction. So I started talking to some colleagues in Japan at the University of Kyoto at the Juno Gallus lab, and they're interested in this this alternative pathway, which is this pathway is called the deoxyribonucleotide pathway, and the reverse will run in synthesis. So here's a DNTP here, and here are your starting points: glyceraldehyde, true phosphate, and acetaldehyde. And there's a series of enzymes that if you take these out of the cell and just feed them these two starting substrates, they'll quite happily make the NTPs. And they're interested in that because, I don't know if you've all noticed, but we've been doing PCR tests and things like that up the wazoo the last 3 years. So there's been exponential increase in demand for DNTPs, but we mostly get them as a byproduct from fish waste, and there hasn't been an increase in fish anytime anyone's looked. So the idea of being able to produce these through synthetic pathways or bacterial fermentation, as my colleagues in Japan are trying to do, is kind of got some applied value as well as sort of the interest of our community. So this pathway definitely will go in the synthetic direction. The problem is that if you try and do this and just give cells these starting materials, it's not going to work, because while it's reversible, anyone who's had a hangover knows acetaldehyde doesn't, it's not a friend of biology. So acetaldehyde levels, the levels required to do this reaction are lethal to the average bacterial cell. So, but we thought, well, it's an interesting problem. I wonder whether can you make a cell use the DARA pathway for building block synthesis? So here's our picture of a modern cell. You're either making or recycling these two pathways. And what we're interested in is saying, okay, well, if we take away ribonucleotide reduction, can we find the conditions where that pathway would operate synthetically? Is that biologically plausible as well as chemically plausible? So what we did was we deleted all the ribonucleotide reductase genes from the bacterium E. coli, and that sounds like a straightforward thing to do, but all my colleagues in the ribonucleotide reductase field said, don't do that, Ant, you're mad. We've all done this, we've all tried and failed. But unfortunately, I had a very stubborn student, Malik Sivaeva, who just did it anyway, somehow. This is devil in the detail, which I won't go into, but basically this figure here is just showing our ribonucleotide reductase knockout line. So these are the operons for the 3 ribonucleotide reductases you get in E. coli, and then these are the shorter bands that you get once the regions have been completely deleted. So I'm happy to go into the background of how we got that to work with anyone who wants to know in detail. So the first thing we did was to characterize these lines and say, well, what happens to them once you've knocked them out? So what we did was we grew them in the presence of, in a minimal media, some MOPs with glucose, and then we gave them different concentrations of deoxyribonucleosides. And this is just dropping down from 1,000 to 50, 50, 10. And then if you give them 0 supplemented deoxyribonucleosides, the orange here is the mutant, and here in the sort of dark blue is a wild type. You can see the wild type is not followed at all because it can synthesize its own. But once you stop providing the ATPs, deoxyribonucleosides in the growth medium, then this line is inviolable. So that's sort of a summary. That's that pathway knocked out, and it's at the moment dependent on this externally supplied source of deoxyribonucleosides. Okay, so then we thought, well, we're biologists, we like evolution. Let's just do a brute force and see what happens if we evolve them in an evolution experiment. So we took a series of these little cell growth plates. The blue here is just a blank, and then we've got 2 concentrations. So the 1,000 microgram per mil and the 250. So the first 2 of those graphs from the previous slide. And this is control. We evolved these for 30 transfers, keeping the concentration the same. And then after that 30, we took the group that were evolving at a lower concentration and dropped them right down to 10 micrograms per mil, and then evolved those for 10 transfers. And that doesn't seem like a long time, but these are quite slow growing. So the experiment took a while. And then at the end of that, we sequenced all the lines using a Illumina sequencer just to see what sort of happened to those lines. Okay, so it turns out that, and this is sort of a a fun thing for anyone who does an experiment is this, what's going to happen. So you start off with an idea, and what actually happened was completely the opposite of what we were expecting. And perhaps in hindsight, we understand why, but not at the time. So 8 out of 10 of the lines have got the transfer 40 have a mutation in this gene here, DEOB. And if you look at the details of the mutation, this gene is absolutely being destroyed. So we've either got truncation mutation or active site mutations. So this is not functional. And we've done the experiment further than 40 lines, and by kind of get this going to 100 transfers, this gene is completely gone by then. So this is a key enzyme in this salvage pathway, the DARA pathway. So we're reducing the concentration of these over time. We don't have this, and this key step in that alternative pathway is taken up. So the DARA pathway is not compensating, at least under the conditions of our experiments. And we're like, wait, hold on, why would that get, this is your chance to make DNTPs through an alternative pathway, why would that happen? So it turns out if you look around, as a biologist, you're used to the thing that organisms have already done most of the experiments for you. So there are a small number of microbes that have lost ribonucleotide reduction, and there's a few nasty pathogens of humans, things like urea plasma, and then some endosymbionts of things like aphids that lack ribonucleotide reduction. And so they're reliant on their host to provide DNTPs for genome synthesis. And if you look at the genomes of the ones that have lost the ribonucleotide reductase, they've also done a knockout. So instead of so they're getting it from their host, and they don't have this, and they knock this out. So what we think is actually happening here is that you're trying to avoid the problem of when you don't have many of these, you don't want to get rid of them by putting them through central metabolism. So if you block that off, then you can reuse them. So sort of don't recycle, reuse kind of situation. So yeah, so that was the exact opposite of what we expected, but we saw some interesting things along the way that I'll kind of cover in more detail. So this, we could say we've not found the conditions for this, but we've been doing some other experiments where we're trying to increase the intracellular concentration of acid aldehyde. This doesn't stick around for very long. First around high 3-phosphate is an intermediate in glycolysis, but that's not such a problem. But making this intracellularly, this tends to be gotten rid of quite quickly. But one of the other things we did say, so that was sort of we got the biology that don't recycle. If you knock out ribonucleotide reductase. But we saw one other thing, which was a thing we're following up now. So we've got no ribonucleotide reductase, no DARA pathway, but we are seeing the introduction of ribonucleotides into the genome in these lines. So I'll just give you a little end on a little piece of data. So this is quite preliminary still. So we've got an E. coli control here, and this is LC-MS analysis of extracted genomes. So this is just a bog standard E. coli, and you can see its genome is made up of DNTPs. And if you take just the an RNA extraction, of course, you get 100% RNA. If we look at our knockout lines, so these RNRs here, this is at time 0, this is actually at time 100 in our experiment. There's 2 different strains that have gone for 100 at 2 different concentrations. You can see that both of them have a ribonucleotide fraction in the genome, so they seem to have accumulated some ribonucleotides. Not a lot, but certainly some. And if you look in the literature, there are other mutations you can do that will lead to ribonucleotides being incorporated into the genome. So we knocked out and did an evolution experiment on a couple of these. So these are RNases, and what these do is that they're part of a pathway called ribonucleotide excision repair. So normally you don't want ribonucleotides in your genome, so there's machinery that comes along and removes those. One of the reasons you get that is priming during DNA replication starts with an RNA primer that's made to kickstart the replication, and this guy here, RNase H1, is responsible for the removal of those primers. So, but we can sort of see that biology seems to be fighting this pretty hard. So if we roll up to 50 transfers, it ends up with being a lot better at dealing with the ribonucleotide that are being incorporated, despite the fact its main enzyme for removing them is being eliminated. So, so it's kind of interesting that maybe we're able to unpick some things and start to see what happens with ribonucleotide accumulation in the genome of these RNA knockouts. So then just to summarize, we've got these RNA lines we've knocked out. We predicted that maybe we could see the their pathway compensating, but in fact it was eliminated. So this a fun quote that always gets pulled out in these cases: the great tragedy of science, it's laying a beautiful hypothesis, finite in fact. That's of course a good thing about science, you always learn things. So we've learned something about endosymbionts and pathogens that we weren't originally trying to find out about. But it's still an open question as to whether their pathway can operate the DNTP synthesis in modern cells. So that's something we're continuing to look at. So with that, I'll stop and just thank the members of my group who were involved in the work. And thank you for listening.
All right, thank you, Anthony, for that great talk. Yeah.
Yeah, this is brilliant. So if you can put back the slide of pathway that you're trying to construct, and I got it. If I go to ELITE 83845, which you just published, I'll have it start. Keep going back a few more slides.
Yeah.
So your problem with this, you mentioned your thermodynamic analysis. I'm going to have to send that to make reference to something, but it's like
Not this pathway.
thermodynamic analysis. The problem is right, the outdoor reaction is the equilibrium constant is concentration dependent. And
Yeah.
you can't get passed out at any concentration. So that means the step that immediately follows that has got to be downhill.
Mhm.
Or at least you got to find some step that's thermodynamically downhill. So the mutase is not downhill. The nucleoside phosphorylase is presumably downhill. So
Mhm.
the point is that if you could find yourself a deoxyribonucleoside 5' phosphatase, right, you would make that first reaction go over the well. So is there any, but it has to be slightly like there's any general documentation for actual organisms. So does the world know about a deoxyribonucleoside nucleoside deoxyribose actually 5' phosphatase that point known to the world?
I don't know.
You see what I'm saying? That would drag
Yeah, yeah. So yeah, I mean, one of the things my colleagues in Japan are playing around with a lot is, you know, that they mostly do industrial microbiology, so they're interested in finding enzymes that are going to be useful. So they go looking in all kinds of weird places. So they found already a more acid aldehyde resistant DARA enzyme, for example. Yeah, but that if anyone's going to find something, it'll be them. I don't know them, but that's a very good suggestion. Yeah, so part of the challenge there is that we want to get rid of the other routes that acid aldehyde would take if it's generated. So a lot of alcohol dehydrogenases, for example, in chloride, you might need to knock out.
But I think that none of those steps going all the way to deoxyribonucleoside are downhill. So even the phosphorylase content.
Yeah, I mean, it seems to be pretty happy to go in the synthetic direction on the right conditions. So it's just, we don't have those to tell them. That's the real question, whether that can happen.
We have time for one more quick question. Yeah.
I'm talking about like what the purpose of the salvage pathway, and the question would be more, you're looking at 0 again because it looked like there wasn't much activity at 10 and then a bit more as we went up. Do you think there might be an optimized low concentration where you wouldn't be activated there? Do you think there's any concentration dependent on the enzyme?
Or when we get there getting inactivated. So it pretty much happened when we dropped to 1,000 was pretty high, and then 250 micrograms per ml was we didn't really see mutation of the DARA for that, so it was only when we dropped it down to 10. So that's still a 25 times change. So we haven't looked at where when it gets stressful enough that you start seeing those mutations, but certainly in that second part of our experiment that, yeah, and that second part, the last 10 transfers, that's when we saw the mutations in there.
All right, thank you very much. Let's thank Anthony again. Our last speaker for this session is Professor Matt Benedix from the UNICEF. You just got to get him switched on through his laptop. Yeah.
Good, thank you. Well, thanks everyone. It's great to be here. For a couple of reasons. First of all, Australasian Society. I only noticed last night, and I'm a proud dual citizen, and I've always hated it in New Zealand when people mix up Australia and Australasian. So I made that mistake myself. Second of all, it's the only venue in the world, really, where I speak, where I'm working on something relatively contemporary. So with the case of motility, we're very happy to have DNA, RNA, proteins, membranes, and a whole bunch of things that you guys work on earlier than the origin of motility, which itself is still quite a long time ago. For the German motor, that's not correct. It's not. You may well know, because it's kind of one of these poster boys of intelligent design and other creationist fallacies, but it's an amazing molecular machine that self-assembles, rotates up to 1,000 hertz, 5 times faster than a Formula One, changes direction in milliseconds, and assembles this propeller, the filament, which can be like 10 microns. So it can do quite a lot of things in the self-assembly and biotechnology space that is far beyond what we do. In terms of man-made nanofab. This came from a long time ago. You can kind of tell we're still in the blobology era of cryo-EM. Everything just looks like a brick. And this upside down paddle pop is the stator. That's kind of the engine of the motor. Ions pass through that and they drive the rotor around. Fast forward to 2020, and oh no, you can't see that with the zoom. It doesn't matter too much.
Oh, you can hide it. Or
I used to command option control H, I think.
Yeah.
Never remember it. Anyway, yeah, so the reason why I have this is, turns out that upside down paddle pop is itself aware. So Monty Bodkin and the Wodehouse models, that was because there were wheels within wheels. Who would have thought, spending 50 years studying a rotating nanomachine, and it's powered by a rotating nanomachine. This was done simultaneously by Susan Lee's lab and Nicholas Taylor's lab. I don't know if I've got them right around, but I think I did. And it's a pretty incredible structure. So it was difficult to solve, it's difficult to purify. It's basically reliant on improvements in single particle cryo-EM. And yes, you have this rotating nanomachine that's either on the inside track or the outside track. It always rotates the same way, and it changes the sort of cogs or the side of the cogs that it's engaging in to change the rotation. I think from structure now, no one's seen it go round and round, but we've got pretty good evidence that it does because of its asymmetry and just the structural resolution. A lot of the superheroes in the field are trying exactly to do that, to purify just one engine and measure it rotating, much like ATPA was done before. But we know quite a lot about this way, top down, how ions come in and go. We still don't really know exactly how the ions sort of go through there. So this is the whole thing. It's been a raft of cryo-EM like in every other field, but in particular, this has always been one of the original heroes of cryo-EM, especially tomography, because it sticks out of the cell, it's easy to find, a large filament, track it down to the base and get pretty awesome atomic level resolution in the whole complex now. Most of what I'm going to talk about today is the engine, the stator, and I'll talk a little bit at the end about the propeller, the filament. Cool. So, going more simple, one thing that I'm going to come back to is I'm just talking about the engine like it powers the ions. I've been a bit vague about it. Well, we have most of them are proton powered, and not that E. coli is representative of most of them, it's representative of most of the published Magellan work for obvious reasons. But we know that some of the marine ones, Vibrio in particular, Vibrio alginolyticus is our model sodium one, that's actually powered by sodium. So we know that these two forms exist, and we also can engineer them to swap parts and change some of that. So the title of this talk was a bit, bold. It was something like Ask Our Ancestors what's the origin of motility. And I'm not really going to talk much about how that works because it's pretty much unnoticeable. We're working on specific parts of that problem because we want to make it a little bit more tractable. So the first thing was to learn or to teach myself ancestral reconstruction. Zapata Trujillo, you probably know, she's a superhero, if we can use that word, in the astrobiology community. And then one of her reviews explaining the process. Many of you probably know this. You take a sequence alignment, generate a good phylogeny, that's probably the hardest part. Then you run some standard ancestral sequence methods, they're getting better and better. Then you do some cloning, and in the Zapa's case, you then interact that with some biogeochemical interpretation. That's possibly the more astrobiology facing thing. When you've got something that interacted with some planetary signature, you can maybe date it and make some claim in the astrobiology space. But nobody had done this on the flagellar motor. And so we had a go very conservatively at first. We took 757 watt Vs. And there's already things if you're an expert in ASI, you're like, geez, that sounds like quite a lot of watt Vs there, Matt. And you'd be right. And people like Zapata and another one back in our days, George Hockberg, they're more snipers in the field, One shot, 2 kills, they build a perfect phylogeny, and they make one ASR directly before and after some transition to learn something. We're a bit more uncivilized. We just blast in a shotgun at the phylogeny and try to scatter a bunch of hits and then work out something about structure and function. So this being the first time we did it, we were very conservative. We know this Zapata is highly conserved. If you knock it out, the stator doesn't work. So we made sure it was there. And we're particularly interested in this FAD-YAD motif because that seemed to be roughly correlated with sodium proton selectivity. In any case, at this stage, we're being conservative mostly because we want things that work. If I shoot the shotgun and nothing works, then that's a lot of money down the drain, and it's not a lot of insight. And we were lucky that, in fact, it worked. So this was done by Imtiaz for his PhD, which he just got confirmed last week. So well done, Imtiaz. And Angela Levin did a lot of the bioinformatics. So this is a classic swim plate. It's unreasonably large on this projector at the moment, to be honest. But what it means is when you inoculate in the center, it spreads radially if it's able to swim because it eats food, gets hungry, and then because it can swim, and more relevantly because it can execute chemotaxis, i.e. it can navigate to where life is better for it, then it will spread radially as it consumes the local nutrient. So this swim plate here, you can see all of them are swimming, and we've got some control. So every vector, no status, no swimming. What AB is wild type swimming, and pomade pot B. I haven't really talked about that, but that's our sodium engineered cone. So that's what we're looking at. But so first of all of them swung, and all of them swung even in the presence of this known sodium inhibitor phenol. So you can see here now our pomade pot B sodium controls doesn't swim, it certainly should, and all the other ones swim. So that's kind of some evidence that it's sodium independent. You could say proton power, but it's very hard to conclusively prove that because it's very hard to remove the protons. But you certainly suspect that. And we also did some other work, which I'm not going to necessarily take you through now, but we actually sit the cells down, and we see them go around and around, and we wash in and out different buffers, and then you can be very exhaustive in reducing that amount of cells. That's MOT B. Then we thought we'll give MOT A a go, given that MOT A is a much larger and more complicated protein. So we did this with 2 phylogenies, not for any good reason, but probably just because of usual scientific disorganization and the timeline over which you do this. This would have been the first one I did, very rough and ready, and this was a much better treat we were doing with better methods. Other things ancestral reconstruction people probably wouldn't do is try and reconstruct nodes so close to the root. But we're again just trying our shotgun methods. We also got a lot better at sort of evaluating the quality of an ASR. There are some standard ways you'd say, what's your certainty across every residue? Obviously, if this was really low, then we'd be able, we're not very confident in that. But actually, the residues that are around 50% are usually pretty interesting. It might be the intersection of, say, this clade with that clade. The residues that are 50% in that reconstruction, they might represent something that's quite important in the divergence of those clades. So typically, we would look at those more closely and maybe order both of them to track those. But again, we're kind of taking this from a structure and function model. So what we do is, in the case of MOT A, we were lucky to have 4 that work. Again, if none work, that would not be that useful because things not working is generally not helpful. You don't know if the protein's folded, you don't know if it's even there, right? But if it's working, i.e. it's swimming, then it's definitely being expressed and it's functioning. So we're looking at the difference between the ones that worked and wild type, which works, and the ones that didn't work. We're able to sort of map these onto our structure, and we can say, look, what parts of MOT A are important? So much of that was already known, but it's nice to see that it correlates, right? We can use ancestral reconstruction to look at what's conserved, and it makes sense. But what was less known was some of these residues that are lipid interacting around the bottom of the motor. This for those of you who don't look, same as every day, it's like top to bottom. You have the inner membrane here, there's an extra bit that binds up here, and your ions are kind of flowing through this. So that's what I say when I say that the in-out part of the ion binding is kind of known, but there's not a lot of hydrophobic residues going through here, and if it's rotating as well, it's not quite like an ion channel, right? It's maybe a clock tube. There's still, that's probably the most exciting stuff. What we show with is ASR here to understand some of the conserved lipid facing and conserved rope facing residues. Okay, so this is getting a bit far away from origins, but we have been able to use ASR to generate functional states. So it's plausible that we could do better jobs and start to answer a question that we care a lot about, which came first, the proton or the sodium ion? I guess we care about it because I've worked on the states for so long, but it's not just us. This is a question that is of broader interest in evolutionary biology. And I'm looking at Jamie et al. In 2008 had this evolutionary primacy of sodium energetics. So as far as I can tell in that paper, the logic is it more likely that sodium energetics arose first by looking at how easy it is to say establish a sodium gradient or permeate sodium ion steel membrane? So second author on this is I think Galpern. He was very excited when I first showed him some of our work because he thought we might have some experimental system that argued it was easier to evolve either towards or away from sodium selection. Now, this is so my point is of interest suddenly to the energetics field. Like what is more likely and what controls selectivity and why would you get there? Why do we care about it? Originally, it was an industrial question. It was like, well, if we could make it sodium powered, we could slow it all down because we can more easily control sodium than protons. We can't remove protons from an aqueous environment, but we can remove sodium. So this question really has to be answered with better phylogenies and ASR, but it's a very hard question because we can't root the tree very easily. We don't have a very suitable outgroup for the status that can split that. We're working on that, and that requires us to get better and better at phylogenetics. But what we can do in the meantime is challenge this system experimentally. So let's say, how did it happen? What happened? How did it happen billions of years ago? What happens if I take a sodium engineered strain and I try to manipulate it in another direction? So that's ultimately the question. Can we evolve changes in ion selectivity? So this is our experimental setup. It's inspired by the Surgic's method where he did the first one of this, where there was no ions involved. He just took one of those swim plates I showed you, took the thing from the edge, re-inoculated it on another swim plate, and he did that 7 or 8 times, just kind of like this. Then it looks like it makes bigger swim lanes. That sounds kind of self-evident, but I don't think it necessarily is. A lot of those experiments, like Michael Bain's megaplates experiments, they look beautiful in the video, but there's a lot going on under the hood to make something so clean like that. They had, for example, in the Michael Bain experiments, they had kind of extra antibiotics. And if you did something as simple as this, just serially iterating a swim plate, you may well promote biofilm formation or something different. When we tried to do it in, say, temperature models or in tanks or in pipes, that's frequently what happens. Anyway, Victor did it. Not only did it make bigger swim rings, when he sequenced it, it was mutations on the motor and in the filament. In this case, upregulation of filament length. We here did it by changing the environment and looking at what happens if we took a sodium instrument and we propagated one line in sodium, and we propagated another line in potassium. When we saw these flares, which is kind of evidence that the mutation occurred and it's able to do something new, we would then propagate those further and further and then sequence. So I mentioned that we had this too, but I haven't really told you how we made this sodium chimera. So the sodium chimera was made by our Japanese collaborators many years ago, and it was known that if you took the A unit from Vibrio, POM A, and you made a chimeric B unit, where you cut together part of the proton B unit and part of the sodium B unit, and you call that pot B, so POM A, pot B, you had something that was sodium powered. We did a slightly different way of doing this because we wanted to have the genomic context as clean as possible. And it turns out also it takes a bit of time, which I suppose is frustrating, but it did mean we actually made this by no-scar methods as well as by lambda-red methods, and then we could compare those two methods as well. Now, chimeras in the evolutionary community, I think gets a bit, sometimes a bit, you get some pushback. They're like, well, you guys aren't even doing anything that really existed now. You're making Frankenstein and seeing what happens. And that's true. But I still think that this gives us hope because we know here we've only changed actually like a few transmembrane messages. The pot B is really mostly just this transmembrane section of the B, and the A is entirely from the sodium. So we've got some hope. It shouldn't take too many mutations or too many changes to get us towards the reversion of that activity. This is what it looks like in terms of day by day. we might see, I can barely see over that side of the projector, to be honest, but we might see that there's a flare and that gets propagated and continually propagated until on the final day, we've got things that are very clearly swimming pretty happily in sodium. And at the same time, we've got things that have learned to swim a bit better on sodium that have never seen the potassium. We recapitulated it. So each time we freeze them and then we put them back on the swim plate so we can put kind of our giant phylogeny on one plate. I guess that's nice, and it also gives us some evidence it's hereditary, right? Because when the freezer came back and it's still there, it's like probably a genomic change rather than some expression change. And then we represent all that in this orange and blue graphic here, where we've taken our granddaddy strain, RP1237, that's a wild type proton swimmer. We've got no star editing taking us to POTS, that's what we call our sodium granddad. So that, I guess, it's the great granddaddy that fell into the nuclear reactor and became POTS, right? Whatever. So here, this is our granddad. And this one, over time, gets propagated in sodium and also gets propagated in potassium. And orange represents it swimming in sodium, and blue represents it swimming in potassium. So it's not swimming in potassium until there's two things I really want to highlight, but this is the main one. POTB-G20V mutation arises here, and then you can see there's now a blue circle, so it is now motile in the absence of sodium. Right? And there's also this one here where it's motile a little bit in the absence of sodium, but it doesn't seem to have had any genomic changes. So maybe that's one of those hypothesis, tragedy, fact, terrible fact situations. But we also see other mutations that correlate with the things of Victor source and some mutations sometimes in the rotor. And interestingly, some mutations in some iron marks, which you might expect as well. I mentioned that we can do our sort of buffer exchange. So this is what it looks like spinning round and round and round. It's like you grab the dog by the tail and swung it around your head, but a little bit safer than that. And we can measure the speed, and we can also change the viscosity and measure the torque. And to be honest, if you really care about this type of stuff, there's many different ways you do this with smaller and smaller beads, down to even 80 nanometer gold, to get really high speed, high almost step resolution measurements. But the overall purpose of this is POTS, our sodium powered granddaddy, doesn't swim when you remove sodium, stays not swimming when you restore sodium, but add that sodium drug phenol, and when you wash it out back to sodium, it starts swimming again. Whereas G20V, whilst it's a bit of a mess, it's clearly different to POTS, and some of those are pretty happy without any sodium at all. We then also restored it back on a plasmid just to confirm that G20V mutation is doing exactly that. Now, if you're a real doubter, and many people are, because it seems unlikely that a single point residue would allow it to function again on sodium. You'd be like, well, there's surely some sodium there, you guys just don't know what you're doing, which is actually fairly valid. So we use a kind of absorption microscopy to confirm that. And I think this is interesting, especially for microbiologists, is how much sodium is there just in the immediate. And there's quite a bit actually, right? So on those plates, we couldn't get lower than about 15 millimolar. So that's because in the LB, there's quite a lot of sodium rendered. But in our buffer exchange, which is why it's so important to do that, we can get pretty low, right? Because you can wash ourselves over and over again in buffer. And we got down to as low as 0.02 millimolar for those tethered cell swimming experiments. But this is interesting because maybe if we could have got less sodium originally, we wouldn't have actually seen the effect that I've been talking about. Because maybe you could only incentivize, maybe a selection experiment only worked well because we actually had from 85 millimolar sodium down to 15 millimolar sodium. If you totally removed it, it might be too harsh and it might not be it. So there's a general lesson in experimental evolution there about the selection, the beautiful selection that you think you create is not necessarily always the same. It's not the same from the bacterial point. So if you care about biophysics, okay, question could be that's stuff pretty high, like how many single ions are in like 0.02 millimolar, that's a lot, right? But it's a question of Benedix at that point. You've actually got more sodium outside the cell than inside. So most of the energy comes from the membrane voltage, but at that point, your sodium is actually, you're having to use energy to pump sodium into the cell. So at 30 millivolts, you basically, if you're in the Fletcher field or otherwise, it's not enough energy to drive it. But this is the energetic profile, so speed versus power tethered cells of sodium, and it's linear, so like 0 sodium is the same as 80 sodium. For POTS, 0 sodium is 0, and it goes up in a sort of classic exponential curve, and this is what our mutant looks like. So obviously at 0, this one's not going, but at 0, this one is going. And the point I'm getting there is, okay, this one is going, sure, that's cool, but it's not like it's just being bolted back to this plateau sodium independent state. It's more as if there's a tweak in the energetics enabled by G20V that allows us to get into this space where low sodium or zero sodium is akin to low sodium.
Except over 10 minutes.
I had 10 minutes remaining. Fine.
Yeah, practically.
I'll get to you in 5 minutes.
Yeah.
What about this tragedy? So tragedy is like opportunity, right? Like maybe genomics is not as important as we're taking on. So Gretchen, the postdoc in this we wanted to find something beautiful that would be for some reason there's an upregulation of the transcript level that's not fixed on the genome. So at that point in time, L5.3 might be some sort of prefixed L183F. Like L183F enables twinning better, and at this point it exists in the transcript that's not fixed on the genome. Didn't seem very likely, but it was worth giving a hoot because I've never done it before and I thought there was more information to get out of this. But indeed, we didn't see any change really across the data. That's what these plots showed that in granddaddy, or you can say granddaddy, grandma, grandparent, parent, and child. Here, there's no mutation, no evidence that any transcripts are like sort of either showing us a G20B, a pre-G20B or a pre-L1. But we also get differential expression information out. And this would be awesome to do with more infinite money and more time, because if you start to ask what systems are involved in the adaptation, what systems are upregulated and downregulated when you're stressing selectivity in order to influence mutation of the genome. We only did this anecdotally because we had the two lineages, and in each case we saw a different pathway really activate. We saw some common elements, but then we generally saw some differences. So it'd be nice to do that more times, I think, enough to get some certainty. And it kind of makes sense around things you think like chemotaxis genes, like they're obviously upregulated. You also have amino acid transport, metabolism, and then on this side there's more about the nucleotide salvage. So I haven't really looked at this in more detail because some of this things don't know anything about, but we saw some commonality and I guess more separation. I think you at least need to do a third lineage or a fourth or a fifth lineage to say, hey, there's only X ways this can happen. Isn't that interesting analysis of some of these pathways? So in summary of the motor section here, we saw spontaneous mutations quickly, and I guess the good thing we did, whole genome sequencing showed that there were status mutated first and then additional things. We identified a point for G20 here, and you can see more about the paper. So in the last few minutes, I just talked about sort of where we're going. So I talked about the propeller, and I guess the propeller is an obvious one because the submarine needs two things: an engine and a propeller. And the propeller is quite attractive because the engine, it looks like it doesn't take much to break it. So you don't have a partially working engine. Whereas a propeller, you can actually make a pretty shitty propeller. But if you can grind it around, you'll still get some propulsion. So you'll still have a benefit versus having no propeller. So we think that there's a more gradiated space. And structurally, there's some interesting questions in there because there's 4 domains in E. coli for the genome, but some of these actually seem to disappear on other species. So that in itself is a question of what's essential. If you can have a filament propeller without those 2 whole domains, why do you need it? And this is even more complicated if you have sheep proteins, and there's some filaments that have multiple other proteins. So why would be the core question. And a new postdoc in the lab, Jacob Scary, he's at the back. If you want to talk to him at the break. He's been doing phylogenetics in this, so it's a chance for us to sort of, I guess, iterate towards better phylogenies for a start. And it's looking quite attractive at this time, because first of all, we've got way better outgroup of the hook. So the hook is the bit before the whole filament, and it sits nicely as an outgroup, and it also can separate some of those clades. We don't have one like that in this data. We don't have a tree state. Okay? And we're seeing clades across species in the tree of life that makes sense, as well as seeing coupling of the groups that have those 2 domains or not. Right? So the firmicutes, by and large, don't have those extra domains. So we can start to map environmental elements onto that, as well as generate ancestral reconstruction. We're doing that. We haven't got one that works yet. We haven't tried too many. I suppose that notion of like a busted propeller is better than a busted engine may be false, because you still need to export that propeller through a tiny hole. Right? So conceivably, if you mess that up, it won't work. But we do know some pretty crazy things work. People have deleted filaments and engineered polyols, so you just don't, you tell it not to stop making the hook. So it just keeps making a really long hook, and those don't work out. So you can make a pretty bad filament. It's like a polymerized hook protein, and it's still working. The last thing I was going to touch on, if you want to talk to me more, because I know maybe you're in the chemistry and droplet space. So you go, I guess this is why motility is too contemporary for the purists in the crowd. If you just want to go back to droplets, we do some droplet work. I won't talk about too much, but we're mostly interested in DNA nanotechnology to manipulate droplets. So we did some work on how cholesterol manipulates bilayers, or better, how best to use cholesterol if you want to manipulate a bilayer with DNA nano. And this is in collaboration with Shelley Wickham. She's famous for making these barrels. In one paper, she's painted yellow and called them a minion, which actually looks like a minion, to be fair. That's pretty cool. And we've got 4 inside that we're trying to release. So the cholesterol question is how do you like bind the barrel? You want it to bind, but you also want to be able to release, etc. And surprisingly, a lot that hasn't done. Too long, don't read. This is the best way to attach cholesterol. And you don't actually want as many as possible because previously people just put 30 on. No point really putting 30 on because it causes aggregation and means less gets to the target. The DNA is perhaps less interesting, but the droplets is what I bring to this collaboration. And we've got experience in making droplet bilayers in different ways. Old school sort of non-ergonomic backbreakers with micropipettes or electronic pipettes and nano injectors. And we want to do this because we want assays where we can confirm that our DNA core is actually a core, and pass it through a core, maybe electric current, gauge current, transfer contents, etc. And so Alex Mason, who is a more bona fide chemist than me, he was doing it the backbreaking way here with the droplets. This is an effort to make the Australia Open logo. And it's just like this. This is like, I haven't got a scale back here, unfortunately, but it's using the micropipette to arrange droplets in oil, which create bilayers. But now we have his robot that does it, which is only about $300, and it's a Creality Ender printer. And it's like the printer prints itself, right? And the first thing you print is the thing to hold the injector, and then you print the thing to hold the plates, and then you program it to not be a printer anymore. So I'll run his trailer. I don't know if the audio is going to blast everyone. I didn't share audio, it's a bit loud. I think you got trigger happy with the Adobe effects there, but anyway, glove probably first time he's worn gloves in the lab, I reckon, this year. No, didn't say that on the record, but that's probably true. And then the plate comes in, and anyway, the point is, it looks like what it does, but the point is that's programmable now. So you can go one droplet, go back and get your other thing. So we've got a pretty good system that's low cost of making 384 wells of 2 droplet systems, so with a bilayer separated of multiple composition. I think in the experimental evolution community, this has benefit. If you wanted to sample a wide library, say you have a reservoir with a very heterogeneous population of DNA, you can make a pairwise droplet and interrogate a large population of membrane proteins in that system. The reason why I'm mentioning it is because I'm looking for a killer application for this. We've kind of done this part, but I think that in many of your fields, or you might have ideas, there could be some application which is like, hey, I really like to do something that's amenable to other plate reader measurements. So please come talk to us because we're lacking that killer application at the moment. Thank you, everyone. People involved. So Ika's finished his PhD, he's on the ancestral reconstruction. Fletcher did the experimental evolution. Jacob is starting on the filament work, and the jury and Alex on droplets and DNA work. And funders, and thank you. And now ready on some of the trades. Next.
Well, thank you, Matt, for that amazing talk. We have a few minutes for questions before the break. Yeah.
We had attacked, okay, so background. So we were resurrecting the elongation factors from bacteria deep in the U bacteria, which this is 25 years ago. And we got attacked because the claim was that we had, by the phylogenetic inferences, the Bayesian inferences of these ancestral characteristics, we were biased towards proteins, among other things, that were more thermally stable. But you see, when you go back in time and your more ancient flagella thing are more thermally stable.
So that's it's a good question. It's a common challenge on ancestral reconstruction, like because you're picking maybe the consensus sequence, you get more stable sequences. There are some people that possibly take it too far, you know, they don't make these proteins and then they're so thermally stable, they're claiming that early Earth was that temperature. I think that the methods are better, and as long as the methods are better, and I think that if you don't need the way that one of the ways that's handled now is not just making the best one, like the best estimate, making the second best, third best, fourth best, making other options down the tree and showing that those also have the protein function that you're looking for. And then you can say, well, it's not just a sort of factor of that. Your broad question, you know, is it all an artifact of consensus ultimately, and that's presented by thermostability. I think if you build your biology in a sensible way and you ask the question in a reasonable way, you can make alternatives at the same time. So to answer your question directly, we don't really purify these proteins to measure any sort of thermostability because they're really hard to purify. We just have them in the organs, which is quite uncommon. Most people in ancestral reconstruction don't do that because in this sense, it's a bit flawed, right? We have a whole E. coli and we've taken one protein and made one part of that one protein ancient, and then we put it in the present. So an ASR purist who wanted to say, I want to understand what that protein looked like back in time, probably wouldn't be trying to put it back into a modern E. coli. They might purify it, take it out, and then interrogate that in vitro. And that's where more of the thermostability work, because they're also doing that protein kinetics. I don't know if that answers your question. Usually I sort of sidestep this, and that's where I say, oh, look, I'm not really interested in evolution. I mean, I am, but it's a bit of a field derivative thing. So synthetic biologists is like, well, it's more thermal, well, maybe that's awesome. Maybe that means I can do my reaction at a lower temperature. I can get a traditionally unstable protein now stable. So it depends on what you work on. I usually avoid saying, this is evidence that this is what happens. You know, it's using tools to get to our best guess, and then using that to infer structure function.
Yeah, great talk, Matt. I just following up on the ancestral sequence reconstruction stuff. I'm just so because you can, in principle, screen, rather than having to biochemically characterize whether a protein is functional or not just by doing your kind of rescue experiment, like putting piece back in. Do you have a sense from you were sort of saying, well, we got better at making trees. Principle of ancestral sequence reconstruction is that the ancestors look to be functional, right? So you showed some of yours functional and some of them didn't seem to work. Does it improve, proportions that are functional as you improve your methods in ASR, or is it really still a mixed bag?
I think if you wanted to do something like benchmarking in that sense, like using the let's say using anthropology and benchmarking different methods and the capacity to generate a functional protein, that would be the way you potentially have to do that more of course. In this case, we haven't even chosen things that we might expect to be time matched or from biology. But your question is bang on in the sense of what does that mean when he says we got better at it? Because the methods are kind of very standard, the maximum methods, and the ASR methods even more standard and older. So ultimately what I'm getting at is something like the states have tons of horizontal gene transfer. If you really wanted to say this is what happened, you need to go and build a proper gene tree and reconcile all of that. That's challenging in microbial systems, can be. And so that's where you might have some errors. Your biology might not represent that because you just made it from a single protein. In our case, I guess we tried to do a little bit of this by taking some really recent ones. You assume that the really recent one, just from E. coli-like things, when you bring it back in E. coli, would be more likely to be functional. We were lucky that in the first pass, the more ancient and the more modern both of them. Then we just got a bit more risky. We haven't got the point where nothing worked, and maybe that's the filament. So we have to do that to be more around the So I don't think it's. I think you're always going ultimately to have to use your knowledge of the protein system. That sort of synthetic biology sense. You're still trying to engineer a functional protein, right? So if the ASR said there's a 20% chance that aspartate is gone, I probably wouldn't even buy that gene because I know that if you mutate that aspartate, it's not functional. So we're using our knowledge of the system to then sort of screen what ASR is telling us.
All right. Let's go ahead and thank Matt again.
Thank you.
So that ends our first session today. Now we have a small break. So if you go back to the reception area, you should see some nice snacks laid out for you. We're a little over time, but that's okay because we have a long lunch break. So let's meet here around 11:05.
Thank you.
Thank you. Yeah.
That's just.
You gotta do more. We're at the top. Yeah.
Okey dokey. Everyone can take their seats. And. Okay, so welcome to the second session of the day. We're moving from chemistry into space in this session. And our first talk was originally going to be presented by Laura McKemmish, but instead we have Juan here to present. So I'll let Juan take it away.
Yeah, hi everyone. As Claire said, unfortunately Laura won't be joining us today due to personal reasons, but I'll be giving her presentation instead. A PhD student in Laura's group. Laura will be here tomorrow for the poster session, so if you have any difficult questions, you can ask her tomorrow. I'll take the easy ones. Yeah, so this talk is on beyond phosphine on Venus. How do we know if we have found elements? Cool. So just to start, this is the McKemmish group here at UNSW. So Laura pretty much, or the McKemmish group pretty much focuses on using computational chemistry and molecular spectroscopy for different applications. One of those is astrochemistry and how we can find molecules in space. Yeah, just all the stuff that we also do that are not particular relevant for this talk. And just for you to know, if there's any reference that you guys find interesting, there's going to be a QR code here that will hopefully take you to the web, to the paper if you want to see it. Cool. So the search for outer space life. The question of whether there's life on planets different than Earth has been a constant in humankind throughout history. So indeed, like ancient civilizations have been wondering this stuff, whether there's aliens or life outside Earth. We have tried to make contact with aliens, so we have the Arizona message that I think was sent like in the 70s or 80s, can't remember. But yeah, when we basically sent some like information about humanity to space, waiting to get a response. You could say probably that this is the biggest thing, if there are aliens or not. Yeah, so aliens have made it into the big screen. We have movies like E.T. and some like a whole universe of just movies talking about aliens and stuff. But I guess scientifically, when we talk about finding aliens, we mean searching for molecules or for molecular signals of life using ground or space-based telescopes. So we have now the James Webb Space Telescope that is like probing the atmosphere of exoplanets. So that is one of the biggest ways that we can use to find life outside Earth. So how does it work? So if you think about this, I don't know, like sort of like planet somewhere in the universe, hopefully this planet will have an atmosphere, and then in that atmosphere there are going to be some molecules there. And so the question is, how do these molecules go to the atmosphere of this planet? So you can think of some like volcanic activity. So some molecules will be produced there and make it into the atmosphere. Some geologic processes as well might bring them there.
weather-related events like lightning and things like that can also bring molecules into the atmosphere. And at the end, or another one, well, potentially life, right? So this is very important because here the question is not so much about which molecules does life produces that can go into the atmosphere, but instead is which molecules geochemistry does not produce that make it into the atmosphere. So that is like one of the biggest questions, and that's how we research it. But the thing is that, I mean, if you think in the planets in the solar system, we can potentially send probes to these planets and like take actual measurements of, I don't know, samples of the molecules that could be in the atmosphere. But when we think about exoplanets or outside of the solar system, well, we cannot go there, at least yet. So how do we rely on, how can we search for the molecules there? Is through a spectroscopy and how molecules vibrate in the atmosphere. So yeah, infrared spectroscopy is basically our gateway to finding aliens. So just as a little recap on infrared spectroscopy, you have a molecule, the molecule absorbs infrared light, the molecule starts vibrating. So in this example, for water, that typical vibration is around 1600 wave numbers. Then you have a symmetric stretch there, and an asymmetric stretch. So yeah, when you record an infrared spectrum, you get all these bands, and each one of those bands correspond to an individual vibration in the molecule. But the important thing here is that molecules, each individual molecule will have a unique infrared spectrum, and that's basically molecular fingerprints, right? So if we find a spectra for a molecule, from an exoplanet, then we can say like, oh, this spectra corresponds to this molecule because there are no two molecules that will have the same spectrum. So yeah, we want to find aliens. We know that the best way of doing it, I guess, is through looking at the molecules in the atmosphere of planets, and infrared spectroscopy is the best way of doing that, just seeing how molecules vibrate. But I guess which molecules should we look for? Because there are like lots of molecules that we can see. So Sarah Seeger and co-workers at MIT, they sort of proposed this list of potential biosignatures, which are molecules that if they're found in an exoplanet atmosphere, then they might indicate the presence of life in that planet. And basically the way they constructed this list is they look for molecules that are stable under the standard pressure and temperature conditions. Then they ask themselves, does this molecule has like some viral detection using telescopes? Are there any like evident potential geophysical false positives for this molecule to be produced in making it to the atmosphere? And after doing all this like filtering process, they end up with a list with more than 16,000 molecules that yeah could be potential biosignatures if they're found in an exoplanet atmosphere. But the important thing here is, or what I want to emphasize here is the word potential. So these are not like biosignatures themselves. They're potential because if they're found on an exoplanet, then we need to do like a bunch of studies just to know if the molecule is actually produced by life, or whether, or if it has like some geophysical source. So one of the best examples showcasing this was the tentative detection of phosphine on Venus back in 2020. So yeah, they found phosphine in the cloud decks of Venus, and if you read the paper, what's interesting is that they couldn't find any source, non-biological source that would produce phosphine to the abundances that they found in the planet on the planet. So it was great. Phosphine is a biosignature, by the way. So it is produced by life. So it was, yeah, just like great news, because it could potentially indicate that there were elements on Venus. Of course, this created like a whole big scientific debate on whether it is or not. But then Laura's group, so this is Laura. Basically, we decided, Laura got together like a lot of people from different backgrounds, so spectroscopists, computational chemists, astrobiologists, basically asked, what are the phosphorus molecules that are important on Venus, and how does phosphine make it into the atmosphere on Venus? So it turned out not to be kind of like a challenging task. task to do. So we look for all of those phosphorus containing molecules of potential importance in Venus, on Venus. But there's little research, there was little research on the phosphorus molecules that were present there. So we couldn't find like any good spectroscopic data for most of these molecules. And the thing is that these phosphorus molecules, they don't really make it into the atmosphere that much. So they're usually like in rocks or like oceans and things like that, or like solid state. So there was a question of how it's like how phosphine make it into the atmosphere. And yeah, with the data, with the high resolution data and stuff, only diatomic molecules containing phosphorus had like available spectroscopic data and phosphine as well. So yeah, it was like an interesting journey here. But subsequent investigations, right, that raised doubts on observations, because they now think that the signal that the astronomers first found and thought that was phosphine could potentially be SO2. So yeah, I think I'm not really sure where the debate is right now. I think it's probably that it was just a misassignment, so it is SO2 what they found and not phosphine. But this raises a very important question when we talk about detecting molecules in space, and is we cannot rely on a single line to make a detection because we need more than that. So if you think this is a very low resolution infrared spectrum for ozone, which is a potential biosignature. So say you're taking measurements with, I don't know, the James Webb Space Telescope or Hubble from an exoplanet, and you find this peak here around 1300 wave numbers, and you can think like, oh yeah, that's ozone, it's great. But then there are actually other molecules that will also have a peak there. So you can see NO2, methane, H2O2, and indeed there are probably hundreds to thousands of molecules that will also have a peak in that same region. So that we cannot rely on just a single line to make a definitive detection. And this was pretty much the problem with the phosphine detection, because it was only one line in the spectrum. Yeah, so there are another 600 molecules there. So to make these detections, we need very high resolution spectral data. And what we mean by that is, so you know how we have the spectrum and the different lines for the molecules. We need to know to like very high accuracy where the lines of those molecules are. And usually this means like sub-wave number accuracy. So like we need to be really, really sure where they are. So this is a list of the current molecules that have that high resolution data available. Yeah, so red is in progress, and I think the green ones, yeah, the green are the ones that have been that do have the data. But you can see that this is not really comprehensive, considering the amount of molecules that exist and that could be produced by life. So the McKemmish group has expertise creating these sort of like high resolution data. So this is a line list. Basically, a line list is a collection of all the transitions for a molecule. The Laura produced for TIO, which is a molecule that has been found in like M stars, I think is the one. But yeah, anyway, so it's just we can produce this really high accuracy data that matches the experiment pretty well. And this is the data that the astronomers are using to find molecules. So yeah, Laura will be happy if you have like any molecules in mind that we could like start to data for. But the thing is that creating this high resolution spectral data for molecules is a very time consuming process. So experimentally, this usually means provide resolution, as I said, with sub-wave number accuracy. So we need to get like really, really accurate where the lines of the molecule are. And just to give you an example for that TIO molecule, this requires over like 25 publications to obtain all that spectroscopic data. And if you think, I don't know, like a publication could be, let's say, I don't know, 6 months or a year, this is probably 25 years of work to get just that data from one molecule. But computationally, that is also very expensive. So CCSVG-like calculations, basically that means really high accuracy calculations that take a lot of time. And for example, for phosphine, it took a whole PhD, so approximately 4 years, to get all these spectroscopic data for that molecule. So if we think back to that list of potential biosignatures with like 16,000 molecules, only less than 1% of those molecules have high resolution data available. So that's like, yeah, very low. And the key thing here is that if we don't have spectral data for a molecule, we simply cannot detect that molecule. So this is a really big limitation in the field. Because yeah, remember that what we get from the telescope is just this sort of like set of different lines. And if we don't have something to compare with or against, then we simply just don't know which molecules are there. So the approach that the McKemmish group is taking is, what if we do a complementary approach to producing this high resolution data? And instead we do like a high throughput generation of approximate data. So we use computational quantum chemistry, pretty much DFT. So calculations are somewhat accurate but fast. And we can do it for thousands of molecules rapidly and get approximate spectra for them. So there are some interesting applications here. We can characterize spectral congestion and molecular ambiguity. So that means if you have, think again of that example for ozone. So if we have that frequency window, how many molecules could absorb in that region and what's the likelihood of a potential misassignment there? We can identify a strong molecular absorbers because with the frequency there's an associated intensity. So ideally, the more intense a line is, the more likely it is to be observed in an spectrum. This data can also be used for machine learning applications. So instead of using quantum chemistry, we can just train a machine learning algorithm to predict the spectra. But the important thing here is that the data that we're producing is not accurate enough to allow molecular detections in space. So it's not like you can just grab this data, go to, I don't know, get some spectra from JWST and just say like, oh, we found this molecule, because it's not gonna, the accuracy is not good. Instead, it's just a complementary approach to sort of like guide which molecules require high resolution data. And by approximate here, by approximate here, we mean like median errors of around 10 wave numbers. So we're about 10 wave numbers off from where the frequencies for the different molecules should be. So we have designed this really big this sort of pipeline for calculating approximate spectra for thousands of molecules. The next question is, okay, so which molecules do we want to calculate? So we can go back to the potential hallucinogens list has 16,000 molecules, but that's probably too much. So we thought, okay, which molecules are, which atoms are elements in the periodic table are the ones that are relevant for life, or at least life as we know it here and now. And we can bring the biology periodic table. So just molecules containing the Schnapp's elements, that's how we call them. So from that list of 16,000 molecules, there are around 2,700 molecules that only contains these elements. So these are the molecules for which we calculated all these spectra. So we have like a really big data set of just approximate spectra for all of these molecules. Yeah, so this is an automated approach just to show you how it works. We get the geometries for all of those 2,700 molecules. We run the calculations to get the approximate infrared spectra, and then we do some data processing and analysis. That basically means, yeah, just plotting the spectra and using it for different applications in finding molecules. Oh yeah, and then as I said before, approximate here means 10 wave number median errors. So yeah, this is the median errors. There are some cases where we are much better, but there are some cases where the frequencies are off as well, like more than just 10 wave numbers. So it was interesting just generating this data because we sort of, for 2 years, we just thought like, oh, this will have like really good applications in looking for molecules, but it was sort of like an idea. But then with the recent launch of the James Webb Space Telescope last year, there was the first carbon dioxide detection on an exoplanet. So it's this one here and this line here. It was really interesting because it was, as I said, the first time carbon dioxide had been detected through this asymmetric CO stretch. So just like, as you can see it here, just the molecule vibrated that way, and they found this signal. They were like, yes, that's CO2. But what was important when they released this like science communication article, like NASA, when NASA did it, it was like, okay, so that's CO2. But then if you look here, there's like a little signal there, and the article didn't report any molecule that could be responsible for that. So like that created a huge scientific debate on, okay, so there's another molecule there, we need to find it, and we need to know which molecule is responsible for that signal. So we thought, you know what, this is the perfect opportunity for us to just play with our data and see what our data can tell us about this molecule here. So before doing that, I just want to showcase, show you some preliminary stuff that we did with this spectra, just to start our filtering process. So as I said, this is CO2 here, and this line here is the one that we want to know which molecule it belongs to. But there is some interesting stuff here, and is that there's no signal for a CH or NH stretches here, which are around this area of the spectrum. And that's really important because that could tell us that if there's another molecule in the atmosphere of this planet, then that molecule probably doesn't have any NH or CH group. So that was a good filtering process for us to like start discarding some molecules from our analysis. Anyway, so we started doing that. This is the peak that we're interested in. So the data in blue is the data collected from the telescope, and then the data in red maroon is the one that we calculated for all these like 2,700 molecules. And we found some really interesting molecules that could be responsible for that line. So you can see, for example, ozone here has a line there, so it's an interesting biosignature. Also this molecule here. Anyway, so we found some really cool molecules, and when we were ready to sort of like continue with this project, well, they found that it was SO2. So as it happens in science, we just got there late. But it was cool. But the interesting thing here is, so this is from the paper, where they showcase that, yeah, this is SO2. But if I go back to this one, you can see that SO2 was one of the candidates that we have in our data. So it really showcases that there's a lot of potential with the data that we have, that sort of like this idea of providing directions into which molecules could be responsible for different lines. Yeah, so we have it there. So yeah, that line basically, this SO2 line here corresponds to combination of different vibrations in the molecule. So it's this asymmetric and symmetric SO stretches. But the interesting thing here that goes back to the phosphine problem is that this detection is made on just one line, which is this one here. So like a definitive detection of SO2 should also include these two lines. And this here is just like modeled. So this is just more, yeah, they model the spectrum because the observations only cover this small like window reading. But yeah, so we need more data to be 100% sure that that's the molecule that we're finding. So yeah, we were sad because we didn't get the SO2 thing, but it seems like life gave us a second chance. So if you look at the paper where they found that, where they made that detection, they talk about, yeah, so this is carbon dioxide, water, and SO2, but then there's another signal in the spectrum that they just haven't been able to identify what it is. And they literally say that in the paper. So this is what it says. You don't have to read it. But basically, what they say here is that they tried all the molecules that have high resolution, that we have high resolution data for. They tried all those molecules and to see if one of those match this signal, and they couldn't find anything. So in other words, there's no spectral data available for the molecule that is responsible for that line. And so that's sort of what we're doing at the moment is, without data, which molecules could be responsible for this line. So if you want to know more about this, you can come to my post tomorrow, where I talk about more the molecules that we found that could be responsible for this line. Our data has some limitations. One of those, one of the clearest one is the accuracy. So our data is not really accurate to make definitive detections. But the other thing is that we need to add rotational profiles to our data. And just to show you that, so the data in black is the one from the telescope. So this sort of like little hill here is the SO2 line that they found. And when we use our calculated data, the frequency is actually here. So it's really off from where it is. And the reason why is because of the temperature. So the temperature is affecting how the frequency, how the band profile for the frequency looks like. And it's sort of like shifted to this side. So we need to include those, and that's like an active area of research right now in our group. Cool. So yeah, we have room intensities as well, as I mentioned before. And we can determine like detectability limits for molecules. We want a comprehensive list of potential atmospheric molecules. So we just want to know all the molecules that could be relevant in the atmosphere so that we can calculate that for them. But when assessing potential molecular candidates, we need to know relative likely abundances. So like how likely is it that the molecule is going to be produced at that point? How much is it going to be produced so that it can make it into the atmosphere and the signal be strong enough. Yeah, so looking for audience is a community effort. So you can think, yeah, so we need observations, do some atmospheric modeling just to see how the molecules interact in the atmosphere, how they make it there, and then ask the question if whether there's aliens or not. But there's more to this like straight diagram. One of the things that we need to consider physical and chemical characteristics of the atmosphere, just to make sure how the molecules interact there. We need the spectroscopy, of course, so we need to fill this gap of data that we have at the moment. But most importantly, we need also astrobiology to sort of like tell if the molecule that we're finding there could be related to life or not. So inputs from this community that I really appreciated would be, yeah, which atmospheric molecules could be associated with geology, origin of life, different areas of early life, photochemistry, and stuff, just to make sure that we have all of this together. Yeah, so this is pretty much the sort of like the pathway that we're following is producing this big set of approximate data to sort of make directions into the molecules that need high accuracy data and run more accurate calculations, perform more accurate experiments, and then just go and see your astronomical observations and see whether the molecule is there or not. Just to summarize, high resolution is essential for definitive detections. As I said, again, I just want to emphasize on that. The data that we're producing is not intended to replace the high resolution one. We still need it. It's just a complementary approach to sort of like provide directions to which molecules require high resolution data. Producing that data is challenging, but it's rewarding because at the end you can find the molecules that is full. So yeah, we're just doing this high throughput condition of IR and microwave spectra. So Maria is also has a post tomorrow talking on this on how we do high throughput rotational spectroscopy. Yeah, so our accuracy is about 10 weight on this. That's why it's approximate. And yeah, so spectroscopy can show us light, and that's pretty much it. Thank you.
Right, we've got a few minutes for questions. We'll start up the back. That's you.
Nice talk, Juan. I've just seen this talk before with Laura. I've been wondering with this, because you've got the STOPS data set, if you sort of like map out a whole planet's atmosphere, like in places where the things don't fit your data sets, can you infer the presence of atoms that are not in that data set, like fluorine?
Sorry, could you repeat that question?
Like if you like for this planet, like a planet, you sort of like have this sort of like spectrum of data, all the rotations and stuff coming from that, and then can you sort of like map out a planet's atmosphere and then sort of infer based on gaps in your data, gaps where your data set doesn't match the planet's atmosphere, that might be like other atoms that you haven't included, like fluorine, for example, or other molecules?
Yeah, that could be something that can be done, especially if you consider so with functional groups particularly, right? So they have like localized vibrations so that we kind of know where the frequencies should be. So like if we find frequency, sorry, I just don't have that data here, but we do have sort of like a plot where all the frequencies for our data are. And so we can find some regions where we just don't have that, but it doesn't necessarily mean that all the molecules won't absorb that. So yeah, if there are molecules with other atoms outside our data set, we should be able to see them in some spare regions of the spectrum.
Yeah, but you can sort of infer it's like, oh, I think that molecule that's from this atom that we don't have additional data set.
I don't know if we can go that far, but definitely fed that the molecule has some atoms that are known in the data set, yes.
Martin?
You mentioned in one of your slides a thermal shift in the wave number for different molecules. So like Venus's atmosphere is 400 degrees. Do you then, when you're doing an analysis of an exoplanet, do you try and estimate the thermal profile of the atmosphere?
Yes, oh, this is why you don't have images. Yeah, cool. Yes, so we actually, we can do that. So there's a program called bGopher, where we can, so what's happening is the vibrational frequency, which is this one, doesn't get affected that much by the temperature, is the form of the rotational profile associated with the band. We have the rotational levels are the ones that are affected by temperature. So yes, we can model how the temperature can shift the frequency. We're still trying to figure out how to do that in a high throughput manner. So that we can do it for one molecule, maybe 10, but when we talk about 2,700 molecules, that's just too much good. I mean, so yeah, we're just trying to figure out how to do it. Yeah, out of one group. Does that answer your question? Yeah.
I was gonna say, in the interest of keeping to time, ask one or Laura questions in the next 2 days, or today. But thank you, that was a great talk.
Okay, thank you.
And now we've got Martin. Whose presentation I will have.
You gotta push resume share, I think. Push that little play button. Oh, push stop share and then share again, I think. Yeah. Yeah. Yeah, looks good.
Yeah, thank you. It's lovely to see everybody here today. Thank you for coming, for those of you who have traveled the distance. I usually talk about early life and here on Earth, and we're lucky that we're going to be going and seeing out in the field in a couple of days. But thanks. I wanted to introduce a new astrobiology program, and that's a new sample return mission Mars that a group of colleagues and I have been developing over the last couple of years. We call it LifeSprings MARS, and it's a new idea that we're trying to promote for Australia, but as part of a multinational consortium. So so astrobiology is all about addressing those big questions. Are we alone? Where did we come from? So it's really about investigating where there might be life outside of Earth. Is it common in the solar system across the universe? And of course, there are many missions that are ongoing at the moment to try and solve that question. There are a number of different rover missions that have landed onto the surface of Mars, and they're all basically following the same exploration concept, which is this idea of follow the water for signs of life on Mars. But they're doing that in a very specific way. They're all looking for remnants of organic molecules trapped in sediments that were deposited by flowing fluids, liquid water plus or minus brine. And so that's a search strategy that is obviously very effective on Earth, because where there's water, there's life, and where there's life, there's water. But there are problems with that exploration model as well. And we try to point this out at the Lancet workshops with the Perseverance rover, that there's risks involved in looking for organic molecules because they do not last for billions of years. They are susceptible to breakdown by radiation that can penetrate through the surface. And finding them in sedimentary rocks can be like a needle in a haystack. They are not everywhere. They're only localized in a very few places. And the examples that were given, for example, to support the Perseverance rover going to Jezero Delta,
was that the Amazon Delta is rich in organic matter. In fact, there are oil plays in the Amazon Delta. We're talking about an entire continent of rainforest, plus or minus the microbial community, that's washing that organic matter down. I don't think anybody is applying that to Mars, but the question is, how do you concentrate organic matter to be discovered in ancient lake or river deposits 3.5 or more billion years ago? So we've asked the question is there a better target potentially? And our research has focused for many years on hot spring deposits. And we got very excited when the Spirit rover in the 2000s discovered these nodular deposits of opaline silica, which were originally interpreted by Steve Squires in the 2008 Science publication as being deposits from hot springs. But at the time of their discovery, this unusual form of deposit had not been recognized before on Earth, and so it wasn't really clear what they were looking for. But since that time, there have been studies that have gone to the Atacama Desert in Chile at about 3,000 meters elevation, in most Mars-like environment on Earth, being very dry, high irradiation, high UV. And there are deposits of opaline silica that have these identical nodular features, as well as these finger-like protrusions. What you see on the right is from a place called Tuja in Chile, but Motaccio is perhaps the better known one where these nodular deposits are found. And what you see in the darker color is the outflow channel from a hot spring that's just at the top of the photograph. And these two images are at the same scale. The picture taken by the Spirit rover on Mars, and this photograph with the geological hemisphere scale on Earth. When you look at those deposits in more detail, they have really frighteningly similar characteristics. In red again is the picture at its highest resolution from the PanCam image on Spirit rover, and then in more detail here the deposits from Motaccio. And you see they have these branching finger-like shapes that are one-to-one matches at the same scale with features found on Mars. Now, what's exciting about that is that those structures on Earth are formed by and in the presence and because of microbial activity. And when you take one of those little fingers and cut them in half, you see these fine scale laminations making this topographic feature, and entombed within those layers and causing that branching and that topographic growth of these structures are fossilized microbes. Here we have filamentous, still green cyanobacteria in these nodules that are formed today. But you'll see that in these laminated parts, there are also trapped and silicified filaments that are deposited during nighttime when they lie flat. And it turns out that analysis of these kinds of structures of hot springs around the world show that A, they're very common in hot springs, and that they are all influenced at least by microbial mediation. So the question is, have we already seen the biosignature perhaps on these? Hot springs are important for a number of other reasons as well. They preserve and trap textural and chemical evidence of life back through the geological record. So our research on the age of stromatolites in the Pilbara show that they are associated with hot spring deposits. My graduate student, Clara Jokic, she's now graduated, found this little rock sample, which is only a few centimeters across. We see these little positive growth structures. That's actually the same kind of finger-shaped structure that were found at Motaccio. And this is a very specific rock composition which formed from a geyser. And there's textual evidence that there's preservation of life in these rocks that are about 3.5, about the same age as the rocks on Mars. The other reason that hot springs are so important as a potential sort of biological exploration space is that because the community in origin of life studies has by and large shifted from thinking about an origin of life in the deep oceans to an origin of life setting on land, and specifically about the geochemical complexity of hot springs. Now, this is a picture that everyone is familiar with. This is Grand Chromatic Springs from Yellowstone, and many of us think about springs as a single spring like this, and that's a chemical entity on its own.
But the reality is that hot springs occur in fields of many tens to hundreds to thousands of springs, and each of them is geochemically distinct and unique. Some have concentrations of iron, some have concentrations of boron, some have concentrations of sulfur, some are acidic, some are hot, some are cold, some are temperate. And they all mix and match their components. Hot spring fields are geochemically the most complex and diverse on Earth. And so there's a lot of research now thinking about, could life have started in hot springs on land? Because of a concept, and Jim talked about it briefly today, but I first got this idea from listening to a talk by George Whitesides here, who discussed the idea that, well, maybe developing the complexity of life is not just purely a linear sequence of steps, but it's actually that it complete one set of reactions in one environment, do another set of reactions in another environment, and then start to mix those. And that's where this idea of complexification and systems development is so powerful for hot springs. So we argue that hot spring deposits would be the place to look for life on Mars. Because if life ever got started on Mars, it likely would have been in hot springs. Hot springs entomb and preserve signs of life for billions of years, and they could be regarded as the first and last outposts of life in an environment that was geologically dead very long ago. So we feel that hot springs would provide the best opportunity to search for them. And so we're advocating this mission that we go back to the Columbia Hills and retrieve samples of these digitate nodules of opaline silica and bring them back to Earth. Now, because any analysis for life has to be done using the best, most sophisticated scientific tools, we regard the mission as just purely a fetch and return mission. There's no point having a fancy scientific rover on Mars, just collect the samples and bring them back. And that allows the mission to be extremely light and nimble and low cost. And in fact, we've been working with engineers from Japan who have devised a mission that can return samples in a single launch from Earth. Now compare that to the Perseverance Mars 2020 rover mission, which requires 3 different launches, each for $3 or $4 billion, and we can do ours in $100 billion. Now, the good thing about Columbia Hills, the digitate nodule of opaline silica are here in yellow. We would collect 3 samples, each only a maximum of 15 grams. So we're talking about a very minimum payload, because the high resolution, high sophistication analysis here on Earth needs only microsampling to enable a huge amount of science to be done. But in the same vicinity, we can get a huge range of different types of materials that can augment our studies to learn about the history and evolution of Mars, both its geological history and its atmospheric history. So in a timeline of its early Mars evolution, this is where we think the opaline silica nodules would have formed, but the range of samples we could collect would give us some important information about this material. And if we're able to get our escapes on, our mission might be able to be the first sample return material back from use. So this is a rough outline of the mission architecture with a single launch from Earth, and you can now just buy a Falcon 9 launch for about $70 million from Elon Musk. You have a dual deployment of materials from that single launch. For direct entry to Mars is the landing materials, so the sampling rovers, the ascent vehicle, and the sample CubeSat, which would then be launched back into space. The second component is a fetch satellite, which would be parked at the Sun-Mars L1 Lagrangian point until such time as the mission was complete on the surface and the samples were launched, and the CubeSat relays to that satellite. And then the satellite is reactivated, it swings past Mars and returns to Earth for deployment of the sinkers into the Australian desert. So this can be done within a reasonable time frame. We are trying to look towards a 2032 launch, and if we're lucky, we might get stuff back relatively quickly. So our team consists of a bunch of scientists who are experts in hot springs. Steve Ruff on the top left was a mission scientist with the Spirit rover and has been a real advocate and main researcher on the digitate nodules from Mars, and now doing that research in collaboration with Catherine and myself here on Earth. Mike Rowe, also at the University of Auckland, is a geochemical specialist who's doing a lot of microanalysis of those nodular materials. Tommaso from the European Space Agency is an expert on microbialites, and he's very excited about the mission. The samples could be returned to Earth, and Asa Nakamura, pictured below myself there, is the director of the Pheasant Memorial Laboratory that has a fully automated remote laboratory that can analyze space materials. And then our team of engineers from JAXA, ISAS, who have developed the mission architecture. So the way that we think this will, and we've got an advisory board with people from NASA, JAXA, and the European Space Agency. Just a couple of notes about why a mission for Australia. Australia is not known for conducting missions, but any nation that's serious about developing its technologies globally and playing on the world stage is now going to Mars as a showcase for technological development. And there's a danger that if we don't participate at this level, we'll be left behind. Australia has a lot of capacity. We don't have capacity to run a mission like this on our own, but we can make a major contribution to it, both from the science side, but also from the space engineering side. And why then LifeSprings MARS particularly? Well, it's a new type of light and nimble, low-cost sample return mission that can be a hallmark for a new type of mission to many different destinations. The science rationale is based on a unique Australian geoheritage of the oldest evidence of life on planet Earth. It addresses those fundamental questions of astrobiology. We feel that it maximizes the chance of mission success by returning to known sites. And this is actually quite a key because NASA decided not to go back to Columbia Hills because they do not go back to a place where they've been before. But as a geologist, as an earth scientist, if we see something of interest, we go back and look harder and look better. And so we actually can reduce the cost of the mission by utilizing pre-existing knowledge of these really extraordinary deposits and the extended sample suite available at Columbia Hills to enable a relatively low-cost mission to bring those materials back. With an idea of a simple fetch rover, we don't need the full, scientific instrument suite of Perseverance or other rovers. It's a simple fetch and return. And we feel that Australia can develop a bespoke technology using its inherent mining technology to develop the sophisticated sampling tools that we need for this fetch rover. And we feel that it also meets a Mars component for the Artemis Accords. So how do we think that'll work? Aside from the architecture, we are now starting to approach this by dividing this mission up into its different components and seeking different countries to contribute to the mission. So we have New Zealand on board, we've got the United States, we're talking to the Japanese Space Agency, we hope that India will be involved. And Australia's part is something known as the Quad Alliance, which is between Japan, India, Australia, and the US. And we feel that if we can get contributions from those different parts. And we just heard, so I'll be very quick, we just heard from Japan that they want to have a Mars mission to land on the surface. And this would perfectly meet their requirements. Australia has a number of different capabilities that could contribute to such a mission. As outlined here, I won't go into the details. Lots of benefits. It can stimulate students, inspire students for STEM. It can showcase our developing technologies. Can bring the nascent space industry together in Australia, academia, government, and industry, and maybe strengthen that Quad Alliance. So there's more information. We've now got a website up and running. We're trying to build consecutively or collaboratively at the same time, sort of a Team Australia to develop our own capabilities and then develop a sort of global collaboration. So if anybody's interested or has ideas, please get in touch and we'll see if we can make this thing go. Thanks very much.
Thank you, Mark. Very exciting. So I'm sure we've got a few questions. Yep.
What other forms of evidence do you have that it is in fact consistent with some of that deposit you're referring to? Is it just morphology? Do you have like prism signature or larger geologic signs of the hot spring here?
Yeah, so there was the initial studies were done by infrared spectrometry, and they found that the signature was very characteristically of opaline silica, which only forms in a few different ways. And that's been investigated by Steve Ruff and others. So in hot springs, there's also fumarolic activity. We have steam that can mobilize and liberate silica. But it turns out that the way that that's deposited is a very different style. And the additional factor that was found in the spectrum was that there was a signature of halite. And halite is helpfully found in those earth analogs because the fluids that those deposits come from are very chlorine rich. And with sodium ions, they combine to make halite crusts. So that was additional evidence. One of the things we don't know from Mars, because all we had was photographs of that sort of centimeter scale, where we're doing a lot of microanalysis on the structures on Earth to be able to characterize them and identify that sort of biosignature, both in terms of the style of the lamination and the internal textures, but also trace element distributions, carbon signatures, et cetera. So yeah, we don't know a one for one. We also have 2 students here who are engaged in PhD studies that are trying to replicate the growth of these kind of structures with biology and without biology, and see if we can identify whether those kinds of structures occur under a biological situation. So it's not proven I've seen a biosignature on Mars, but gee, they're pretty good.
Yeah, just going to follow up on that, Martin. You probably thought about this, but if you look at very ancient rocks, do you see any examples that you think could be abiogenic, or do they all look biogenic? Is there a way to sort of say, well, if I saw this, I'd say this is pre-life, and it's similar, but it's not quite the same.
Yeah, that's been a huge controversy within the field of early life studies. So almost every claim that's made for evidence of life has been counterclaimed as being abiotic. So there's still a lot of room for improvement there. Interestingly, I've been contacted over the past 2 weeks by 2 independent researchers that are now looking at new ways of identifying biological carbon signatures without finding biomolecules. But looking at the distribution of the carbon chains and stuff to say like so there's still a lot of movement in that field, and there are absolutely discrepancies in the way people think about it. And part of what we're trying to do is emphasize the importance of context and texture. So we've put NASA into the field in the past, and have to come next week again to investigate these outcrops and start to really get that sort of community involved. Understanding it's a biological signature.
We might do one really quick one. Are you really quick? Yeah.
How long does stuff last on Mars from what you know about it? So if you chop one of those Chilean things and you just drop it on Mars, what would it look like after 1 billion years? I guess we know a lot about the conditions, but how would it last? You have fossilized cyanobacteria?
Yeah, so one of the problems is that anything that's exposed on the surface of Mars, because there's no atmosphere, is exposed to very high radiation, and that can damage organic molecules. It doesn't damage the structure of the rock, so it hasn't broken down those hook lines to look at the rocks. One of the only other suggestions for making those branching structures in the Mars is by wind blasting, by sand blasting with wind and stuff. But we've done, Steve Ruff has done some studies on that, and they form quite different textures, distinctive textures. So we feel that the materials will probably be integral, still be whole from what they were. And it's also very likely that those materials were covered for a long time by younger rock successions and have since been exposed. So they might have only been exposed for 1 billion years, but still, exposed to surface radiation. So we don't expect to find biomolecules in the structures, but we're looking for the trace elements, the textures, that sort of.
Anyone else have questions? Please accost Martin during today and tomorrow. He leaves for WA on Thursday. So
Yeah, in the afternoon, we'll be here all day.
Yeah.
Anyway, thanks.
Yeah.
Thanks very much.
and it is now lunchtime. So
Yeah, I can make it.
yeah. Albert?
All right, so if you go out to the reception area, there should be some lunch waiting for you. It's a nice day. Like I said earlier, I suggest walking either up the steps or down the steps where you can take a right. There's outdoor seating aplenty, and please come back by 1:20.
Can we leave stuff here?
You can leave stuff here. Yeah, I will make sure somebody is guarding it.
I feel like I didn't answer the question.
It might be me.
Very nice.
Yeah. Do you want to use your own computer? I think we're ready. Do you want to start?
Hi, everybody. Hey, good afternoon, everybody. Welcome back to the rest of the session on space exploration and technology. My name is Astrobiology and I'm a postdoc from the Anabank Research Group. And it's my great honor to be the session chair this afternoon. So before we proceed, just a few housekeeping details. So we've got 2 invited talks and 2 oral presentations. So one of the presenters couldn't make it, so we have 3 talks in total. So invited speakers will speak for 30 minutes in total, which means 25 minutes for talk and 5 minutes for Q&A. And the oral speakers have got 20 minutes in total, so 15 minutes talk and 5 minutes for Q&A. So I do really appreciate if the speakers can keep things on time. So without further ado, I'd like to move on to our first speaker, who is Professor Gretchen from Curtin University, and she's going to talk to us about the machine learning techniques and how we can get cheap sample return from Mars. Welcome.
Thank you very much. So I'm really pleased that Martin set us up for the transition from biology to Mars earlier, which is really good. I'm going to argue that we already have sample return from Mars, and so I think LifeSprings needs to go absolutely 100%. But I'm just going to say that we've already done. So what I'm going to actually talk to you today about is how we have used machine learning to find the source crater, or the place on Mars, the location on Mars that the Martian meteorite NWA7034, which is also known as Black Beauty, is the most unique Martian meteorite that we have. And I'm going to show you, or attempt to convince you, that we think that the crater we think is the source crater from whence it was launched off the surface of Mars is indeed correct. So, very brief overview. I'll give you a little bit of background. I'm going to kind of do this in the way of, it's kind of a mystery, and we have clues from the rocks and we have clues from the planet. So I'm going to go through those clues so that we can try to figure out how to get to an answer. Then I have to talk to you about the importance of machine learning and how machine learning is the reason we can do this. And then we'll talk about how we define the source crater. So I have to start you with meteorites. I am a meteoriticist by trade. Since moving to Australia, I've branched into looking at Martian meteorites and looking at the surface of Mars more. But my background has really been in meteorites from all over. So the story of meteorites from Mars is quite interesting, because up until about 2001-ish, we didn't have very many. But we had decided, we had speculated and hypothesized that they were from Mars, because this group of meteorites was very different than any other group of meteorites we had, primarily because their ages were so young. Most of the meteorites that we have in our collections, they're around 70,000, they're all in the 4 to 4.5 billion year old range, which it means they're really, really old. Some of them have never melted, some of them have completely melted, but they're all still very, very old. These rocks came in, they were definitely melted rocks, basaltic rocks that had ages that were in the hundreds of millions of years. And the only way you can do that is to have an active body, an actively hot body that's generating lava, until very recently in solar system history. So we started, the hypotheses were all around, well, where could this come from? Would it be Venus? Would it be Mercury? Would it be Mars? Would it be, and so Mars became the default option. And so what that meant was we kind of thought, okay, well, these come from Mars, and then we weren't sure how do they get from Mars. So maybe that's not it. But then it turns out that when rocks are lava and then they cool off, sometimes they get little gas bubbles stuck in them. The gas bubbles don't go away when they get launched off of other surfaces. We can measure the gas inside those bubbles. When we did that, the scientists found that the air inside of that gas bubble was exactly the same composition as the air that was measured by Viking while it sat on the surface of Mars. So we're really quite sure that these come from Mars. What's been interesting is that over the last 20 odd years, we've gone from a total of 15, which is how we started talking about stuff, to we're now sitting at around 350 different pieces of about 188 or 190 specific faults. So what happens is meteorites hit the earth, or as they're flying through the atmosphere. You may have seen this recently in the Queensland fireball. It exploded. That's causing things to break. So it's a single rock that's breaking apart. So we get lots of pieces. So that's what I'm talking about there. The other interesting thing about most of the Martian meteorites that we have, and you can see there's an exponential discovery of them, is that we have only witnessed 5 faults, and 4 of those were witnessed before 2000 or before 1994. So the most ancient Martian meteorites we have are the few that we've actually seen to fall, and there's only been 1 since then. Martian meteorite types. So originally they were called the SNCs, which stood for sugartites, nachtlites, and chasignite. And this is when there were only about 15 of them, and the majority were the sugartites. And the sugartites were primarily basaltic in composition, so that's lava rock, just like you see in Hawaii. And this, over time, has exploded into a whole bunch of sub-definitions that are just depicted here. This is from Arya Udry's recent Martian meteorite review paper from 2020 that's really got all of the things that we know about Mars based on what we can see from the rocks themselves. In 1984, the Antarctic meteorite search team from the US brought back a rock that they very, thoroughly named ALH-84001. ALH-84001 is probably the reason any of us here are doing astrobiology. So ALH-84001 was the rock that came back, initially misclassified as a rock from Vesta, later turned out to be a rock from Mars, and then was purported to have or had evidence that was best defined as being brought about by biological activity on Mars. So there was, some aromatic hydrocarbons, there was magnetite, there were these very strange carbonates that had these rosette fields or things to textures, and then there was also this tiny little what we decided might have been a nanofossil, which still I think the ruling theory is that it's not a nanofossil. So that's where we kind of sat for a while, and then in about 2012, so the first year I got here to Australia, this new meteorite called NWA-7034 was presented at our Meteoritical Society Council meeting. And this meteorite is H here, it's a regolith breccia. It's the only Martian breccia. All other Martian meteorites that we have are whole rocks that have been shocked quite heavily due to impact processes, but they are recognizable as igneous rocks. NWA-7034 is the only rock that is brecciated that we know has to come from a heavily impacted area. And when it was first recognized, it was thought that it might be from the moon because it resembled the moon rocks that we have. So that's been a really interesting ride. So NWA turns out to be a really, really important rock. All of this is some very brief background to say Martian meteorites exist. They sample a whole range of different compositions and geologic processes. And as a geologist, what we're looking for is we're trying to figure out what's going on Mars. So for instance, when we do geology of Mars, we do it primarily remotely. So we have these rocks in our hands here, and then we go and we send rovers and we send orbiters, and we get lots and lots of data. That helps us build up a whole bunch of different data sets. So we have elevation, we have, this is CRISM data overlaid onto an optical image showing compositional information in the Jezero crater delta. This is a geologic map that's been created. This is the optical imagery. We can get a rover on the ground and it can start roving around. So this is Perseverance's view of where it's going and driving around with Jezero. And part of that, as Martin pointed out, is that Perseverance rover is actually going and collecting samples and catching them as it goes. So that in whenever 2030, whatever, JPL's little Martian sample return weird rocket is going to be popped up into the air and lit up like a Roman candle to bring samples back, and I'm really excited about it. But I argue we already have sample return. So how do you find a source crater for the Martian meteorites? Well, it turns out ages. So we can measure ages on Earth in rocks really, really well. Turns out we can, using different geochemical mechanisms, radioactive decay, dating schemes, we can get different kinds of ages that date different processes. We also can get at chemistry, very, very detailed chemistry. We also can analyze things until we're glued. I'm going to focus right now on the ages because that's where a lot of this, the clues start by getting the ages right. So this is a plot of crystallization age versus ejection age. So ejection age is the age at which the rock was launched from the surface of Mars, got out of its very thin atmosphere and started to collect cosmic rays. So that's based on a cosmic ray age. And if we look at this for all the meteorites we had at the time, you can see that we can use these to help us kind of pinpoint certain things. So from this, we can say that there are very specific events that launched these rocks. We can also look at the different types of crystallization ages, and that tells us when was that rock formed. So we have that kind of crystallization and ejection, and there's some other ages we can talk about as well. For NWA 7034, this rock has been studied very, very extensively, and you can see just from this one rock all the different processes and all the different events that we can actually figure out by looking at the different parts of that rock. So we have crystallization ages that are quite old, we have some resetting ages, we have some ejection ages. We'll be coming back to all of this, so just giving you the context of how this works. Now, we can also determine ages from space. So if we look at the surface of a planetary body, we can start to understand differences in ages that help us pinpoint things. So this is Mars. This is the elevation map of Mars. White is very, very tall, and dark blue, indigo is very, very deep. So Hellas crater is really, really deep. Olympus Mons is really, really tall. What you can see is that the northern hemisphere and the southern hemisphere are quite different. So the northern hemisphere is lower and it doesn't look very cratered. The southern hemisphere is very cratered and is much higher. Now, we can use that to establish relative time frames. So if over the course of the solar system, you've got bodies that are smacking into the surface of Mars, it's going to leave impacts. Now, if you have a volcano that comes along and erupts, or you have a really big resurfacing event, you'll have a flat surface again to start counting. So you basically have a clock. So if you have craters on the surface of a body, you can use that to help you figure out time. And because we've been to the moon and we collected samples in situ, we have a way to calibrate that. And by doing that, we come up with this scheme called crater counting. So some graphs, but basically what you want to look at here is that crater diameter on the bottom on this right-hand side, crater diameter on the bottom versus cumulative crater frequency per a specific area. And when we look at that, if we look at surface age versus this cumulative crater frequency, we get this nice curve that says over time, this is how many craters of a certain size would have formed over time. So the green box is actually a three-dimensional version of that green line in that isochron map. So basically, we can do this. If we narrow down an area and we can count craters, we can actually convert that to an age. then we can make some comparisons. Now, the other thing I want you to know on this is that if you look at the very small crater sizes, so the smaller your diameter, your isochron numbers go up. So if you want to get to really young ages, you have to be able to count small craters. Okay, so here's our stuff. We have got from the sample, we've got a crystallization age, we know when it formed. That is very close to, within error, the equivalent to the surface age, as we would measure it using craters. We also have cosmic ray exposure ages, which we can equate to the age of the crater. So now all we got to do is actually go through and figure out how to match them together. So this is where the machines come in. Machine learning. We used a convolutional neural network to allow us to actually recognize objects. So trying to have a computer recognize circles turns out to be really hard. Computers need a lot of training and a lot of education. And so we use this convolutional neural network. We use an algorithm called YOLO, which is you only look once. And what it is designed for identifying multiple different features in images. So their advertisement is you can recognize a horse, a dog, and a person in the same image. And it does it. And it does it really, really well. And as it has progressed, it's gone from recognizing big things to recognizing very small things. So we had to train that algorithm to identify what we wanted it to identify on the surface of Mars. I'm not going to go into vast detail on this. If people want to know, I can explain it later. But basically, we had to manually tell it, we had to create a data set that told it what it was looking for. Then we took 2/3 of it and we said, that's what you're looking for. And the other third, we said, okay, did you find what we told you to look for? And we iterated through that until it worked really well. We trained it on medium or with high resolution and super high resolution images. So 100 meters per pixel and 30 centimeters per pixel. We have access to some amazing imagery from the surface of Mars. We applied that to 2 global Mars data sets, the Themis data set at 100 meters per pixel, and also the CTX controlled data set, which is 6 meters per pixel, which means we can see things that are very, very small. The reason this works is that manual counting has been the way to do it for decades since we've been to the moon. There is a manual database for both the moon and for Mars. For Mars, that hand-counted database is 385,000 craters greater than 1 kilometer in diameter. Now, like I was saying before, the smaller your craters, the more they are. Well, that's an exponential. So the smaller your craters go, the exponentially large the number gets. What this allows us to do is find a way to interact with that data set more quickly. And I'm going to go super speedy. This is the manual data set as counted by Robin. So this is all craters on the surface of Mars bigger than 1 kilometer. Now, nothing's perfect. 2 different people counting the same area will count differently. So even if our data set doesn't count exactly the same, if it's in the ballpark, we're close. So we're looking for around 85–80%. Now, I just want to throw this one out here because this is where the machine learning comes in really handy. So originally, for the amount of time it would take to analyze a single image that was 26,000 by 17,000 pixels was about 3 minutes or about 15 minutes. We knocked that down to around 1.5 minutes or 2 minutes. And that allowed us to actually figure out how to use it over the, it's fast. It's fast. You need a cluster of GPUs, but in order to analyze the entire CTX mosaic, which is something like 9 gigabytes or something, that doesn't sound very big. It's a really big image. It only took 24 hours on the GPU cluster, and that's counting craters down to 100 meters per pixel, or 100 meters in diameter. So this is getting very, very different. Now we're able to look at things in a much different way. This is just very quickly to show you that, especially over the area less than 1 kilometer or between 1 and 5 kilometers, the automated version versus the manual counting is really, really close. And we can talk about those error analyses later, but in effect, we're within 85%. So compared to, if we're comparing to the manual data set, we're doing as well, the computer is doing as well as 2 or 3 different humans counting the same area. So here's the, just for reminder, here it is, manual data set. Here's what it looks like when you do it with a machine, 94 million craters. This is now presented not as individual craters, but as crater density. But it is showing you for craters down to 100 meters in diameter, the density of those craters. I just want you to look at the difference. You can see, you can recognize Martian things there. But with this one, you start to see some of that topography coming out that you might have seen in the elevation now. And then we can plot it. If we look at only the very smallest craters, what we're looking for is areas of interest. So this is only 89 million craters. So you can see how that exponential works for the smaller crater sizes. This is what happens when you look at just the small ones, and then if you put them into a red, green, blue, you can actually map them quite differently. I know I'm running really late. I got 5 minutes. Okay, we're going to speed through the source crater. All right, so on Mars, any crater bigger than 3 kilometers is a potential source crater. That's 70,000 craters. That's way too many, right? Using our data set, if we find stuff with significant rays, that makes them young, that fits better into our cosmic ray exposure age constraint. That knocks us down to 19. So we got 19 craters that we think will work. Here's the paper. This is the paper we've written. That's the QR code if you want to go get the paper, but I'm going to skip that right now and we're going to keep going. So back to our crystallization versus ejection. We talked about that. So we're going to take a look. This is NWA. That's what we're looking for. Here are some ages we're very interested in. So we're looking at oldish. We're looking for the different types of ages that we want to find something on kind of an old age, but we also can see that there's a young ejection age, and that it's got some younger ages in it that say things about what kind of environment it might have been in or around. Okay, we got crystallization, we got reset, we got ejection. This is the important bit, chemistry and magnetization. So what we can see is that NWA is a little bit of an outlier on a chemistry map, potassium versus thorium, and also on magnetization. So if we start looking for that, so here's our 19 potential craters. The top layer here is magnetic intensity and magnetization. So if we are looking at all these craters, what we're looking for there is we want to find something that fits with that previous graph on magnetization. And we also on the bottom have a potassium and thorium map. Now, the resolutions of these maps are completely huge compared to the resolution of the data we can get for the composition or for the craters. But what this does allow us to do is it allows us to pick craters or actually get rid of craters that are in absolutely the wrong areas. So by doing a couple of things, first we knocked out everything because of the age of the rock, which is in the billions instead of the millions, hundreds of millions. We kind of knocked out everything that we thought definitely it can't be, which are most of the northern hemisphere things. And then based on the chemistry and the magnetism, we picked a couple that we thought were the best options. And so what we're looking for is kind of the green options are the ones we ended up going with because they matched most of the things. So even though the potassium and thorium is pretty off, the magnetics is very good. So these are the two that we ended up choosing to work with. We're going to go look at this one that we named Ratha, and it's next to another one. So this other one is not that name, Gaza. So Gaza was the small crater inside the bigger crater. We were looking for a small crater and a bigger crater because of an age constraint. So these two both have that. We were looking for an age constraint that indicated, because of the shock zircon, that it might have sampled the central peak of an impact crater, which is where you get a lot of really highly shocked material. And so this is where we ended up. We took a look at both of these, and the ages are determined in these ways, and we've got some age constraints there. We ended up picking this Karratha crater as the launch crater because of all of these things. And this is the story. You have this small crater formed first. We have a secondary crater that actually comes later, and that's where we get an argon age. That's a later reset age, and has materials that got dropped into it. And then Karratha is smaller. It sits in down here, and it's the one that actually allowed the meteorite to get launched off the surface of Mars. And that is the story of how Black Beauty got from Mars to Northwest Africa. I want to thank you all very much. I'm sorry I've gone long. I hope I can have a couple of questions. Australian Research Council funded a majority of this, along with time on the Posey Supercomputing Center and Curtin University support. Thank you.
Thank you very much for the interest. Does anybody have a question?
Can you put that code back up so I can see that?
Yes. All right.
So can you do this with every one of the Martian meteorites we have?
We've done it for that meteorite, and we've done it for this other set of sugatites. They're called depleted. We've found, we've pinpointed the most likely source crater for that group as well.
And if you had to know the amount of accessible boron there is on the surface of Mars, which of these rocks would you look at?
That's a good question.
We have a donor who's given us, bought for us 5 grams of Black Beauty, and we're trying to decide whether to dissect it.
Oh. So the thing with Black Beauty, which is awesome, is that it has so much stuff in it. So it's got rock class, it's got mineral class, it's got just stuff. If you're looking at, I don't think I've seen a boron map of Mars, so I think it's worth just checking it. Let's talk about this, because I have to think through.
It's a regular, right? It's
a regular Gretchen, so it's
not an individual part.
Yeah. It's had a whole bunch of things happen, and there's shocked stuff and unshocked stuff, and so it's a really big mix of things.
Some of the basaltic sugar types might be better.
It might be, yeah. There have been boron isotope studies done on some of the sugatites, so it might be worth having a look at that.
Just out of curiosity, how many lithologies are in that stone? Do you know?
In Black Beauty?
Like hundreds or 50?
I wouldn't say hundreds, but it's a lot. I mean, it's basaltic and monomineralic and fine grained and aqueously altered and.
Any chunks of rhyolite or andesite?
I'll check we can check Aria's paper. I don't know off the top of my head. Marita.
The last question.
You said, I think, when you were trying to find the source crater, you were looking at the ones that were larger than 3 kilometers, and then you culled it down to just the ones that had a ray. I think you had like 70,000 or something that were larger than 3 kilometers. How did you cull it down to the ones with ray? Yeah,
so that is a step that I went over very quickly. As part of understanding the cosmic ray exposure ages, the ejection age, most of the ejection ages are quite young. And what that means is that you're looking for a young crater. So young craters on the surface of Mars will still have rays coming off of them because the erosion, there's erosion of stuff happening. And so those craters get buried. And so this is, that was the way we kind of narrowed it down, is finding all the sort of really clearly rayed craters.
So were you looking at them visually, or are you just looking for young?
No, we were looking for young, but there was no way to differentiate that until we got the small craters and we could see the rays.
Yeah, okay.
And yeah. Thanks.
All right, thank you very much. Let's thank Professor Gretchen once again. Yes. Oh. Yeah. Did you put the?
Yeah, I think.
Okay, I gotta let you in.
Oh.
Let's see.
No, sorry.
Oh, I don't see it yet, actually. Can you try clicking on the? Oh, yeah, I don't see you. Yeah. I'll just make you a co-host. Okay. Oh, just unmute yourself. Or mute yourself, I mean. You don't need to. We have a microphone. Yes. The words is, can you mute yourself? It looks, except for that.
Yeah.
Sorry for the delay.
Yeah. Okay, great. Oh, good. Just speak loud.
I can do that.
Yeah.
Sure.
Everybody. Next up, we have Dr. Rita from Gretchen University, who will talk to us about the hydrothermal environmental detector.
One of these. Oh, hopefully, can everyone hear me at the back? Yes, okay, cool, thank you. Well, thank you very much for having me, and I'd also like to thank the Benedix people who are the custodians of the land that we're meeting on today. And I'm coming to you from Wajak Noongar Budja from Perth in Western Australia. So I'm going to be talking to you today about the initial, so very early development of a planetary hydrothermal environment detector called FIND. And I'll get right into it. Let's see if it's quickie. No, that's okay. I will stand next to my laptop. Okay. So, yeah, cool. All right, so the key messages of the talk, I'm going to talk about HAPs. So that's this hydrothermal alteration products, just so I don't have to say so many words. So essentially, I'm going to talk about identifying these HAPs, which are indicators of hydrothermal environments. So we're looking for these mineral indicators using remote sensing as indicators of these hydrothermal environments on other planets and also on the Earth. Also going to talk about how we can differentiate between these hydrothermal products that may be generated by impacts versus products that were simply excavated by impacts and deposited on the surface. So how can we tell between the two when we're just looking at this orbitally? And also talk a little bit about, well, mostly actually I'll focus on the development, the method behind how we're developing this FIND, this detector to look at finding these environments on other planets. And I'll show a few test cases where we look at it for the Shoemaker Teague impact crater on the Earth and also Toro crater on Mars. So this is what I'll talk about, but not in this order. But these are the topics. So essentially the method, what are the minerals that we're looking at? What minerals should we find in association with impact craters on Mars and on Earth? How do we find them spectrally? How do we relate this to impact crater thermal modeling? And then these early test cases of FIND. All right, so I probably don't need to provide really the motivation for this audience as to why we're interested in looking at hydrothermal environments. A lot of that has been provided by the speakers that have gone before me today. Essentially, I really like this definition here. I just kind of reworked this from Georgieva et al., but this is a definition that I've found really broad and encompassing about what these hydrothermal fluid environments really are. But the motivation really for looking for hydrothermal fluid environments on other planets is because they can really be some of the earliest evidence for life on Earth is from these kind of environments here. And so when we look for them on other planets, we know that they have the potential for these environments where life could potentially get started in these chemical rich kind of building block environments for life. So this is where we want to try and target, seeing if we can find these environments on other planets and seeing if there's evidence for life having gotten started in other places in the solar system. As you heard from Martin earlier today, so I won't really spend any time on this,
but there's lots of interesting evidence for potential hydrothermal processes on Mars in the form of potential hot spring deposits. So this here is a slightly different to what Martin was showing, that these are the pastorally soil high sulfate rich deposits at Gusev Crater at Columbia Hills. And you saw earlier the opaline silicate nodules that Martin was showing. But it's not just those sorts of environments. There are also other ways to generate hydrothermal fluids through a process that occurs broadly on bodies in the solar system, and that's through impact cratering. So impact craters can make hydrothermal environments through essentially, if you have an existing water table or a cryosphere, a frozen water table, then essentially through delivering heat and through fracturing that crustal environment that you have, and through heating up that water table and giving you boiling fluids and melting and vapor, and fracturing that gives you that permeability for those fluids to circulate and to flow, you can get a short-lived, a transient hydrothermal fluid system. So this is a schematic from Sinski et al. That gives an example of where they believe you are most likely to see evidence of HAPs, of hydrothermal alteration products. So that's those 6 numbers there. So the key thing here is really the concentration of those mineral signatures that form from the alteration of the rock by those hot fluids is really concentrated predominantly within the interior of the impact crater system. So predominantly within the rim, but it is possible to get some evidence of alteration, although less likely can occur in the ejecta, but predominantly you would expect it to occur more concentrated within your crater rim because of this impact cratering mechanism. So what is FIND? So it's essentially a tool, an automated Python-based tool that's being deployed in a Jupyter notebook to search for these hydrothermal alteration products around impact craters on other planets. So at the moment, it's being developed in parallel, FIND Earth and FIND Mars, but they will be combined together. It incorporates multiple satellite sensors and will potentially also fuse those products together. But the purpose of that is to encompass a broad range of wavelengths from visible across to the thermal. So, for example, in the Earth context, it's currently using Sentinel-2 and ASTA. In the Mars context, it's using Themis and Prism, and we'll certainly also incorporate Omega and High Horizon GTX, of course, and onward. So the purpose is for it to include as much wavelength information as possible and fuse it together to capture these mineral signatures. And of course, once you have these candidates, potential hydrothermal alteration product signatures, then you would go to the next step of attempting to verify what particular mineral candidates they might be. And then, of course, you can look more detailed into the astrobiology of it. So what are the potential temperatures for those minerals to form, what potential fluid, what rock ratios, fluid pHs could you potentially be looking at for the formation of those mineral environments. Okay, so getting a little bit into the development of this in a Martian context, obviously with some variation, but on Mars we're predominantly looking at the basaltic rock. So how can we differentiate the kinds of minerals that form in hydrothermal alteration versus other kinds of fluvial alteration settings, such as lake setting, for example, or lower temperature water alteration of basalt? So what we did is we started at looking at all minerals that have been found in association with impact craters on Mars and have been found orbitally, so in spectral data from Mars. And what we found is that there's 17 essentially hydrated minerals that have been observed in association with impact craters. So really quite diverse, a lot more than just the clays that are talked about broadly, but quite a diverse suite of minerals. So here is a subset, and it encaptures most of them. And along the top, you can see the temperature and the colors, temperature in Celsius, and the colors are these broad mineral groups. So you have essentially the clays, the phyllosilicates in brown, you have some other silicates, and then you have your carbonates and non-silicates there. So if we look firstly here, that box there captures most of the minerals that are seen around impact craters on Mars. So the most common ones detected are the iron, the magnesium smectite, so in that black box there, the aluminium clays, so like montmorillonite. Most of the detections around impact craters are those ones there. And as you can see from the temperatures there, they're not very strongly hydrothermal. So you can also make these minerals, they can be formed in lower temperature aqueous settings as well. And in fact, they are on Mars detected in many non-impact settings as well that are more consistent with other fluvial morphologies and lake beds. We also see quite a bit of kaolinite in association that is up there in terms of frequency, and that is more strongly hydrothermally explained. But if we look down here, we would say, well, the most obviously hydrothermal minerals would be these ones, would be like illite, chromite, serpentine, chlorite, for example, because they form at much hotter temperatures. So these would be more strongly hydrothermal alteration products for us to sort of target. We can also consider, I'll just touch on this briefly because I don't think I'll have time, but we can also consider the other end of the spectrum, which is, well, when do these minerals break down? And this sort of comes into the differentiating between impact excavated versus impact generated, is looking also at what temperature do these minerals break down. So where would you not expect to see them if they were impact excavated versus impact generated? So now we've collapsed them into a few more groups that just really behave the same in terms of their formation and their breakdown or their water removal temperatures. And the main thing to get from this really is that the strongest evidence for us to target in terms of what we would say really strongly is a hydrothermal alteration product, if we can detect that from orbit, or it's more likely to be, would be these particular minerals that I've highlighted down here. So things like chromite, serpentines, talcs, micas, fluorite, illite, cristobalite, epidote, those kind of minerals would be for us the strongest evidence of being potentially formed through hydrothermal alteration rather than any other kind of aqueous setting. All right, so how do we find them if we know that that's what we're looking for in association with impact craters? So we go now to the spectrum. So what we want to do is we want to devise a way to differentiate those groups from essentially the target, the basement rocks, the background kind of terrain. So that's kind of your background kind of stuff up the top there. At the bottom, you've got that first group that we were talking about. So that's the clays. And so what we do is we define some key regions that we want to look at. So those colors there, so 5 key spectral regions that we would start looking at to differentiate these hats from other mineral groups. So if you look here, you would say, well, in the A region, obviously there's a strong absorption there. In the B region, there's an absorption, although that's a bit of a tricky region because of Mars's atmosphere. You have to be really good at your atmospheric corrections. In the E region, there's obviously a strong spectral slope going on. So you can compare the two areas. You can also look at, say, our other sheet silicates. So this is where we've got those ones that were more strongly hydrothermal. So the crenite, the chlorite, the serpentine. And you can see the same regions here. So that's the previous, more phyllosilicate clays. And here we have the more strongly hydrothermal ones. So you can see in A, okay, they both have strong absorptions in A, but if you look in B, they're quite different. So this has not got such a strong absorption, but in the B region there, it does. In E, it's flatter, but in E over there, we have a strong negative spectral slope, whereas here it's a bit more of a positive spectral slope. So you get kind of the gist. So the way the detector is built up is through looking at these different regions, looking at the mineral groups and looking at how to differentiate them using absorption bands, using spectral slopes, that kind of thing. Okay, so that's just a brief background to the methodology of building up the detector. So the next question would be, where then do we target? So we know some initial steps of how to target these hydrothermal alteration products and what products we want to target. But where specifically should we target and relate that to our impact theory? So back to that initial image that I showed of where we expect if impact generated hydrothermal system existed, where did we expect those fluids to potentially interact with the surface and leave us with potential hydrothermal alteration products that we could potentially see from orbit? Where would we expect that to happen? So that's where we go back to the theory. So this now is related to some of the impact modeling that my colleagues do that you'll hear about a bit more on Thursday. So this is just a really brief overview of the logic of the ISAIL damage models and how it relates to giving us essentially a zone in which to focus the remote sensing. So the logic here would be that if there is a damage model for the impact cratering, then from that damage, you can infer that that could potentially lead to fracturing. That fracturing then could lead to fluid pathways. And if those fluid pathways are seen to be connected to the surface, then that would give us zones where we could potentially look for orbital evidence of these hydrothermal alteration products. So an example that we're looking at here is Toro Crater, which is known to have had strong evidence of potential hydrothermal fluid system in the past based on previous spectral work that's been done on the central peak. So we're now looking at a different area of the crater. So this is Toro on Mars. There it is. It's a layered ejector crater. So it's got this lovely Lowe ejector around it. It's in the presence of other layered ejector craters. There's one up there. So this now is a zone of radii. So you can see over there. That gives us mostly in the interior and a little bit up on the rim and a little bit outside on the inner parts of the ejector. So here we have 3 prism images, and we're looking now, so this is hyperspectral. We're looking here at the prism hydration index, and you can see that it gives some strong color, which indicates essentially that there's strong blue color means that there's evidence of hydration. This is a typical Mars spectral product that would be used, but I would say it's very ambiguous mineral interpretation based on these initial images, but it shows that there's some strong signature worth looking at. You can also see that there's very strong inter-image differences, which can happen for a whole heap of reasons. When we now look at our one of our spectral composites, it really smooths out that inter-image difference. So you could actually mosaic these and the pixel values align really nicely across each other. And the interpretation here is that we're discriminating between the clay minerals and the other sheet silicates that are more strongly hydrothermal that we talked about before. So what we see here is that there's some interesting signatures. So if we zoom in and have a look, and we're overlaying here on CTX imagery. And so they correlate really nicely with what appear to be these bright mounds near the walls at the base of the inner rim of the impact crater. So this is something really interesting and is our first essentially really interesting detection of potential hydrothermal alteration products, but really this is the first step. And the other observation was up here on essentially correlates with, if I zoom back and forth there, is like this distal edge of one of those fluvial layers of ejecta. So this is really interesting. This correlates more strongly with the clay minerals, which is actually consistent with temperature models. So that was good. This is spectra, lots of work to be done. Okay, if we have a quick look at a terrestrial example, which really is our ground truth. Obviously on Earth, it's a lot more complicated because the target rocks have a lot more variety. So this is pie charts of all the different target rocks that have impact craters impacted into them. That's for English. But you know what I mean? On Earth, this is from an Earth impact crater database published in 2021. So as you can see, there's lots of variety. So if we do one example, which is the Shoemaker T crater, these are the hydrothermal minerals that have been directly sampled. So this is not orbital, this is on the ground, in situ, geologists going and measuring it, boots on the ground. I love that expression. So here we are at Shoemaker. If we zoom in, that's a lovely geologic map. You can see it's got an inner ring and an outer ring. So that's the inner rim. And that's where we see these hydrothermal products that have been mapped. There it is, absolutely beautiful in WA. That purple there is where they have directly sampled the hydrothermal alteration products. And then here is our orbital detection using FIND. So you can see that we've really nicely essentially captured the shape of what they've matched on the ground, but we've also found, ignore the small pixels, that's noise, but we've also found some large spatial areas that may be false positives or may not. There's a lot of work left to do. So next steps are to do a lot more ground truthing and to continue the development, and many more questions left to answer, and to keep comparing this to the modeling as well. And that's all for me.
One question.
Can you just clarify, in the case of Toro or the other craters, the step where you actually identify the specific.
Right, yeah. So that would be the final step, absolutely. So in this, I only spoke about essentially mineral groups and detecting them via mineral groups. But once we have identified targets, so the FIND detector at this stage is identifying mineral groups to identify high priority targets that have those HAPs, so those hydrothermal alteration product groups. And then from that, the next step would be to actually do the best fit mineral and mineral mixing ratios and go into that next step. But I didn't mention that at all. But that would be the next step, absolutely, to provide the stronger evidence. Yeah, no, that was developed separately. Yeah.
All right, let's thank Dr. Riddell once again.
Okay, let us do start. It's doing something weird. I don't know why. Let's try this again. Yeah, it's fine. Yeah, I think I fixed the audio. Let's see if it works now. You have pretty plug this in.
So if I just share, can we just go through that one? Is it easiest?
I think it'd be easier just to like plug in. Can we do that?
Yes, that should work.
Yeah. You know where it is.
There we go.
It's good. Let me just check on mine here. Yeah, that looks good.
All right, next up, we have Dr. Ben from UNSW, who's going to talk to us about the search for habitable exoplanets. Take it away.
Great, thanks. So yeah, the solar system is great. I will, concede that my favorite planet is in the solar system. But there's 100 billion other stars we could be thinking about just in our galaxy. So I'm going to give a little bit of an overview of exoplanets and what we know and what we wish we knew, and what we might know someday. So I know this is the last time before coffee, so we'll just start with the conclusions, and you can kind of tune out for a little bit if you're hitting the end of your attention span. But we found lots of planets. There's a lot we wish we knew about them. Understanding habitability and ultimately life means understanding their host stars. That's really, really important. And large-scale transit surveys, we'll talk about what those mean, have fundamentally transformed our field over the past 10 or so years. So with that in mind, let's jump in. This first question we might have is, how do we find planets? And here, because this is an astrobiology workshop, let's limit ourselves to how do we find potentially habitable planets. Well, there's a few different ways. The original way that was the most successful for the first 15 or so years of finding planets, from 1995 until about 2010, was through spectroscopy. That's not, oh, let's do that not now. Cool, okay. This is the spectrum of a star, this is the spectrum of the sun. And we see lots of narrow features in there, absorption lines in the spectrum of the sun. And those give us a reference of the velocity of the star through the Doppler shift. If the star is moving away from us, these are redshifted a little bit. If the star is moving towards us, they're blue shifted. So we use this for all sorts of galactic structure, understanding the motion of stars around the galaxy. But if a star has a planet, as the planet goes around the star also goes around the center of mass of the system. And so we see some motion here. And if this GIF or GIF, as your preference, plays, there we go. As we see that this motion, we see all the spectral lines for the star move towards and away from us in a periodic way. So the size of the shift here tells us how massive the planet is, and the period for the shift tells us the orbital period, the year on that planet. Only problem is this is pretty small. Planets themselves are not particularly large in the grand scheme of things. And so the motion here on the y-axis here, I'm loath to move away from the microphone, so you won't hear me as well, but I'm gonna do it anyway, is the motion of the planet around the star. This motion is, in this example, several meters per second, which is faster than I can move right now. But for a planet like the Earth, this motion is 9 centimeters per second, which is quite a bit slower than I'm moving. So we need to find, get back to the mic here, a planet change, or star changing its velocity over a year from 6 months, from one side to the other by 9 centimeters per second. So think about what that really entails. That's a hard job. That's why they pay us the big bucks. This is challenging when we look at the real data. When we look at a spectrum, this is what it looks like. And so, things move around, but everything's quantized. We're looking at, detectors with pixels. And so 1 pixel of motion is about 1 kilometer per second for the star's motion. So if you're measuring, a meter per second shift, your star's light has shifted by 1/1000th of a pixel. 10 centimeters per second is another, zeros beyond that. And so this is hard. Planets are small is the fundamental limitation here. For Jupiter, this is 12 meters per second. This method was really, really powerful for finding Jupiter-sized planets. We can get down to 5 or 10 Earth masses pretty robustly these days. To get to 1 Earth mass, to get to these rocky things that might have, a solid, surface like the interior planets of our own solar system, we're not quite there through the Doppler method. So this is hard. And this is also something that you need really large telescopes, these 10-meter class things that a few universities and a few collaborations have access to. So this is a field that was really just, led by small teams working at the right institutions. This all changed in about 15 years ago now, in 2009, with the launch of the Kepler mission. Kepler was a phenomenal telescope in space, about 1 meter across, and it did one thing really, really well. It only did one thing. It's not like it did a bunch of things poorly. It's observed a field about as big as your hand at arm's length, measuring the brightness of all the stars in that field about every 30 minutes for 4 years. And it did that because occasionally these stars have planets, and occasionally these planets are just lined up, this laser pointer points. Maybe not. You can see when it's in the upper corner there, these planets are just lined up so they pass along your line of sight between Kepler and the star, cast a shadow on the surface of the star, so the star appears a little fainter for a few hours. See that once, it could be anything, but you see it over and over and over. The star gets fainter periodically. The size of these dimming events tells you how big the planet is. Again, the spacing between them tells you how big your planet is. You might think just look for the planets or the stars with these little curvy lines around them based on the star's conception, but you don't see those. So there's three problems here. The first is that transits are intrinsically rare. You need that orbital alignment to be just right. If the planet's going around like this, you never see it. And just statistically, that's unlikely. For the Earth, it's about a 1 in 200 shots, which means if every star had an Earth, you'd have to look at 200 stars just to find one. Conversely, if you find one, you know that there's a lot that you're missing. You can do statistics that are pretty robust pretty quickly. So because these things are rare, you need to look at a lot of stars to find a few transiting planets. That's why we had this big field, hand at arm's length. You know, there's something like 200,000 stars that are observed by the Kepler mission. This is just one image from Kepler. There's a documentary that described it nicely back in the day. So lots of stars. Second problem is that transits are infrequent. All you get information about is when the star passes, when the planet passes between you and the star. For the Earth around the sun, that's about a 13-hour-long event once per year. So then you have to wait 364.5 days where you see nothing. So a lot of the time, even if you had a system with a planet around it, you're wasting your time looking. So to find it, we need really continuous monitoring at a high cadence. So for Kepler, this is every 30 minutes. Some stars every 1 minute for 4 years. And the third problem is that transits are small. For the Earth, the signal that you measure is the relative areas of the planets, how much of the shadow from the planet across on the surface of the star. So it goes as R squared. Earth is about 1%, 10 to -2, the radius of the sun. So the transit signal is 10 to -4. So that's 100 parts per million. So you might have seen it, you might not have seen it. There's a small dot on the sun there. That's the actual relative size of the Earth to the sun. You need to be able to find that. You might not have seen it. If you missed that one, there's a blue square right here, which is also the relative area between that square and the slide here. So if you're feeling cocky about finding that one, you see that one. This is a hard measurement to make. And we also need to understand star spots. The star itself varies at much more than the 80 part per million level for the Earth. So we need to understand what the star is doing really, really precisely as well. So Kepler did this for 4 years, from 29 to 2013. Wonderful telescope. I did all my thesis work on data from Kepler, so I'm biased. TESS launched in 2018. TESS is the Transiting Exoplanet Survey Satellite. You can probably guess from the name what it does. It's kind of an all-sky version of Kepler. Here are some stats that there'll be an exam later. We'll go through. But the takeaway is, millions of stars at 30 minutes or faster cadence, 10 to 20 parts per million noise with really small telescopes. Kepler is 85 centimeters, is kind of here. TESS is 10 centimeters, which is here. You're asking an American about the metric system, so but it's not big. And so TESS is cute. Let's see if we can get this to play. Because it observes the whole sky over a couple years. It has this nice field of view where it's about 20 degrees on one side and 90 degrees, 4 cameras stacked on top of each other. And so you'll see it here that over a month, it observes this field from the equator to the pole in the ecliptic plane, just like that. Beautiful. There we go. I always feel like I should play this at 1.5, then I forget about that. There we go. So it observes that for a month, and then it steps by about 30 degrees and observes another field, and then over a year continues to do this, observes the entire hemisphere, flips over and does the south. So every star gets a month of observations, and square detectors on a spherical sky means that these all overlap at the poles, and so you get a year of continuous data at the northern and southern ecliptic poles, which also happen to overlap with the continuous viewing zone for James Webb. It's the part that's easiest for JWST to observe. So there we go, gets the whole sky. And we got a lot to talk about in 25-minute talk, so believe me that it does the other hemisphere too. There we go, it's flipping. Good job, Tess, keep doing your thing. So it's been observing for, it's in year 6 now. It's been doing this really, really well. And so the end result is that we know about thousands of transiting planets all over the place in terms of radii and orbital period. Lots of planets as big as Jupiter. You see my cursor if we do this? Yeah, there we go. Planets as big as Jupiter are rare, but not vanishingly so. And one of the things we know is that smaller planets are really, really common. That it seems like the most common size of planet is about 2 Earth radii, something we don't have in our solar system. But they're all over the place elsewhere. Planets Earth-sized and smaller are common. Especially in short periods, as we move further out down this way, this is observational bias. These planets are harder and harder to find. And so, 365 days, 1 Earth radius is really empty here. That's probably because those are hard. But we know larger planets at a year are common. We know Earth-sized planets at shorter periods are common. You can extrapolate and say probably Earths are common. Other things that multiple planet systems are common. These are colored in kind of cutesy ways where multi-planet systems, the shapes are the number of planets, like 3 planet systems are triangles, 4 are squares. It's cute. And so, multiple planet systems are super common as well. But there's a lot we don't know. That's what we learn. From the size of the dips, we learn about the radius. If we can do Doppler observations, we get the mass. That's what we know. There's these beautiful artist conceptions when a planet gets discovered and announced. This one has oceans on it, and clouds and all this, wonderful surface features. Be great if we knew that. This one's, I love the artist's conception trick of making the planet very dark and shadow as the, pointed, we don't know anything about this planet. And that's accurate in most cases. We don't know anything about these planets. And so, what can we learn about these systems now and in the future? We get the mass, we get the radius. If we have both of those, we know the density of the planets. That's important. Density tells us about composition. It tells us about could it retain an atmosphere. There's a lot of really useful things to know about density. We know the orbital period. And so from how bright the star is, how hot the star is, we know something about a temperature. That's an equilibrium temperature, it's ignoring an albedo. So, Venus and Earth would be about the same temperature under, if we discovered an exo-Venus and an exo-Earth. And if you have transits, you observe at multiple wavelengths, as we heard earlier today during transmission spectroscopy. That can give you some information about atmospheric composition. We can't do that for rocky planets yet, except in really, really, truly exceptional cases. But for giant planets, we're learning quite a bit about their atmospheres. So this is the good. These are things that are useful for us. These are things we might want to know that we don't. What's on the surface? Yeah, if we can rule out an atmosphere, we could say what's not on the surface, probably. But we don't really know what's on the surface in any case. What about magnetic fields? Those are certainly important for Earth. We have genuinely no idea for basically any exoplanets. Are there oceans, plate tectonics? No answer, no hope to have an answer anytime soon. What's the obliquity of the planet? That's probably important. Is it aligned? Is it tilted by 23 degrees? Is it like Uranus by 90 degrees? That's a hard observation to make. Does it have rings? Does it have moons? If planets transiting, we know a little bit about the eccentricity, but we don't really even know the shape of the orbit. Is it circular? Is it very, very eccentric? These are things that we would like to know, but are hard. And things that in most cases we don't have hope to measure with current data. So, for an example, if we found a system that looked just like this, first off, we wouldn't measure this. Mercury would miss under almost every detection technique. It's too small. Uranus and Neptune are too far away to really have any detectable signals. And so our hope are the 5 middle planets. Mars are probably missing 2. Venus and Earth would be really struggle to distinguish between the 2. We would say there's 2 habitable planets in the solar system. I don't know about you. One feels more habitable to me, but we wouldn't have the data to say that for an exosystem. Jupiter we'd find, Saturn we'd find, they would look very, very similar as well. There's lots of things that planetary scientists would say those are different. Yeah, they're Jupiter-sized planets for us. So we know a lot. We know a ton. We know that planets are common. Most stars have planets. Probably Earth-sized planets are super common, but there's so much that we don't actually know about these. One of the things that is really fun from my perspective is, and since we're here talking about astrobiology, let's think about habitability a little bit. Basically, as soon as we started looking at stars, we started seeing signatures of activity. One of the first things that, when we had the first telescopes 400 years ago, Christoph Scheiner pointed the telescope at the sun. I cannot advocate for that for you to do, but we learned a lot. And one of the things he found right away was what he thought was a planet going across the star. And we now know, people knew at the time too, he looked at the sun too much, knew were star spots, sunspots. We see them moving along, evolving. Over the past hundreds of years, we've gotten quite a bit of evidence from the sun about the sun's magnetic field through sunspots. They come and go over about 11 years. They evolve on several month timescales. There's a lot we know about the physics of stars from observing sunspots and stellar flares as well. When we look with Kepler and TESS, this is exactly the same story. There's a lot we can learn about the stars that can inform us about the habitability potentially of planets around them. So that eventually when we want to look at these planets with something like James Webb or its successor, the Habitable Worlds Observatory, I think it's called these days, we know where to look. Where are we wasting our time? Have really no hope to ever see some sort of planetary atmosphere? Where are our best places to look? To understand that means we need to understand these stars better and understand what is possible. And that's what our group does. So we'll spend a few minutes talking about what we want to know here. First off, super young stars, early in the sun's life, sun-like stars that are less than 100 million years old, are extremely active. We see variability because of star spots at the 5% level. The sun gets, over a few days, 5% brighter. The star gets 5% brighter or fainter over a few days. This is a factor of 50 more than the sun does. And we see different features here. First, this periodic signal, that's the star spot signal. So that's a dark region on the surface of the star rotating in and out of view. The size of this tells you something about the size of the spots, convolved with its contrast. We see these sharp features up here, which are stellar flares. And so ejecting off some material. And here there are still planets. This is a Jupiter-sized planet. It really gets overwhelmed by the spot signal because the stellar activity signal is so large. You can say the same thing here for another. There's a 20 million year old star and a 40 million year old star. You see the same spots and flares. And there's a planet in here, but I couldn't even tell you where it is. Yeah, good luck. So one of the things we want to know is, what do the surfaces of these stars look like? When do they change? How quickly do they change? And how does this affect planetary habitability? Stars are kind of like dogs. I got a puppy during the pandemic, and so I'm obligated now to tell you about her. This isn't my dog, I'm sorry. When stars are young, they're like a puppy. They're very active. You really, can't tire them out despite how much you want to. And they're just, always giving off energy, moving around all the time. And as they get older, still not my dog, sorry. They calm down quite a bit. And so stars do the same thing. They begin rotating very rapidly. Over time, the stellar wind, analogous to solar wind for the sun, is carrying away the momentum, causes the star to slow down. Magnetic fields become weaker over time as a result. And so one of the things we want to find are stellar flares. These flares have very characteristic shapes. You see this all the time, kind of rapid increase in brightness as the material gets ejected, and then a slow exponential decay as it spreads out a little bit. Looks like this. This is the it's called Fred signal, fast rise exponential decay. All astronomers love acronyms at all times, but we know just what they look like, and so you could probably guess, especially if you were here 2 talks ago, this is an opportunity for machine learning. We can train something to find these directly in the data. Machine learning used to be the future, now it's the present. And so we have lots and lots of data, 1 million stars, data point every 30 minutes. Some people have had the good fortune to be, as PhD students tasked with identifying flares in data. And so we had a nice training set to use. And so lean into that and train an algorithm to find all the flares. Turns out it works really well. There are various heuristic-based methods, just identifying certain outliers, this many points, some sigma above some mean, that have been used for a long time. They work pretty well on, supermassive flares are easy to find. Smaller flares are harder, but neural network finds those really nicely. And so just as an example, let's see if this plays. Yeah. So we just essentially slide a box through every data set and ask it, is there a flare in here? And return some probability that there's a flare in there, which has two benefits. First, we didn't have to do this by eye. And secondly, then we have some sort of probabilistic, or something that can be turned into a probability if there's a flare here. So we don't need to do any sort of cut by eye of only take these certain things. If there's some events that are low probability, we can kind of statistically account for those rather than needing to make some decision along the way. 10% probability flares are as good as 100% probability flare in some sense once you can account for all your biases. And so when you have thousands of flares, this is an example of a young sun-like star. We can do statistics on that and see how often these flares occur, what are their energies, what are their rates, how do they change over time. And one of the takeaways is that, not a surprise, low mass stars, M dwarfs, have much larger and more flares, more energetic flares than solar type stars. The coloring here is the amplitude compared to the brightness of the star. So bright yellow means the flare is half as bright as the star itself. So you're adding a lot of energy into the system. And other colors are in between 0 and 50, but we're talking, many 10 to the 30 ergs. We use CGS, I'm sorry. I don't know what an erg is either. For young stars, there's not a relation between flares and spot phase like there is for the sun. We know for the sun that you're much more likely to see a flare when you're near a star spots. What this probably means is that there's spots all over the place. Then when we see a star varying by 5%, it's not that it's going from 0% covered in spots to 5% covered. It's going from 50% covered to 55% or 70 to 75%. In fact, this level of insignificance of variability is consistent with about 80% spot coverage. So most of the star is covered in some sort of magnetic signal overall. 80%, the sun is 0.3%. So these are really dramatically different than the sun, even though it's a sun-like, stellar abundance, stellar mass object just 4.5 million years ago. This is just examples of spots. We'll skip on that. It turns out that 80% filling factors, it seems like meaning spot coverage, are common on young stars. Through different groups have tried to understand this through different data sets, including looking at spectral features, really temperature and magnetism sensitive spectral features like titanium lines in the surface of the star, and have found that 80% is not an unreasonable number. So it seems like genuinely this is true. These stars are about 80% covered in star spots. And over time, that calms down to the 0.3% we see for the sun today. We also see that sun-like stars at any age don't have regular superflares, which is probably good. Flares are important, right? You need some sort of, probably, I'm going to speculate a little bit, but some sort of UV something, to use a technical term, is probably important for some of the reactions that might be important for life at the start. This is my understanding of astrobiology. But too much of it can destroy your planet's atmosphere. That as you have more and more UV, you're stripping off the hydrogen and all the other good stuff in the planet's atmosphere, and you can end up with just a bare rock. So there's some middle ground that's important. What that middle ground looks like, we don't know from a stellar physics perspective. But for the sun, we don't have these giant flares that are many percent of the star's brightness at any age, even the youngest ages. We can see that flares on low mass stars significantly affect planet atmospheres. That when you use models of UV emission, how that might affect the planetary atmosphere, these generally don't account for flares at all. When you put in realistic flare rates, at the end, you have about a 10% difference in the amount of atmosphere that's retained at the end. So if you're not including flares, you're overestimating your atmosphere at the end by 10%. And this might be a conservative estimate. We're using a flare model that people have used for a long time, based off the sun, where flares are about 9,000 Kelvin. This might be extremely conservative on low mass stars. For tests, we have a single wavelength, and so you see a brightness, you have no information on the temperature. There's a team at UNC in the States, North Carolina, doing really cool work looking at stars simultaneously with tests, but in a much bluer filter. And so they have data from what they call the every scope at the same time as tests. And so we have information in two filters. You can infer the temperature of events when you have two different wavelengths if you're observing it. And they see that some of these flares can be as hot as 40,000 Kelvin. And so 40,000 Kelvin means it's, based off the energies we're seeing, that can increase the UVC flux by, it says 100 to 100, which is a very narrow range. It should be 100 to 1,000 times at the top of the planetary atmosphere. 1,000 times, that matters quite a bit. And so what about the most sun-like stars? Stellar age too. I've gotten I should be wrapping up here, so I won't say too much here other than even the most sun-like stars, the sun seems like an outlier. When we take stars and we think they're similar in rotation period and composition and mass and everything else we can think of, these stars are still more variable than the sun. Here's a couple examples where we see variability at kind of the 0.8 or 0.3% level. The sun is at the 0.07% level. The sun is really less active than other solar-like stars. That is kind of at the bottom 10% or so of variability of sun-like stars, which means 90% are more variable than the sun. What does that mean about our own solar system and why we're here around the sun? I don't know. It's one data point that's difficult to draw conclusions from. But maybe it's something as we find more stars that we can look for life around. That's perhaps a really important data points. If you want to read more about this, I like titles that are very descriptive, and this one is. Just to wrap in my last minute, 30 seconds here, the exciting thing that's coming up next is the Nancy Grace Roman telescope. It has a great story. I asked me about it at dinner tonight, but it's going to launch in late 2026. It's a Hubble-sized telescope, 2.4 meters, and one of the things it's going to do is a survey of the galactic bulge that is a really, really solid transit survey, except with a 2.5 meter telescope, very precise, very, very big image, much, much wider than the Kepler fields in terms of stellar density, in terms of number of stars. And so it's going to be something like 100 Keplers at once, where we found 5,000 planets with Kepler. This thing is going to find something like, if you add up all of these numbers, 60,000 planets, and something like 20 million stars in its field of view. So anything that Kepler found one of, it should find 10 to 100 of. Anything that Kepler didn't find because they're too rare, it could find a few of. And so it's a really exciting mission for us to find new weird things to explore and to better understand planets across the galaxy. So thank you. I'll leave my conclusions up and I'm happy to take any questions.
Very interesting talk, and I know nothing about astronomy.
Yeah.
So is there any difference between stars at the rim of the galaxy compared to the stars as you go closer to the center? And so should we be looking at different parts of the galaxy then for the stars that we think might be closer to the light?
Yeah, absolutely. When we get closer to the center of the galaxy, stars. So one of the exciting things about Roman is that it's going to look specifically into the galaxy. And so it's going to give us a very different field of view and basically help us understand how do things change as we go across the galaxy. We know that planet occurrence is the likelihood that you have a planet is a really strong function of composition. The more metals and things that are in your disk when you're first forming your star, much more likely to have a planet. So we expect there to be much more Earth-sized planets over there. The converse to that is that also stellar activity is much higher when you have a more metal-rich star. even when you control for age. And so probably there are more planets that are riskier places to be around. There's also some evidence that when you get out into the higher regions, so further higher above the plane of the galaxy, planet occurrence decreases. I think that's not controversial at this point. There's enough data to say that's true, and we don't understand why. So that's something where you get N theorists in a room, you're going to get N different opinions as to why.
There's this plan into the sun
Yeah, that's a good question. We like stellar evolution. Yes, the sun was definitely fainter 3 billion years ago. What does that mean for, maybe that's something about the activity that you're getting a lot more UV emission that helps balance things out in some way. From the stellar physics standpoint, we don't have an answer.
One micro question.
Micro question.
Wondering if it's in terms of like example of the actual wondering if, like obviously they're very small objects, they're kind of wondering if that's sort of scattered free of objects would take up a larger area or like small free objects.
Yeah, that's like a Miller question or like a Centa question more than a micro question. But yeah, so the short answer is it's hard. Not with Doppler or transits, it's just too small. There's one system where we think we found an exocomet, but that's hard. It's much more tractable in very young systems when you still have everything hot and lots of thermal emission from formation of these things. And so there's evidence for early debris disks, so remnants of planetesimal systems around young stars. But once they cool down, we'd really struggle to find an asteroid belt. We can do slightly better with a Kuiper belt, just because it's so much more extended. You use some sort of infrared imaging, something to get things at much wider separations. But the classical 3 to 5 AU thing would be really, really hard to measure around a solar star.
Right, let's thank Ben once again. And that brings us to the end of this session. Since Poole couldn't make it, and we're going to have a slightly longer break, I think.
But I have an announcement.
Yeah, there's one more announcement to make.
Since we have a little bit extra time, I'd like to invite everybody to the stage. It's like a quick photo. Everybody's welcome. The more the merrier. It'll only take a few minutes, and then you can all go outside and have some snacks. So come up. Just be careful. There's fours in front here. Don't trip on them.
What was it?
Welcome. Don't be shy. Why don't you guys kneel down in front? Yeah, just careful of that.
Laura. All right, 1, 2, 3.
Because I'll take one and hand it together.
Albert's very useful.
Hey, cheese. Cheese. That's very enthusiastic. Okay, we're good. Thank you, everybody.
Oh, thank you. Back when we made them.
Are you,
Yeah, I did. Yeah, it's better.
My name is Jeremy Tran. I'm a student of Dr. Albert Fahrenbach, and today it is my honor to be chairing this session about science, communication, and outreach, which is one of my passions. For the first talk, I would like to welcome Associate Professor Carol Oliver, who will be talking about astrobiology virtual field trips.
Hello. This session is about science communication, and I'm going to be talking about virtual field trips, but I think science communication actually begins within the university environment. But I actually want to begin with the story over the tea break. I was just chatting with one of my tutors, and she'd seen the virtual field trip, or one of the virtual field trips I'm going to be talking about in my course, because she had to guide students through it. And this was during the pandemic, and her master's was about the Pilbara. But she couldn't go out to the Pilbara because for 2 years we couldn't go out there. But she said when she did get to go, because she had seen the field trip, she went straight out there and was able to identify everything straight away. She saw the context and everything. So it's actually a use that I'm aware of. But to hear it actually, to hear the story of how it impacted her was really a great feeling for me. But anyway, as I say, I'm going to talk about this in the university environment. And university education is really about learning to think. And if you teach undergraduate students or the postgraduate students here who remember when they were undergraduate students, then you're aware of the increasing challenge that we have in getting them to engage with us. And for us to turn out educated citizens who are able to employ evidence-based thinking. Now, move this on is to down here. So we spend a great deal of time trying to prevent and catch cheating. I've given up on it a long time ago, then late last year we had the arrival of AI. It's been around for a while, but ChatGPT made it openly available. And ChatGPT says itself, creativity is a complex and unique trait that is difficult to define and even more difficult to replicate in a machine. And that is what we have to play to when it comes to the university environment. Because I'm going to say something that's quite controversial. I think that a lot of the assessment that we do in the university environment is not fit for purpose. When you think about all the rote learning that we do in the university environment, sometimes that's necessary if that rote learning is then employed later on. But if this is rote learning to just to tick a box that the student at that point in time has learned that fact, it's not going to last longer than 3 months. And actually, another story I can tell you is of a postdoc who told me that she had rote learned her entire undergraduate science degree and felt betrayed by that. So I think we've got a lot of thinking about what assessment means in the university environment. So almost everyone in this room will understand the value of a physical field trip. And there's no doubt, there's no replacement for that. Here at the Burick locality in the Pilbara, WA, where we see the earliest, most convincing evidence of life on Earth, we can see either a student or a researcher out there actually examining the rock. But what if we could use that instance to educate students in the university environment? And actually, that's where my story began way back in 2007, where I was out there in the Pilbara and I thought I want to bring this experience into the classroom. So it's taken me since 2007 to get to this point where I'm going to be talking about today. So The problem with virtual field trips is they can be very expensive to do. So we see things like ThingLink, where you can use commercial software and go out there and upload your data onto an external site to a university, but that's very dangerous to do. For one thing, you could put a lot of work into doing that and you could lose it. For another thing, you've got to protect your student data. But the problem is in making these virtual field trips, it's been very, very expensive, typically somewhere between $100,000 and $500,000 per field trip. And clearly that's not viable for a single university course. But that changed last year here at UNSW, and I know at Arizona State University as well. And I think that other universities will think this through and do the same thing, which is to create a tool, an authoring tool, which academics and students can use to make their own virtual field trips. And then the cost comes down to zero. And the data is protected within the university environment, and you can track the student learning as they do that. But there's an extra problem to deal with. The first attempts, because they were so expensive, were aimed at high school students and public audiences. Why? Because that's what you could get funding for. And here you can see Martin at the Buick locality in the Pilbara under one of these funding schemes. It was actually, I think the funding came from NASA for Arizona State University for a large astrobiology grant, and actually funded this. But there's a problem with this. That's great for high school audiences and for the public. But how can we use that in the university environment? Well, there's a philosophy that the Australian Centre for Astrobiology and Arizona State University developed together out of virtual field trips. And it is education through exploration. It's a great way to learn, and we know it's a great way to learn because that's what we do when we go out into the field. We take students out into the field, and that's how they learn. They're learning through exploration. But now we have this tool. It means that we can simply make these virtual field trips and create them so you can make the experience different to that experience by a high school audience or the public audiences. This is not advancing. Okay, so this is the result of using the tool. It's called Talia, Teaching and Learning Authoring, Teaching and Learning Authoring Tool, Immersive Authoring Tool, that's right. This one here, and I'm just showing you a flat image here at the moment of the Buick locality, that same locality where you saw Martin standing at, and now it's been seeded with hotspots. And in the lower right here, you can see this little icon here, which tells the student where the hotspots are. So as it turns, they know whether that hotspot's behind them or in front of them, because this is from a 360-degree image. And let me go on to the next one. And this is what it looks like. I'm going to show a little video here because I didn't want to go into the virtual field trip. It will take me too long. And you can see now the context of the Buick locality and what the student can explore. And they can pull out those samples and look at them. But you might actually say, well, that isn't, Carol, hang on to you, because then that's still guided. No, it's not. Because, go on. Okay, because the students are told this is the environment of the Pilbara. And my virtual field trip has 10 different sites for the students to examine. And those 10 sites are seeded with a total of 60, 70 samples. And they're told what the story is of the Pilbara. I don't want you to read this teeny weeny writing, but just look at those images of the 4 stages when life appeared in the Pilbara. We're looking at a time 3.48 billion years ago, where over a 13 million year period, a volcano arose from the sea. It started to collapse with the hot spring environments that were created, and then it slowly collapsed into the sea and then disappeared, and life disappeared. The students go through these 10 sites trying to figure out which are the best samples that tell that story. Okay, they do that individually first. They have handwritten field notebooks. And I'll go into why we use handwritten field notebooks in a moment. They use those field notebooks, and then they are in groups of 3. They then interpret their field notebooks to create a story and make a video between them. So here are the field notebooks. Again, I don't need you to read the teeny weeny writing, but the point is this looks like approximately like a field notebook that a researcher might have. And what we found was, as the literature told us, active learning took place as opposed to typing. We got really concerned about students just typing these field notebooks because they were just taking everything and they were just copying from the explanations. But once they had these field notebooks and they had to draw where they were taking samples from, they weren't restricted in any way, but they got much more selective about their samples. And when it came to doing their videos, the marks to the videos went up. So collectively across the class, we'll see that in a moment in the data, from kind of middle of the class around the credit level to up to distinction and high distinction. And the same occurred for the notebook as well. And that we did that made that change 2018, 2019, and those scores have persisted throughout. So let's look at the data. You can see 2018 on the top. On the top left, you can see the samples. They're all over the place. They're collecting a lot of samples. And on the bottom of that, you can see now it's shifted to the left and they're collecting fewer samples. On the right-hand side, in the upper one, you can see what I was describing with those scores being pretty much in the middle of the class. But in 2019, they soared on average towards the high distinction mark. So this has been so successful, we started collaborating with the University of Auckland because we were interested in the hot spring environment, looking at an acid environment and acid sulfate and alkali chloride environment, in particular because of what Martin was talking about earlier, about the digitates, little nodular things on at home plate. And that in these environments, you can see these kind of micro stromatolites as well as El Tatio. And just coming down the line is I've just had a researcher out there collecting images from El Tatio. So we'll be able to see that with the images that we've got from Mars to have another field trip that will follow this one. Get on to the next slide. Go. Okay, so where's the proof in the pudding? So I'm going to play you a video, and it's just under 3 minutes. Just want you to sit back and relax, and watch this video. But it was made by 3 students in my upper level astrobiology course after doing the Pilbara virtual field trip. It's one of the best ones I've ever seen, so I must say it's near the top of the class. But the interesting thing was the students weren't all science students. 2 of them were art students. One was a science student. All 3 are now undertaking higher degree research, high degree research degrees, and they are all in science, and they're all in our school, biological, earth and environmental sciences. And when I asked them about this experience, they said it was the experience that made them think about where they wanted to go with their careers, which was really heartwarming to hear. But anyway, you can see for yourself how going through a virtual field trip, these are third level students, postgraduate students. These three were not postgraduate, they were year 3 students. We've tested this and I hope it works.
Water, fire, air. 3.48 million years ago in the Pilbara, Western Australia, the 4 elements combined together in harmony to enable the development of our life.
Now, only the stromatolite, layered deposits created by microorganisms, remain as concrete evidence of life from this period. These remnants can be used to indicate the physical composition and changing nature of this early environment.
3.48 billion years ago, the region now known as the Dresser Formation existed in the caldera of an ancient volcanic system. The fossilized evidence can still be seen in the localities that curve across the landscape. And after looking at the evidence, we ourselves try to piece together what this environment could have looked like.
The early environment was characterized by hot spring lakes, now preserved as barite-filled hydrothermal veins. These veins provided energy for early microbes, which can still be found fossilized close to the barite deposits. Many sites contain evidence of shallow water preserved in light rivers of sandstone. The lakes were calm and enclosed, such that crystal starbursts were formed from evaporation, which we still have the imprint of today.
The hot spring lakes were fed through a network of underground hydrothermal veins, which circulated the hot water to fuel microbial life. From observing the positions of stromatolites within the site, it is apparent that they grew on the edges of hot water springs and around hydrothermal vents. This can be seen at the geyserite locality. The preserved wrinkled stromatolites are found directly next to geyserite, a rock which indicates the presence of hot springs and is still found on the edge of similar environments in Yellowstone and New Zealand.
However, this calm, shallow water environment did not last forever. The appearance of rounded pebble deposits and fragmented sediments indicates increasing water turbulence, as seen here at the Buick locality. The caldera, in which the sites are located, was slowly collapsing, allowing streams and rivers to flow where lakes once dominated. With this collapse, the water also deepened in places, causing increased sediment deposition, in some instances covering the stromatolites. Since the stromatolites changed the way the sediments accumulated, scientists could conclude that the formations were biological. This changing environment and water depth is also evident in volcanic pillar formation, which require deep water to form and indicate a marine influence.
The volcanic nature of this environment cannot be denied. Particles of volcanic ash and crystals can be found preserved in the layers of stromatolite. Recognizing the physical composition of these ancient environments allows us to compare it with modern volcanic hot spring environments, improving our understanding of the possible conditions faced by early life.
Thank you very much.
Oh. You have 2 audiences. There are people who are going to go on and be geologists and people who are not. I guess the question is, for people who are not going to be geologists, what do you want them to take away from this whole experience?
For students who are not geologists, who are studying astrobiology, the context of getting them to go out to the Pilbara is for them to think about the exploration of Mars.
Yeah, I think about those who are going to become lawyers, they're going to become, going to not be in.
Well, okay, so if you're wanting to be a lawyer and you're doing this virtual field trip, it's getting you to stop and think and explore a scientific environment, and that they can apply that thinking in everyday situations. Yeah, so it doesn't really matter what for astrobiology, my students come from all over the university. They're not going to be, all of them going to be astrobiologists or geologists or even scientists. But the impact on those students is very clear from the feedback that I get, that they have seen education in a way that has not been presented to them before. It also causes them to ask questions as well, which is what I want to do. I want to prompt curiosity in the students that they can apply in other situations, but also appreciate the stories in the rocks. But, you know, I think that that's what fascinated me, that a rock could tell this whole story. And that's what I wanted to convey to the students.
We have time for one more question. Carol, fascinating talk. You gave the example of Italia offering software being used to make much cheaper PhD.
Zero.
Zero, yeah. Could you tell us a bit about what inputs you have to give the AI in order for it to get started?
Okay, so it's that's a great question. Thanks, Simon. It's actually quite as simple as PowerPoint. Okay, so it prompts you, you have the software in front of you, it prompts you to put in the 360, you just drag and drop the 360 in, and then there's another icon that allows you to place hotspots where you want to, and to manipulate those in the environment, and then publish it. Is as simple as that. The one I showed, I think I said it took me about an afternoon to do, just a few hours to replicate the Pilbara into the Italia tool. But anyone can use it. You can give it to anyone and not give them any training whatsoever, and you don't need any technical expertise. And that we could add a lot of bells and whistles to the software, but we have to stay firm on making it so there's no training is needed, because academics don't have time to learn it, and students don't have the patience.
Thank you, Carol, for the great
talk. Okay, thank you.
Next up is Dr. Kobe Calix, right?
Calix.
Who will be talking about bringing plants to space.
All right. So if you can get my...
Is this working? None of these are working right now.
I think people just use the mouse.
We're using the mouse.
The laser. You turn it on.
Let's see how I go with the mouse.
Yeah.
Great. So we have some people on Zoom. I'll try and stick near the mic. Hi, I'm Dr. Kobe Calix. Calix, if you're American, it's a botanical term, so you can ask Google how to pronounce it later if you forget. This is a little bit of a change of scene in that I'm going to be talking about, so I was actually talking with Steve Benner at home, Steve Benner and Carol. I'm going to mention our conversations that I had with you just before about how we define astrobotany versus, for example, exobiology. And what I'm actually going to talk about a bit more is space ethnobotany. So Alice Gorman has done a pretty good job of kind of making space archaeology a thing. So I'm going to start making space ethnobotany a thing, because we are now in this era of plants in space, very much plants from Earth in space. And so that's what I'm going to be talking about. And part of what I wanted to do was kind of almost survey a room of astrobiologists on some of your thoughts on this. And I've actually added a question mark at the end of the talk that I'm doing here for you, because I wanted to make it a question, choosing Australia's first plants in space, question mark, because I want to explore whether or not we're actually making choices or whether things are just kind of happening. So before I move along to the next slide, I'm interested to know if anyone can tell me what plant germinated on the moon in 2019. Or put your hand up if you knew that a plant germinated on the moon in 2019. So that's about I'm going to say that's probably 3% of astrobiologists, which suggests to me that knowledge in the general public is probably lower than that as well. So we're now in this era. So my PhD was in deliberative democracy and science policy. And so I'm interested in what the public knows about what's happening. One of my case studies was synthetic biology for that. And we are in this era now where things are happening, you know, with the Artemis Accords and other missions. And this is what we're in. We need to start grappling with whether we want to make decisions about this as democratic societies or just kind of have them happen as kind of technological assemblages of action. So in 2019, part of the Changi 4, far side of the moon biosphere kind of container thing that they had going on, germinated some cotton seeds. There were a few on there. There was Arabidopsis, what we call canola in Australia, and cotton. And cotton was the first plant to germinate on the moon. That's as of 2019. Which is kind of, so as someone who's interested in ethnobotany, the idea of how humans do things with plants, what we choose to do with plants. All of the plants are Arabidopsis, canola, or some people call it rapeseed oil if you're from northern parts of the world, but I think that has dodgy connotations. So let's stick with canola. I kind of feel like cotton from China is, I'm like, yeah, I can vibe with that because I wear a lot of made in China cotton stuff, really, as much as I try to avoid it. I try to do op shopping and stuff. But to me, this does kind of speak to humans in space, right? This is very much what humans are doing in space. It's not extraterrestrial. This is not exobotany. This is humans making decisions about plants being alive in space. And yeah, there's surprisingly little awareness of what's happening. So this was back in 2019. And more recently, the Australian government has announced the new ARC Centre of Excellence for Plants in Space. That's just been in the last couple of years. They're still ramping up now. They're probably going to be operational by the end of the year. And this is an image generated by this new ARC Centre of Excellence. And so as someone who's kind of looking at space ethnobotany, as I'm terming it, as opposed to astroethnobotany, which some people in the Northern Hemisphere are talking about, I'm interested in the people making decisions about these things and what this says about us as society. So the majority of people, so I've spoken to several of the researchers in this centre, and most of them have a background in plant biotechnology and this kind of vertical farming thing that we can see in this visualization. And this vision of what's happening, very much looking towards the Artemis Accords, humans living on Moon and living on Mars and potentially beyond and having a new lunar gateway and things, you know, is a very particular relationship to plants, and the kind of plants that we've grown on the space station reflects the kind of societies that have been doing, have been existing in space so far. So particularly, you know, the kind of North American and Russian crops, for example. And they haven't, so there's this biotechnological basis for what is happening at the moment. And so if I were just to do a kind of interview, so if I wanted to do kind of a space ethnobotany study of the people who are doing plants in space at the moment, I would have this kind of group of plant scientists who basically pivoted into space, more or less, synthetic biology, and some people who were working with genetically modified crops, which might be more palatable in space than they are to some people in Earth. You know, if you're going to be eating foods affected by radiation on Mars, then if it's, you know, genetically modified to be a bit more tolerant of that, then maybe that's a good thing, as opposed to on Earth where there's all these other dynamics. But then outside of that, there's also some other things that have been happening. So, for example, in the last few years, led by an NGO, this One Giant Leap Foundation in Australia, wattle seeds have gone into space. So this idea of choosing the first Australian plant in space arguably has already been made. Depending on whether you count seeds going into space and coming back down, like we've had a lot of that, we've got the moon trees, there's all various types of seeds that have gone into space since mid last century and then come back down to Earth and grown, right? So we know that a lot of plants can survive space flight and then come back and grow successfully. And yeah, this was a partnership really between JAXA and Australian non-profit. And by the time it did go up, the Australian Space Agency exists now. So there's an Australian Space Agency sticker on there. But does that count? Does that count as astrobotany? If the seeds go back, you know, they go up to the space station, they come back down, they get planted. Should that be kind of should that be the first Australian plant in space? Does it matter? Does it matter which nation states are choosing what plants are going into space? Do nation states matter in space? We have a, you know, moon time zone. I'm sure that some of you astrobiologists do have thoughts about this outside of the kind of narrow framings of the kind of geochemistry and things that you're talking about as well. And there are steps beyond this that could potentially open up a wider sample of people I might interview as astrobotanists, but some of them are children, so that's going to prove harder to do. Might have to wait a few years. But I'm guessing that some of the young people who are involved in this experiment are probably going to, as Carol talked about, end up on a path where this becomes something they do for work. And this is this year, right? So this was just March, a few months ago. And there was an announcement that this NASA associated program of the Fairchild Tropical Botanic Garden was being introduced to Australia. And there's some Victorian school students now who have started this project. They've got these veggie-like modules. Veggie is one of the NASA acronyms for a plant growth module. And this is when we started hearing about bush tucker and Aboriginal traditional foods going to space. And as someone who's got a background kind of in some of this development studies / decolonial studies and things like this, or amongst the sciences, the way this is worded is interesting to me. We've had some comments like this in the last few months where we've got someone from the Royal Botanic Garden in Melbourne talking about the juxtaposition of deep knowledge of tens of thousands of years and modern science, and talking about the benefits of working in partnership. And I've gone, okay, well, based on this list of partners, I can't actually see anything Indigenous led there. So we've got NASA, we've got an American institution, Royal Botanic Gardens Victoria, Royal is there, right? It's colonial institution. Fairchild was a American explorer. Latrobe was a colonial administrator, and the Melbourne Archdiocese of Catholic Schools. So at the moment, it's a Catholic education thing, which to me doesn't scream equity. So as someone who's done some work with Aboriginal communities and Indigenous communities internationally, I've been thinking about what would benefit sharing in astrobotany look like. So we've got various international agreements now about benefit sharing to do with genetic resources. So if we're actually being serious about wanting to use some of these Australian bush foods because, of these knowledge of tens of thousands of years, then we want to make sure that the people who've been transmitting this knowledge for tens of thousands of years are meaningfully involved in the process. And yes, I know that the Royal Botanic Gardens in Melbourne is involving Aboriginal facilitators in the process. And I know that this is works in progress for these various people that are working on these projects. So I definitely don't want to say that, this is not happening. It might be something that's happening slowly in the background. But I guess I wanted to center it amongst this community, for example, because I feel like some of the people in this room might have opinions about it that in democratic societies like in Australia and the US, in theory at least, we might want to have a say on this and kind of advocating for people being involved. So yeah, there's the Convention on Biological Diversity, and then we've got things like the United Nations Declaration on the Rights of Indigenous Peoples, and some of the works that I've done, for example, in Asia, with satellite imagery documenting forest rights and carbon offsetting things, are about some of these rights. And so as we look to these programs for astrobotany, I think we need to be questioning who is doing the astrobotany and whose interests are being represented in that, given that we recognize that the past hasn't been ideal and that the present is also not ideal. I'm not going to go in with this crowd into too much detail on my opinions about this, but I have talked about this with other people who are more on the botanical side of things. There's been a lot of talk about warrigal greens, for example, going into space, but all of the knowledge about that, so warrigal greens is one term for them, but it's also known as New Zealand spinach, right? So, and also throughout the Pacific. So even if we were to shift towards models where, traditional knowledge was better represented or better respected as a partner, it's not like there's one place, right? These are cosmopolitan species in a botanical sense. And I get a bit nerdy about purslane, because in contrast to this kind of biotechnological approach that we were talking to at the start of this presentation, purslane is all over the place. Portulaca is like a Latin term that kind of means portable, edible kitchen food, but it's a weed that you find in the pavement. But it's got omega-3s, you can eat the whole crop. It's a really good candidate for astrobotany, but there's no research into it because there's been no research into it in the kind of agri-biotechnology framing of research that happens, that's the kind of thing that's happening for astrobotany at the moment. But yeah, I chose this Atlas of Living Australia example because, so in some of the work that I've done with different communities, I know that there are names and traditional knowledge about some of these plants. And in this case, Damu and Uwalare is documented on this scientific website, right? So there is also some of this traditional knowledge is embedded in scientific, like Western scientific systems. But I'm really interested to ask you kind of astrobiologists, how you're feeling about this idea of earth plants becoming, you know, elsewhere, and how that relates to exobotany or exobiology in the sense of looking for life elsewhere, because we've already got cottonseed germinating on, we've had it, sorry, it didn't survive like the first lunar night that it had. So it didn't do very well. It germinated and then died. Does anyone know that tardigrades crashed on the moon in 2019? More awareness of the tardigrades crashing than the cottonseed germinating. But I'm interested in this idea of cosmopolitan plants, right? Cosmopolitan plants that travel across shores and that we consider maybe weeds, even though they're entirely edible and nutritious. Are we eventually going to have these plants being cosmopolitan to the lunar gateway, potentially to other planetary bodies? How does that relate to, you know, figuring out where other types of life come from, or, you know, potentially rendering them extinct as we've done all over Earth already? And so the reason I'm framing this as space ethnobotany is because I'm interested in how these kind of human origin stories for how we use these plants and how we're taking these plants into space relate to how things are unfolding in the next 50 years. As we do the lunar gateway and we revisit the moon and we go onto Mars and we start doing potentially horticulture on these other planetary bodies. So I wanted to allow plenty of time for questions because I'm really interested to hear from you about this, because I'm trying to figure out how to frame this research project in a way that is a bit more open than just interviewing a bunch of people who've been plant biotechnologists and then are pivoting into space. But given the theme of this session, I kind of wanted to end on this idea of how does astrobotany relate to the democratization of space? There's a whole bunch of corporations who talk about the democratization of space. But I think that we can go a bit better than just corporations talking about it. Are there ways we can move forward with plants in space that don't replicate the inequities and injustices that continue to shape how we live on Earth, and that don't replicate the kind of agricultural, commercial interests in plants that we're seeing at the moment kind of taking the lead, but not in ways that just appropriate other forms of knowledge with the same actors as well. So I'm going to finish a bit early to allow time for questions, and I really welcome your contributions. I was saying before to Albert that I'm kind of tempted to, so this is being recorded, hello people on Zoom, but I would love to have a bit of a discussion, and so we might potentially cut out some of the discussion from the recording forevermore. If you want to say some things that you don't want to have on the record, we can kind of cut this because you might be able to inform my research direction with your questions. Any questions before I wait for our chat? I see a lot of like eager faces who are really interested, but maybe nobody wants to talk about it.
Can you just define ethnobotany for me a little bit? I'm not quite sure exactly what you're trying to imply by that.
So ethnobotany on Earth is the study of basically how people use plants and why people use the plants that they do. So there's already been about 25 species of plants grown in space on the space station. And most of them are for food, but then there's also things like zinnias, Asteraceae, which are kind of just more for enjoyment of astronauts and because we can. So I'm interested in which species we're growing and why.
Those plants that were picked maybe how the air. There's so many different aspects of living in space And, even to the point where when the Apollo astronauts were going to head into space, they weren't even sure what they could, if they could eat. And so there's usually in a lot of this stuff, there's usually like multiple ways to use a single thing to benefit multiple things.
Yes, definitely.
So I mean, I don't know. I had heard about the cotton thing, but I couldn't find any detail about it. So thank you very much for reminding me that it was. But there is like what has been grown in space so far, and how do we kind of create the popular art? How do we get everybody's opinion to be able to say, well, actually, this would be a really good place to start? I mean, we can always go to the Martian and we can always go potatoes. They are the universal food.
And potatoes have been to space, like a lot of food crops have been to space. But you're right, the NASA Clean Air studies from last century really kind of, we're not food-based, and we're very much about air cleaning. And one of those examples of NASA research that had applications in a lot of other ways as well. But yeah, the majority of crops that are grown at the space station now are kind of quick life. There's chilies and things, so things that are interesting for astronauts to eat. So you can't eat the leaves and stuff so much. But yeah, the majority is like pak choi, kale, lettuce, things like that. But then there's been experiments with microgreens, sunflowers, and there's also been experiments with, there was, I think, an astronaut who reused their underpants to grow like cucumbers and things like that. So various things that will be interesting as we move towards. Exactly. Yeah. So as we move towards kind of whole closed ecosystems in space, how do we use these things more effectively? Yeah.
And if it were kind of creating connections and all of the other. So where do the plants fit in the whole?
That's such a profound question. We could say the same thing about Earth, right? So the Changi lunar biosphere experiment, in which the cottonseed germinated, also had Arabidopsis, canola, and also fruit flies, and none of those did anything. So there was an attempt to kind of make it sort of closed ecosystem that didn't work. And yeah, at the moment, most of the plants on the space station have their own kind of enclosed. So on LADA, there was a kind of a greenhouse, and then there's various modules of greenhouses, and also the biological research in canisters that have been happening on the International Space Station. So they're kind of contained. So rather than being cleaning the air focused at the moment, it is very much these experiments looking at the impact of space on plant growth and little enclosed systems, so that then they can be studied most effectively. And the Growing Beyond Earth one that the students are working with is very much that model of, here's a contained little system that you can measure all of the inputs and outputs. So that's still where we're at, as opposed to kind of the art of supports working towards these systems that are actually supporting potential communities elsewhere. I feel like 1, 2, 3. Can we go that order at the back and then forwards? Just because you're, privileged place at the front of it. Go. I forgive you already. Oh, and that's only just 20 minutes now. And I think we have a speaker who's left out, so we could have these 3 and then save more to the end. Yeah, well, I mean, we're already terraforming in the sense that MOXIE is working on Mars right now, right? The Mars in situ resource utilization experiment. I missed the oxygen part in there, which is part of the Perseverance mission. which has been working 7 or 8 times now. So it's interesting that we have a machine, we have a robot doing what plants do on Mars now. We know that we can, right? So I kind of like to remind people that that is still terraforming. It's not earth mining. It's not planetary body mining, it's atmosphere changing. But we're already doing that experimental technology as of 2021 as well. So I think it's interesting in the context of as well what's happening on Earth with robots. That sense that we're using robots to do something that plants do on Mars because the robot can take it, whereas the plants couldn't take it. But are we working towards the stage where we do have these things on Mars? I could do a whole separate presentation on the research about fungi as a building material for Mars that NASA also funds. But I feel like fungus is a step too far for today. But yeah, I think that, that's kind of what I'm interested in. As the majority of people in this room, I think research, life from elsewhere or how life could develop elsewhere, as opposed to what's going to happen as we contaminate / grow things on other planetary bodies. Yeah, how do you feel about terraforming as a thing? When a lot of these plants that are designed to stabilize sand dunes, for example, it's interesting to me that Fairchild is a tropical garden, but in Australia, a lot of the things we've been looking at are kind of coastal. Things that might do better in, regular and things like that. Are we ultimately going to get to the stage where the moon dust is stabilized by some of these plants? A green moon. Doesn't really answer your question, it just kind of throws a whole bunch more stuff in there tangentially.
I think we'll wrap up with Martin's question.
Oh, but I had a question. He hasn't really got to speak today.
Oh, okay. I was reflecting on this question. Are there certain things you wouldn't want to take? Like tobacco or is there any plant that we grow on Earth that, obviously we wouldn't want to replicate? Is there a list of some sort?
There's definitely, I feel like there's definitely not plants there. There's not yet. There is a list. Okay, so actually Carol touched on ChatGPT. I think it's really interesting. So I asked ChatGPT, because this is related to my teaching and stuff, to generate a list of the plants that have already been experimented, that have already been into space. And it came up with about 25, which is around about right. And I was like, okay, now can you divide it into plants that are edible and plants that are inedible? And ChatGPT was like, okay, well, here's these ones that are edible. And I'm like, not too bad. And it's like, these ones are inedible. And it was like celery, amongst other things. There's a lot of people actually who agree with ChatGPT on that. And it was really quick to get to ChatGPT being wrong about what is edible, because it clearly doesn't have a good grasp of what is edible. And it was like, no, but the research was just an experiment. People didn't eat them. And I basically got into an argument with the AI about what was edible based on the list. So I'm like, no, sunflowers are edible and celery is edible. Put them on the other list. And it just kept getting the list more and more wrong. And it was very confused. Yeah. So I think this is a really interesting area, actually. If you want to maintain a human job, then looking at how humans use plants is something that the AI finds quite confusing. It doesn't really answer your question, but the answer is no, there's not a list. There's things like death cap mushrooms that I would suggest we don't take. But in terms of plants, things as well, like poison ivy, maybe we don't want to take. But maybe that's got really good, formaldehyde scrubbing properties and things, so maybe it's got redeeming qualities.
You want to get subsidies to certain crops, for whatever reason, you have to grow them in space one day. And you're buying for, cash crops versus food crops, something like that.
Yeah, well, I feel like cash crops are probably not, economics is a pretty earthbound system at this stage. I hope that's going to remain the case for a while. So I hope they're going to be other kinds of crops and plants. But yeah, there's definitely been a trend towards the ones that are already the subject of a lot of scientific experimentation. And what I consider to be like people pivoting from just straight up plant biotechnology to plant biotechnology in space.
Yeah.
Thank you, Dr. Coley.
And since our last speaker could not be here because of a visa rejection, Juan will be our last speaker for today, and he will be talking about training students into research.
Is it good to? Yeah, cool.
Cool. So hi everyone, it's me again. As Jamie said, I'm Juan. I think I'm gonna introduce myself properly in this one. So I'm a final year PhD student here at UNSW in the School of Chemistry. And my research basically focuses on using computational chemistry to do molecular spectroscopy and apply that molecular spectroscopy to find to like astrobiology or astrochemistry related topics. It's a really fascinating topic. Pretty sure most of you would agree with that. And it's a great opportunity to involve senior high school students in looking for aliens in space. So we do this within the SciEx summer school here at UNSW, which is a week-long research integrated learning experience for senior high school students where PhD students like myself work as mentors in small groups for the students. And I want to emphasize here on the, on the PhD students as mentors, because the fact that we have PhD students as mentors means we're sort of like more approachable role models than if you remember that we're talking about senior high school students, so probably year 11 and year 12. So yeah, we're just more approachable than a senior academic or something like that. Just to give you a little bit of an overview of what SciEx does, as I said, well, students come to learn about research and explore their own hypotheses. So they actually pursue a research project in that week in a supported environment. So it happens in late January, so it is summer here, which is great. The students get exposed to what research and university life is like here at UNSW. As I said, they get support from the mentors, which are PhD students, and the academics behind the projects as well. And we get to go into a lot of lab tours and things like that. So just to showcase their research facilities that the university has. It is a very EDI-friendly approach. So just in 2023, there were 40% fee waivers and 60% of female enrollment in the program. So I feel like I've been talking a lot about Laura today, which is a bit weird, but yeah, Laura is the main SciEx coordinator, and she will have a poster tomorrow if you want to know more about SciEx as a whole, as in like the whole school, summer school. So if you have any questions about SciEx itself, I do recommend going to Laura's poster. There are lots of projects that the students can choose from. So as in this year, we had 19 different projects. You can see that change from like air quality to genetics, materials and stuff. But particularly I focus, the one that I focus on is on this one. So this is my project, or the project that we run, quantum chemistry and spectroscopy. And in this project, we basically talk about aliens. Well, I mean, not quite. We do more stuff, but aliens is definitely the most popular like project choice here. And so basically, the main idea with the astrobiology part of the quantum chemistry and spectroscopy project is basically how molecules absorb light and how can we relate that absorption of light from molecules with signals from light. So yeah, we just show them things like this where they can see like different vibrations and stuff from the molecules, and basically just start a discussion on whether those molecules could be related to life or not. And yeah, so we use computational chemistry as a tool to do this sort of like research. So yep, we introduced in the project, we introduced key concepts in quantum chemistry and spectroscopy, exploring its applications in different research fields. So one of those is fundamental chemistry. So some of the students want to investigate, I don't know, things like how does the bond in a molecule changes when you change different atoms. Another field is infrared spectroscopy in chemistry. So how can you follow chemical reactions using infrared spectroscopy? There's also a bit on climate change and global warming. So some students will want to investigate what's the global impact of releasing certain kind of molecules into the atmosphere. So calculating the global potential, the global warming potentials and things like that. And of course, astrochemistry and astrobiology and aliens is another one of the areas that we that the students choose to perform their projects. So yeah, as I said, it is a week-long project, so we need to organize it so that we can deliver the most. Yeah, so that the students can get the most out of it. So I'm just going to show you how we align the different goals that we have for the school with the sessions in the week. So the first goal that we have is we want students to learn the basics of quantum chemistry and basically get something exciting that they can just go home and show to their parents. So you can see that on Monday, well, Monday morning is basically like an intro session where we get to know each other and stuff. And then on Monday afternoon, we get a hands-on activity on quantum chemistry where we actually get them to calculate a spectra. And then on Tuesday morning, we actually introduce the concepts of quantum chemistry. And so it might be a little bit weird at first, like, so why are we doing the practical first and then explaining what they did the day before? But the thing is that we found that doing this is actually more engaging for the student. So the fact that the students can get this spectrum, just take a comment, like start discussing, makes it easier for them to later understand the concept of quantum chemistry. We want them to take part in the research project design process. So there's a session on project planning where basically students get to formulate their own hypotheses and how are they going to tackle the research project over the course of the week. We want them to learn Python, or basically just to get more confident and less scared about Python. So we have a whole session in Python here. And then at the end, they have, we wanted to pursue their projects, because remember that this is a research integrated experience. So they actually choose a project and then they just get to do it. So they have the rest of the week with the support from us to just pursue with the project. And then on Friday, they also do like a mini presentation to their peers. And here we mix them. So like, remember we have like 90 different projects. So when they do these presentations, we actually get them to go to students from different groups that different than the one they were in. We also introduced this year particularly something called science interludes, where basically even though we were doing just quantum chemistry and spectroscopy, we want them to get a gist of basically what other stuff is happening in research. So we got all the PhD students from the school to come and talk about some interesting stuff, so like catalysis, water purification, climate change, drug development, well, so on and so forth. So the students actually enjoyed this a lot. Now with Python, so as I said, that's one of the main components of the research pool, is just to get them more confident when using Python. So we do that through Jupyter notebooks. Yeah, through Jupyter notebooks. These Jupyter notebooks have some pre-written functions for the data analysis and visualization, so that they don't have to start from scratch when they want to do, pursue like the processing of the data. It is extremely well commented throughout, so that the students know exactly what's happening in each part of the code, but at the same time, they don't have to read it to make the code work. So it's just, if the student is interested in learning more about Python, then there are the comments for them to know more about that. But with that, we also provide links to additional resources here within the School of Chemistry for them to enhance or maximize their Python skills. And it's just an excellent starting point to get them into Python, honestly. Yeah, it's just because of how user-friendly the notebook is, the students just get less scared about it. Well, what about like aliens and Python? So Jupyter notebooks offer a great opportunity to showcase how spectroscopy can be used to find aliens in space. And so one of the notebooks that we provide to the students basically is useful for them to understand and visualize the different effects that affect the spectra of molecules in space, demonstrate gaseous mixtures, and at the end they get to apply the concepts that they have learned into like a mini task of the Jupyter Notebook. So this is actually part of the Jupyter Notebook, so they can go and just click and see how the spectrum changes when they choose different molecules, which is a fundamental concept in vibrational spectroscopy. So it's just a great way for the students to visualize that. And then this is probably one of the most exciting parts for them, because once they're finished with the notebooks, we sort of have this section where we said this whole narrative of, oh yeah, imagine that you just got data from this exoplanet and there are like signals for, I don't know, 3 different molecules that could be related to life. How can you identify which molecules are the ones that are in the atmosphere of that planet? So basically what the students have to do is just change here, choose different molecules and try to match the spectrum from those molecules with the unknown mixture or the spectrum from an exoplanet. And the students just love it. The confidence in Python actually significantly, well, it increases by the end of the summer school. And they recognize its value in research. So this is just data from this year's summer school. Basically, we asked them, how confident do you feel prior to summer school with Python, and how confident do you feel now with Python? So 0 is no confident at all, and 10 is like super confident. And we can see that by the end of the summer school, there's like a 2-point increase in the confidence that the students have in Python, which is really good. We also asked the students what's their favorite, least favorite part of the project. So those are just quotes, but basically what they're saying is they really enjoy being part of the research project, making friends and sort of like the networking aspect of it. Learning from the PhD students, literally just talking to us, asking questions and things like that. They seem to enjoy that a lot. Networking, and then the Python bit. As you can, I mean, this is just like evidence from the previous slides, right? So like the students actually see the value of learning Python. In terms of the least favorite part, because they have to decide on, so we only have 1 week. Some students find that overwhelming to think of a research idea or like a research project in the week. So they just that process of trying to decide what to do is a bit challenging for them. This one is funny because, of course, this is a computational based project, but we have different, there are over 19 projects in the school, right? In the whole summer school. And some of these projects, they take them into labs and things like that. So they have friends, right? And they talk to those friends and they're like, oh yeah, we went to this lab and stuff. And so they think that we don't have any like practical sessions, but there's a problem here in like the message that we're conveying, because our practical sessions are the computers themselves, like doing the stuff on the computers. So this one was an interesting one to see. Just stuff related with the lectures and stuff. I guess this is very specific. And then the presenting bit of it. Some students just were really scared of presenting, as anyone could be, which is okay. And then, and so we also asked them what advice would they give to future science students if they had the opportunity. You can see that this one relates pretty much with the first least favorite part before, and is that they recommend that students do some prior like research so that they have an idea of what they want to do during the summer school. Just get good at Python and do the coding beforehand. But at the end of the day, they get it, so it's fine. This is really interesting. They seem to just have enjoyed it, like the whole summer school. They just seem to be very like motivational to other students, which is great. And some of the students got lost during the summer school. So just doing the campus tour earlier. Just because I'm running a little bit out of time, I'll be done soon. Some things to improve for future for SciEx, and in case you are thinking of sort of developing a similar project, is we need to find a better way of delivering the program's content beforehand. And this is pretty much related to that bit of where the students thought that because we didn't go into labs, we didn't have like any practical sessions. So we need to convey that message better. Remove some heavy theory that is actually not needed. So like, yeah. Make sure that the students have the Python and all the libraries and stuff installed beforehand. Believe me, it was really challenging to do it on the fly. We lost a lot of time, but yeah, you live and you learn. Yeah, like with the science interludes, just trying to combine them all together. And then ensure that the students are comfortable and relaxed for those presentations, because the students are not marked during the summer school, but they still get really anxious about the presentation, and it shouldn't be the case. Just to finalize, this is the quantum chemistry and spectroscopy cohort for SciEx. I'm wearing the same shirt. I've been a SciEx mentor for the last 3 years, so I started back in 2021, but 2023 was the first time we got to do it in person because of the pandemic. And it was funny because engagement in person is better, but at the same time more difficult. People like talking, and I wasn't prepared for that. But it's really cool because we got some homegrown avocados. One of the students just had an avocado tree and brought like 10 avocados for us. But yeah, thank you.
Clare? Good talk, thank you. Some of the issues that you've mentioned with either things that students didn't enjoy or the things that science needs to do better, in particular working out how to like come up with your own project or just installing a bit of software before you start. Those are issues that we face teaching undergrads here at uni. Like, where in the educational pipeline do you teach people those skills? Because they don't have it when they come to uni. They clearly don't have it in like year 10, 11, 12 either. When do we need to start teaching kids how to do things on their own?
Yeah.
They're not slacking.
I mean, that's a great question. Yeah, I don't know. That's my answer. I think definitely potentially projects like this one would motivate that from an early stage. But yeah, I don't know.
I can't get my students to download Google Earth in like a week's morning. So
yeah.
I don't know.
Yeah. That was, as I said, it was really challenging. Super challenging, yes.
Albert? Yeah, have you ever followed up with some of your students about where they end up? Like, what is their major, for example?
Yeah, that's I think Laura has a better answer for that, because it is really hard to track students once they finish the summer school. But we can talk from example that from SciEx 2021, we actually have one undergraduate student joining our research group this year. So I guess some of them do end up doing computational chemistry and a spectroscopy. But yeah, in the great scheme of things, I don't really know.
I guess these students are, you said there's no formal assessment, so they're all very self-motivated. Like,
yes, yeah. So students come to SciEx because they want to. Yeah.
Okay.
Yeah, no, that's great. We actually do. So this yeah, I'm not gonna go back, but in one of the sessions we have some time where we give them at least the advice we know on how to present. So we do like train them, I guess, I don't know if that's the right term, on how to present their research, especially because because they're presenting to people that are outside the research projects. So they're gonna, it's pretty much like giving like a general audience lecture on their findings. So we take them to, try to minimize the jargon, because it's really interesting, right? Like, because once you start learning or understanding what you're doing, you feel like everyone does as well. So like with high school students, they tend to just like use all the jargon that they can. But then in these sessions, we actually tell them like, hey, nobody is gonna understand that. So, but yeah, we do have the training with them beforehand. I think it's just, I don't know, the fact that they're also in high school is probably more like scary for them, and that they don't know anyone, like all the students that are in the summer school as well. But yeah, and long story short, we do have like a training session on how to present. Yes.
Maybe that sort of thing can be done more informal. We've had examples where when you sit around in a group, you actually just discuss and the meeting starts. Or then quite happy to chime in and say, oh, I have this experience, or I have that, so that it's not necessarily, I mean, I don't know what you're trying to get out of this for the student. But if it's just really sharing what they've done, it'd be better to have that more as a group process. Because like I said, everything in high school is mortifying. I mean, I get in third year, my students just get mortified by giving presentations. They have to do it every year.
Yeah.
So it might be a way of enhancing them into that, where it's more group discussion, and maybe think about having talking points for each of them, saying, what was your methodology, what did you have a problem with your methodology, so that it's more a relatable fact, stand up.
Yeah, I think that's great advice. When you look at the schedule for the students, like each year for the summer school, there's this box on Friday that says presentations, and I think probably we should have started there, just like changing that naming from presentations to sort of like discussion or something like that. Like that would probably make it easier for the students.
Yeah, or a workshop, something that's more inclusive.
Yeah, no, I think that's great feedback. Yeah, thank you.
Yeah, I was just going to follow up on that. I guess you've got a good number of people in your team. I don't know how many students you have, but I've done similar things where you have one of the mentors is sort of they're coaching a group who are going to present together, so that they've got someone they can bounce ideas off of. You can get that positive feedback and say, hey, that's a good idea. I don't know if that's something you guys do as part of it or not, but if not, I don't know. It's a way to get the confidence up and get the jitters down.
No, that's also great feedback. I think we just need to like see better how we can deliver those, because it's also the problem also is that we have such a short amount of time. It's only a week, so we want to teach them all that we can, get them confident with the skills and also confident presenting. So it's probably an overkill to try to accomplish all of that in a week. But yeah, I think just having that mentorship more like one-on-one would be beneficial as well. Yeah, thank you.
All right, I think we'll wrap up there.
Thank you.
And for those who signed up for the optional dinner, that would be happening at 6:30.
Yeah, I'll make an announcement. Yeah. So thanks everybody. That wraps up day one. It was a lot of fun so far. I had a lot of fun. And for anybody that's going to the dinner, you basically have like an hour and 40 minutes. You should have got an email from Anna Wake just very recently to give you more information. So you can use that time to go back to your hotel room, freshen up, do what you got to do, and then come back to campus. The roundhouse is just down the steps. It's on campus, very close. And you're also welcome to stay here as well. But you have a bunch of free time.
The roundhouse is open. You can have drinks down there as well.
Right, I mean, it's a regular bar, so like you just want to hang out there, but there is going to be an open bar at the dinner, so you can pace yourself. I would recommend it, like, against doing that. If you really want to, you can leave stuff in my office if you, yeah. All right, thanks a lot. I'll see you guys tomorrow where we have another excellent lineup. Thanks a lot. And a round of applause for all the speakers again.
I'm not having trash.